Hi everyone. I hope I'm perfectly audible and visible to everybody. Hey students. Hi everyone. I hope I'm perfectly audible and visible to all of you. Hi. Am I? Students, am I perfectly audible and visible to all of you? Wait. Okay, so welcome everybody. Welcome to the session. Welcome to Unacademy NEET English. Today it is the class 12th zoology or not zoology, not botany. It is the biology marathon. So we are going to talk about the chapters which we were not able to complete in the last lecture. So today we are going to start with the genetics. This is what I'm planning. You tell me, what do you want? Yes, you tell me, what do you want? I have planned that let's start with the molecular, let's go for the biotech, then let's go for the ecology. Rest, whatever my students will say, I'll follow that. I'll follow that. So you tell me what you guys want. Which chapter you want me to start next? You want me to start first? MBOI. MBOI. Okay, guys. So let's get started because we are going to target the most important chapters first, MBOI, and then we will go for the biotechnology. Okay, then we will go for the biotechnology. So let's get started. Let's get started. So you tell me in the chat section, are you guys excited for the session? Yes. Are you people excited for the session? Not MB, it's MB. Molecular basis of inheritance. MBOI. Some students are saying MB. MBA, अरे नहीं यार, why MBA? It's MBOI. Okay, so let's get started, people. So there is one more good news for all of you. I hope you guys have seen that, right? We have already completed our class 11th marathons, right, students? So what am I going to do this? There will be complete class, right? There will be complete class 11th revision, right, for the biology. And here what will be confirmed? 180 marks are confirmed for sure. Yes, bache, and in this marathon it is not going to be for 10 hours, it is not going to be for 20 hours or sorry, not 20 hours, 10 hours or 12 hours. No, here in this particular marathon, in a very concise manner, we are going to revise the entire NCERT. We will not touch anything beyond NCERT. Here in this particular marathon, you will get line to line NCERT practice. Yes, you will get line to line NCERT practice. Tell me, how's the idea? Yes, voice is low. Evolution, okay, we'll pick up the evolution as well. Even I was thinking the same. Right. Literally, I was thinking the same, that from so long we didn't revise the evolution. So we'll do it. And if not now, if not in the starting, I'll pick it up in the short video revision session. What say? Now, is the video clear? Human health and disease, so we have completed, Simran, in the last class. Okay, we'll touch the evolution as well. Happy? Now the voice is fine. Voice is fine. Voice is fine. Respiration we have discussed. Yes, voice is low. Still low. I think it's fine now. Okay, what about voice? Tell me about the voice. Is it fine or not? Okay, so let's start the genetics. Okay, so do not forget to hit the like button. Do not forget to click on the notify me, because this session is going to be very important. And here, as I said, I'll be picking up the most important lines from the NCERT biology. So line to line practice is not going to be there. Okay, line to line practice is going to be there. Okay, so let's get started. So here we are. We will start, and if you are asking why not 180 plus, yeah, 180 marks, so don't worry, they can come. But the point is, majority of the questions, if you will see, they are from class 11th, right? In your exam, if you see the majority of questions, they are from class 11th, right? Even they say that 60% is 11th, 40% is 12th. But yeah, it is not a fixed trend. But as per previous year paper, it is like that only. It is like that only. So let's start. Let's start with MBOI. Ready? Let's start with MBOI. Ready? Ready? Are you ready? Tell me. So we will start with molecular basis of inheritance, right, students? And then let's go for the biotech. Let's pick principles of inheritance. Let's pick evolution, and then let's pick up the ecology as well. So don't worry, don't worry, don't worry. Otherwise, right, whatever is required, I'm still putting the short videos for the quick revision. Do watch them, right? They are going to be very amazing for all of you. And from the evolution also, I'll make a short video with all the important topics of evolution. Don't worry. Okay, so let's get started. Let's start with the molecular basis of inheritance. And when we talk about the molecular basis of inheritance, the very first thing we need to understand is nucleic acid. Yes, I'm starting from the scratch. Don't worry about that. If your genetics is weak, this session is going to be helpful for all of you. So we are going to start with the nucleic acid. When we are saying nucleic acid, they are the polymers. They are the polymers of nucleotides. What are they, people? They are the polymer of nucleotides. Right, so polymer of nucleotide means they are made up of many nucleotides. So that is why we also say that when you talk about the nucleic acid, they are polynucleotide chains. What are they? They are polynucleotide chains. And here, students, we talk about RNA as well as DNA. This is what we have to discuss here. RNA as well as DNA. So when we discuss the first thing here, the first thing that you need to keep in your mind is the scientist name, that is your Friedrich Mischer. Right, which name you need to remember? Friedrich Mischer, F. Mischer or Friedrich Mischer. He was the one who discovered nucleic acid. He was the one who discovered nucleic acid in pus cell. Right, he was the one who discovered nucleic acid in pus cell, right, in pus cell, okay, in pus cell's nucleus, whatever you want to say. So because he discovered it in nucleus, so he called it as nuclein. So initially he called it as nuclein. So initially it was not the nucleic acid. Right, from the starting itself, it was not the nucleic acid. He started with what? He was like, he discovered the nucleic acid in pus cells, in nucleus, so he called it as nuclein. Is that clear? He called it as nuclein. Is that clear? Is that clear? So term nucleic acid was given by Altman then. Term nucleic acid was given by Altman. Right, he was the one who named it nucleic acid after, of course, after checking its acidic properties. Is that clear, everybody here in the class? Is that clear? Yeah, the class is going to be from the scratch only. So as I said, nucleic acids, they are the polymers of nucleotide. So when we talk about the nucleotides, what we need to see in the nucleotides? We need to talk about the nitrogenous base. We need to talk about the sugars. We need to talk about the phosphate, my dear students. This is what we need to discuss. So firstly, we will start with the nitrogenous bases. What are we going to start? We are going to start with nitrogenous bases. So as the word is saying, my dear people, as the word is saying, it is the nitrogenous base. What is it? What is it? Nitrogenous base. So it means it contains nitrogen. Am I right? It means it contains nitrogen. Am I right? Yes, everybody, do let me know in the chat section. Do let me know in the chat section. Yes? Yes? So nitrogenous base, we have two words here. It is a common topic of biomolecules as well. So we have purines and pyrimidines here. We have purines and pyrimidines here. So when it is purine, the trick here is puga. When it is pyrimidine, the trick here is cut. So what is the meaning of PUGA here, my dear students? G stands for guanine, A stands for adenine. That's the meaning here. G stands for guanine, A stands for adenine. Guanine, adenine, guanine, adenine. Look at the spellings here. 9 is there in the spelling. 9 is there in the spelling. So do remember it's a 9-membered ring. Do remember it's a 9-membered ring. Are you getting my point? What kind of ring it is? It's a 9-membered ring. Then it is pyrimidine. So when it is pyrimidine, you have C for cytosine, U for uracil, T for thymine. That is what we have here, my dear people. And it is a 6-membered ring. What type of ring it is? It is a 6-membered ring. Am I clear? Now, when you are talking about the 9-membered ring here, purines and pyrimidines, so remember at 1, 3, 7 and 9, at this position, nitrogen is present in the ring. At this position, nitrogen is present in the ring. At this position, nitrogen is present in the ring. And here at first and sixth position, N is present. Nitrogen is present. Are you getting my point? Sorry, at first and third position, nitrogen is present. So 1, 3, 7, 9 and 1, 3. Is that clear? 1, 3, 7, 9 and 1, 3. Is that clear? So is it important? It is very, very, very important. It is very, very, very important. I was directly about to start with the DNA structure, right, and the packaging of DNA helix, but I believe many students, they struggle in this part. So that is why, bache, I am discussing here. I'm discussing it here with you all, right? So when you talk about the what? When you people talk about the pyrimidines, so what you guys will see in the pyrimidines, first of all, this is the ring that we have. Okay, first of all, this is the ring that we have. Are you getting my point? So as I said, 1, 3, 7, 9, right? The position here is very important. 1, 3, 7, 9. So I'll give you just one example, and I will give you the example of what? I'll give you the example of cytosine. I'll give you the example of cytosine. I think I have discussed it multiple times with all of you. Right, bache? With all of you. So I believe you remember that one alternate double and single bond is single bond and double bond is there. Is that clear? Is that clear? Yes, bache. So this is the structure of what? This is the structure of cytosine. So you people can clearly see that it is a 6-membered ring. 1, 2, 3, 4, 5 and 6. Are you getting my point? So we have cytosine here, we have uracil here, we have thymine here. So what we need to remember, that thymine is also known as 5-methyl uracil. Means the structure of uracil and thymine is same, right? It's just that in the thymine, at 5th position in the ring, methyl group is there, CH3 is there. That is why it is known as 5-methyl uracil. So it is our previous year question. Which question it is? It is our previous year question. Am I clear here? Am I clear here? Then what do we have? Purines. It's a 9-membered ring. It is a 9-membered ring. So that 6-membered pyrimidine is also there. Plus imidazole ring is also there. Plus imidazole ring is also there. So here we have one pyrimidine ring. We have one pyrimidine ring. One pyrimidine ring. So when I'm saying one pyrimidine ring, so it is a 6-membered ring, right? So 4 carbon plus 2 nitrogen are there. This is what your purine is. And trust me, that is a PYQ as well. That is a PYQ as well. So we have one pyrimidine ring here, my dear students. One pyrimidine ring here, my dear students. Along with that, we have one imidazole ring. We have one imidazole ring. What do we have, people? We have one imidazole ring. So when I'm saying imidazole ring, I have 2N plus I have 3 carbon. So it's a 5-membered ring. It's a 5-membered ring. Here 2N, 4 carbons are there. Here 2N, 3 carbons are there. So it is a 5-membered ring. So you guys can take the example of your. So 1, 2, 3. Yes. So that is how the ring is going to be. That is how the ring is going to be. And here double bond, this is a very simple structure. Single bond, then again the double bond, then again the single bond, then again the double bond is there, and here also we have the double bond. Right, here also we have the double bond. So it is your NH. So basically, if I have to name it, so it's 1, it's 2, it's 1, it's 2, it's 3, it's 4, 5th, 6th, then 7th, 8th, and 9th. So this is the 9th nitrogen, 7th nitrogen, and here at 6th nitrogen, here at 6th nitrogen, at 6th position here, at 6th carbon here, the NH2 group is attached. So here I am talking about adenine, and if you people will see the guanine structure at the 6th position, double bond, O is there, and second carbon will have NH2. These are the differences. So if you think of pyrimidine, 6-membered ring, if you think of purines, 9-membered ring, it is going to be, so at 1 and 3, at 1 and 3, at 7, 9, you have nitrogen, 1 and 3, you have nitrogen. Any doubt here? Any doubt here? Now the next thing is sugar. So when I'm saying sugar, I am talking about the pentose sugar here. So when I'm saying pentose sugar, so it is C5H. So now how can I check it? Do remember, it is a monosaccharide. So this is a general formula that we have. CnH2nOn. This is the general formula that we have. CnH2nOn. So N is equals to 5 here, right? So it is a pentose sugar, and here we talk about two examples, my dear students. One is ribose, another is deoxyribose. One is ribose, another is deoxyribose. Okay, one is ribose, another is deoxyribose. So C5. So here it will be C5H10O5. Okay, C5H10O5. Again, a previous year question it is. Right? They even ask the question from the basic structure of these monosaccharides. Just keep it in your mind. Okay, just keep it in your mind. So this is what we have. Right, now, bache, firstly see the structure, then I'll come to deoxyribose. See, it's like this. So it is your first carbon. It is your second, it is your third, it is your fourth. Okay, so this is the first carbon. Let's say this is the alpha HOH, HOH, H, and here it will be CH2OH. Just look at the structure. Just look at the structure, everybody here. Just look at the structure, everybody here. So what do I have here? See, here, this is the first carbon. Hydrogen is below. So it is the beta ribose that I'm talking about here. Which ribose? Beta. So what am I saying? Hydrogen below, beta at 6,1. Hydrogen above, it will be above, A for above, A for alpha. Right? So this is the ribose. Now when you look at the deoxyribose, now when you look at the deoxyribose, so look at this. So look at this. This is what we have, right? This is what we have. So if you will see here at second carbon, because this is your first carbon, this is second, this is third, fourth and fifth. And here beta, it is your first carbon, it is your second carbon, it is your third carbon, it is your fourth carbon, and it is your fifth carbon. Okay. So when you talk about the second carbon here, so you will see oxygen is absent. And yes, you have to be specific. You have to compare the second carbon here. What do you have to compare? What do you have to compare, everybody here in the class? What do you have to compare? You have to compare the, right? You have to compare the second carbon here. Am I clear? Am I clear? What do we need to compare? We need to compare the second carbon here. So here in deoxyribose, there is one less sugar in comparison to the ribose. So the formula will be C5H10O4. So it is an exception, right? It doesn't follow the deoxyribose. It doesn't follow the general formula of monosaccharides. Okay, deoxyribose, it doesn't follow the general formula of monosaccharides. Just keep it in your mind. Now next is phosphoric acid. Yes, it is your phosphoric acid. That is what you have to study here. Phosphoric acid, ick, nucleic acid, phosphoric acid. That's the way to remember that. H3PO4. So it is going to be like this. And when we say phosphate, we are talking about the ionic form here. Is that clear? So because it is an acid, it is giving that negative charge to the DNA. Okay. Now the next thing is, now these molecules, all of these biomacromolecules, all of these biomolecules, they need to join. So when you talk about the nitrogenous base and sugar, when they join with each other, my dear people, you will get nucleoside. You will get nucleoside. When your nitrogenous base, sugar and phosphate, when they join, you will get your nucleotide. That is also a PYQ. For an example, N. N stands for nitrogenous base. S. S stands for sugar. N stands for nitrogenous base. S stands for sugar. Here, nucleotide it is T. Three things are there, beta. So nitrogenous base, sugar and phosphate is there. Nitrogenous base, sugar and phosphate is there. And here you have nitrogenous base and sugar. This is something we need to keep in our mind. Now, sugar is sweet. So everybody is like, I want to join with sugar. Right? I want to join with sugar. Sugar is sweet. So everybody is like that, I want to join with sugar. So this is the formula that, this is the structure that we have just formed. Let's suppose that is the scenario. Let's suppose that is the scenario. Just imagine. Now, this first carbon here, my dear people, it will, the OH here, it is going to form bond with the nitrogenous base. Right? It is the one that will form bond with the nitrogenous base. Are you getting my point? Yes, everybody? Yes? Yes, everybody? Am I clear here? Am I clear here? So let's suppose if I have to make a structure just to show you this, 1, 3, this, okay, this, right? So here, in the case of purine, so this ninth nitrogen, it is having NH. So here, my dear people, the bond formation will be there, right? So when it is the purine, your ninth nitrogen and sugar's first carbon, they will form bond. That bond is your N-glycosidic bond. Okay, so dehydration is there in this bond. Okay, this is your nitrogen of the ring. This is your carbon. It is having the OH. Water will go out. You will have N-glycosidic bond. What do you have here? You will have N-glycosidic bond. This is the first thing that you have to remember. Right? This is the first thing that you need to remember. Anybody having any doubt? Yes. Anybody having any doubt? So once, just see, this H2O will go out. What will you get after that? Of course, after that, it is going to be like this. It is going to be like this. So when your sugar, it joins with the nitrogenous base, the structure is known as nucleoside. Okay, so here in nucleoside, which bond is linking them? It is the N-glycosidic bond. Which bond is linking them? It is the N-glycosidic bond. Keep it in your mind. Keep it in your mind. Now, here itself, you have this P double bond O, OH, OH and OH. So when you look at this part, right? When you look at this part, yes, my dear students, here what is happening, beta? Here also your water will go out. Here also your water will go out. So the bond will be like this, right? So the bond will be like this, right? The bond will be like this. So which bond is there? It is phosphoester bond. This is phosphoester bond. So you can keep it in your mind like this, that this is our sugar. Sugar is sweet. Everybody is interested in having relation with the sweet people, right? So sugar is sweet and everybody is interested in having relation with what? With sweet people. So that is happening here. So in between sugar and nitrogenous base, you have N-glycosidic bond, and in a nucleotide, because it is a previous year question. So if it is a nucleotide, which bond will be there? Phosphoester bond. Phosphoester bond. Right, bache? Now, here, this is a purine. That is why the bond is in between ninth. The bond is in between, right, ninth nitrogen and first carbon. And if at that place purine will be there, so purine's N1. right? Purines N1 means nitrogen at first position of purine will form the bond with the sugar with the same carbon. That is what you have to keep in your mind. Now I'll repeat the structure. Okay, I will make the same structure again. Now this time I will not write, I'm not going to write rather this thing, this structure. I will simply write here the nitrogenous base. Okay, okay, right? Now here you know that CH2O, this is P double bond O, OH, or you can even mention the ionic form. It is like that. It is like that. So this is your first nucleotide, bache. This is your second nucleotide. This is your next nucleotide. Right? This is your first nucleotide. This is your next nucleotide. This is your second nucleotide. Now what's going to happen? You have a look. This is your sugar, right? So everywhere it is a sugar that is forming the bond with everybody. So here you can see nitrogen is attached here. Here also it is attached. Now this is the point. Now again the water will go out, bache. Minus H2O will occur. Here also minus H2O will occur. So after that minus H2O, what is going to happen, my dear people? After that minus H2O, what is going to happen, my dear people? This is going to happen. This is going to happen here. This. Are you getting my point? Can you see that? Can you see that? This is going to happen. So now, here in this case, you can say it is the phosphodiester bond. Which bond is there? It is phosphodiester bond. Here, this is your phosphoester bond. Now, this phosphate is also attached to this sugar. So, it is phosphodiester bond. So, you will see, we also call it as 3-5- phosphodiester linkage. Anybody in the class having any doubt here? Right? Anybody here in the class having any doubt here? What I just said, people, what I just said, I am saying when it is the, right, I am saying that when it is the single nucleotide, I am saying then it will show phosphoester bond. And I am saying if it is a dinucleotide, or I would say that if it is a polynucleotide chain, then what you people are going to see, you will see 3-5- phosphodiester bond. 3-5- phosphodiester bond. This is important. So in single nucleotide, phosphoester. If there are many nucleotides attached, phosphodiester. Look at this end. This is the 5th carbon here. So 5- will have what? 5- will have what? Yes, please tell me. 5- will have what? It will have, yes, everybody. Can I say that it is PO32 negative? Can I say that 5- end is having the phosphate here? Right? Because here in this chapter, again and again, here in this chapter, again and again, you will talk about this thing, that synthesis is always in 5' to 3' direction. 5- end is having phosphate, 3- end is having hydroxyl. So just imagine, this is your first nucleotide. So of course, the first carbon here, just look at this part. The first carbon here will have, or the starting part will have what, beta? It will have the phosphate group, 5- phosphate. Okay, now this bond here, because this is the first nucleotide, it is phosphoester. It is not phosphodiester. It is phosphoester. It is not phosphodiester. This one, I told you, N-glycosidic bond. If there is purine, then 9th carbon. Sorry, if there is purine, 9th carbon. If there is pyrimidine, right? All these things are indicating. If there is pyrimidine, by mistake instead of pyrimidine I have mentioned purine. So if it is pyrimidine, it is N1. So all these things, na beta, all these things that I am doing, these type of blunders, they indicate that I need a cup of tea. Nahin? Don't you think so? I need a cup of tea. Hai na? I am also feeling sleepy. Chalo, just have a look. Phosphoester bond, N-glycosidic bond. Now, this is one nucleotide, this is next. So here, phosphodiester bond is there. So kindly do not make the mistake here. I am writing it again, right? So when you are talking about polynucleotide chain, so it is 3-5- phosphodiester bond. It is 3-5- phosphodiester bond. It is 3-5- phosphodiester bond. Okay? 3-5- phosphodiester bond. Is that clear? Anybody having any doubt? right? So that is why 5' end is having phosphate. Whenever you will see, because now here you just have two nucleotides. Listen to me very carefully. First nucleotide, second nucleotide. How they are attached? 3', 5' phosphodiester bond. Now imagine there will be more nucleotides, one after another, one after another, one after another, one after another. 5' end will always have this free phosphate group. 3' end will always have this, right? 3' end will always have this free OH group. Are you getting my point? 3' end will always have this free OH group. That's what you have to keep in your mind. This is all about the bonding, my dear students, right? And this is something you all need to keep in your mind. Okay, so in the NCERT, I do have that, but I am interested in having a quick revision. So what type of revision do you want? Quick revision or slow revision? What type of revision do you want? Quick revision or slow revision? Quick revision or slow revision? Quick, ma'am. We want you to finish the entire syllabus in 5 hours and tell us all the important things. Nahi? Tell us all the important things. Okay, ma'am, this is what going to come in paper. Guys, we have discussed everything, right? If you will see our channel, just pick up Game of NEET 2.0. You'll get all the chapters. You'll get all the chapters, right? Human health and disease, because of some technical glitch, I was not able to continue. So if you remember, in the marathon, I started with that. We have discussed that. Okay? Okay. So you will get everything on this channel. Okay? All you have to do is, you have to be consistent. All you have to do is, you have to be consistent. Am I clear? Am I clear? Okay. Okay, so let's go. Done? So just have a look of these structures. So you have the pyrimidine rings. This is your cytosine, uracil, and thymine. Here it is the purine ring, guanine and adenine. Look at the structure. Look at the structure first, right? I hope it's clear. Then it is the sugar, deoxyribose, ribose. Deoxyribose is an exception because it doesn't follow the general formula for monosaccharide. Now, this is the bonding that I have discussed, right? So 3', 5' phospho, 3', 5' phosphodiester linkage is there. So this is your sugar, this is your nitrogenous base, this is the phosphate attached here. This is the third carbon of the sugar which will get attached to the next nucleotide. And here you people can see 3', 5' phosphodiester bond will form. So if you guys are having any doubt, feel free to ask, because I believe this time the question will come from this part. So it is most expected topic. It is most expected topic. Trust me. Trust me, right? And now look at this diagram also. 5' end is having phosphate. If in the paper there is a diagram based question, at the place of phosphate, if they put hydroxyl, that will be incorrect. If at the place of at 3' they at hydroxyl, if they put phosphate, that will also be incorrect. So this is the format that we have, right? So sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. So that is how the things are, and I hope all the doubts are crystal clear. Right? I hope all the topics are crystal clear to all of you. Done, bache? I hope all the topics are crystal clear to all of you. Now, the another thing is from the nomenclature as well. Yes, you heard me right. Here also there is a nomenclature that we have to follow. Now, see, when you are talking about the DNA, we have to discuss their differences and RNA. In the case of DNA, the sugar, as the name is suggesting, the sugar is deoxyribose, and here, of course, the sugar is ribose. Here, what do you have? You have adenine, guanine, cytosine and thymine. So purines are same in both the cases, but when it comes to the pyrimidine, cytosine uracil is there, cytosine thymine is there. Just keep it in your mind, right? Then after that, right, so these are the differences that we need to know. So here in the DNA you will have thymine, here in the RNA you will have uracil. RNA is, right, our single stranded DNA. RNA is our what? It is our single stranded DNA, and when it comes to single stranded RNA, this one is double stranded. This one is double stranded. Am I clear? Double stranded, single stranded, double stranded, single stranded. Chalo. Now after this, if I join adenine plus ribose, means I will get nucleoside. So the nomenclature has to change. It is adenosine. That is again a previous year question, right? So adenine is a nitrogenous base, adenosine is a nucleoside. Let's suppose if adenine at the place of ribose, if it will be deoxyribose, then what will you get? You will get deoxyadenosine. You will get deoxyadenosine. Now I'll pick up cytosine. So cytosine plus, let's suppose if it is deoxyribose, right? I'm just giving you examples here. So it will be your deoxycytidine. Okay, that is how the nomenclature changes. So here you are talking about the nucleoside. Do check it for others as well. Then comes what? Then comes nucleotide. So tide, T for 3, right? T for 3. So it will be your, let's suppose adenine plus ribose plus phosphate. Plus phosphate. That is what we are going to get. So what will you get here? What will you get here? You will get adenosine monophosphate. Right, students? Adenosine monophosphate. Are you getting my point? So because single phosphate is attached, adenosine monophosphate, I can also call it adenylic acid. I can also call it adenylic acid. Same way, you will have thymidine monophosphate. And here at the place of thymidine, you can also write deoxythymidine. It's all same. And right, so thymidine monophosphate or thymidilic acid. Like guanosine monophosphate, guanylic acid. Yes. Do you have any doubt in the nomenclature? Anybody having any doubt here in the nomenclature? I believe this part is clear. Now, DNA, this is the direct information, and of course, we are not allowed to miss that. So when you are talking about the organism, if it is bacteriophage 174, 5386 nucleotide, if it is bacteriophage lambda, 48,502 base pairs, if it is E. coli, Escherichia coli, right, bache? So it will contain 4.6 into 10 raised to power 6 base pair. Haploid content of humans will have 3.3 into 10 raised to power 9 base pair. So it will be 6.6 into 10 raised to power 9 base pair in the diploid content. Am I clear here? Yes. So take the screenshot, everybody. Take the screenshot, please. Everybody, please. Done? It is a nucleo. See, if it is deoxy, right? See, if it is your cytidine, so it is simple. Cytidine is your nucleoside. Done? Okay. So now the structure we have discussed, right? I'll share these points. Don't worry about that, right? I'll share these notes, but the most important thing is at this point of time, you need to be attentive so that you can revise. Okay. So Misher in 1969 named it Misher in 1969 discovered the this genetic material in the nucleus, called it nuclein, and then later Altman renamed it as nucleic acid, already discussed. Now, bache, this information is very important, and this information is a direct previous year question. Yes, this information is again a direct previous year question. I told you, the revision will be very quick today. So it is on the base of X-ray diffraction. So when I'm saying X-ray diffraction, along with X-ray diffraction, I can also call it as X-ray crystallography. I can also call it as X-ray crystallography, right? X-ray diffraction or crystallography. So the data was produced by Maurice Wilkin and Rosalind Franklin. The data was produced by Maurice Wilkin and Rosalind Franklin, right, bache? So let me tell you here, the main lady behind the discovery of DNA is Rosalind Franklin. She's also known as the Dark Lady of DNA. It was her work, that X-ray diffraction. It was her work. It was her data. She obtained the best image of DNA, photo 51, but she didn't get the credit for her that discovery, right? And Maurice Wilkin, without her knowledge, shared this information with Watson and Crick. So they just, explained all the structure, the base pairing part, they got the credit for that. And later on, when Rosalind Franklin, when she published her research, it was considered as a kind of supporting document for what Watson and Crick was saying. So this is the kind of thing that happened. Okay. So overall, when you talk about this, yeah, we say that on the basis of X-ray diffraction or X-ray crystallography data, right, produced by Maurice Wilkin and Rosalind Franklin, Watson and Crick, they proposed a simple but double helical model of DNA in 1953. So year is also very important. So the DNA that they explained is bDNA, my dear people. It is the right-handed DNA. Okay, it is the right-handed DNA. So we have A DNA, B DNA, C, D, E, all of that are there. So remember, it is the Z DNA, that is zigzag, and that is left-handed. That is zigzag and that is left-handed. That is zigzag and that is left-handed. Am I clear? That is what? That is zigzag and that is left-handed. Understood? Sure? Sure? That is zigzag and that is left-handed. Okay? So, yeah. So, it is made up of 2 polypeptide chain. Simple. Let's suppose it is like that. So, this diameter, it is bache 2 nanometer or 20 angstrom. I'll directly discuss the points which are required for your NEET. One strand will have polarity 5 dash, 3 dash. 5 dash end is having free phosphate group. 3 dash end is having free hydroxyl group. Here, it is 5 dash end. This one is 3 dash end. That is how it is. That is how it is, right? So if I have to tell you here, so what am I saying actually? I am saying that 2 strands are anti-parallel. What am I saying? That 2 strands are anti-parallel. One strand is having polarity 5 dash to 3 dash. Another strand is having polarity 3 dash to 5 dash. It is like that. So we say that when it comes to the DNA, it is anti-parallel. What is it? It is anti-parallel. One strand is having this polarity, another strand is having this. So 5 dash end is having free phosphate, 3 dash end is having free hydroxyl. Keep it in your mind. Diameter is 2 nanometer. Now here you have one base. So when it comes to the base pairing, we talk about Irwin-Chargaff. We talk about Irwin-Chargaff, right? Irwin-Chargaff's rule we discuss, right? Irwin-Chargaff's rule we discuss when it comes to this base pairing thing. Right. So basically, James Watson and Francis Crick, the X-ray diffraction, they, took that X-ray diffraction data, they studied that. And even the Irwin-Chargaff's, it's Chargaff's rule also helped them to, crack or decode this structure. So here, you remember one thing, that we have the basis, right? Your adenine, which is a purine, will always form 2 hydrogen bond with thymine. Cytosine will always form 3 hydrogen bond with guanine. So we have 2 hydrogen bond with adenine and thymine. Sorry, 2 hydrogen bond with adenine and thymine, or 3 hydrogen bond in between cytosine and guanine. So If it is adenine thymine, if they two are attached with the hydrogen bond, base pair, if these two are attached with the hydrogen bond, that is also a base pair. So that is something you need to keep in your mind. Okay, so adenine thymine, cytosine guanine, adenine thymine, cytosine guanine. Like, it's not like they are parallelly, but this is what I'm saying. So when they are attached, so it means you are talking about the base pair. Okay, you are talking about what? You are talking about the base pair. So now the next thing that you need to remember is, now the next thing that you need to remember here is, see, if you have one base pair here, I'm talking about the bDNA, one base pair here, this is your another base pair here. Okay, in between these two, the distance, the gap is of 0.34 nanometer, which is also known as 3.4 angstrom. 0.34 nanometer, which is also known as 3.4 angstrom, right? 0.34 nanometer, which is also known as 3.4 angstrom, number one. Now, when you talk about this DNA structure, it's each turn, right? It will contain 10 base pairs. And when I'm saying each turn, I have to consider both. Let's suppose this is minor groove, this is major groove. Same way, this will be the minor groove here, this one will be the major groove, right? This is what we study in the DNA. The proteins, they interact there. The proteins, they interact with this DNA here. Okay, so one minor and major groove together, they are forming one turn. Each turn will have two base pair. If beta, my two base pair, right, each turn will have 10 base pairs. So if two base pairs are having a distance of 0.34 nanometer, then of course it has to be like this, 0.34 into 10, right? So of course it has to be like this, 3.4 nanometer, yes. So it will be 3.4 nanometer or 34 angstrom, right? Because 1 nanometer is equals to 20 angstrom. So here it will be 3.4 nanometer or 34 angstrom. So what I just did, I am saying that each turn is having 10 base pair and the distance between two base pair is this 0.34 nanometer. So of course, here, if the distance will change, but number of base pairs will remain same, you can multiply, number one. If distance is same, number of base pair changes, again you can multiply and you can check this distance. So these are the kind of things that we need to understand here, right? That we need to understand here. So here you guys can see, right? Here you guys can see, it's very simple that two polynucleotide chains are there. Two polynucleotide chains are there that are making the DNA, right? Two chains have anti-parallel polarity. One chain 5' to 3' direction, another chain, like let's suppose here you have 5' end, here you have 3' end, 3' end, 5' end. Okay, so this is what we have. So two chains are coiled in a right-handed fashion. Pitch of the helix is 3.4 nanometer. That's what you have to remember, right? Because roughly 10 base pairs are there in each turn. So the distance between a base pair and a helix is about this nanometer, right? And if I have to convert it into the meter, it will be 0.34 into 10 raised to power minus 9. I have to multiply it with 10 raised to power minus 9. Any doubt? Any doubt? Any doubt? Sure. So the plane of one base pair, it will stack over other in this double helical. So this in addition to H bond will provide stability to the helical structure. It will provide stability to the helical structure. Is that clear? Right? Chaliye. So let's discuss the packaging of DNA double helix. We have to be very quick here. So when you are talking about the packaging, right? In a typical mammalian cell, this is the number that we have to see, but in a diploid cell, my dear students, where are we going to see this? In a diploid cell, you people will see that we have 6.6 into 10 raised to power 9 base pair, right? And if it will be haploid, 3.3 into 10 raised to power 9 base pair, that is what we have to see. So now, if I have to check the length of the DNA, so for that I have to multiply the number of base pairs, right, with the distance between that base pair. Like if I have to check the length of the DNA, right, length of the DNA, so this is what I have to do. Wait, I'll go for I'll go for NCRT here, right, so that you can relate with the NCRT as well. So this is the double helical model. Okay, this is the double helical model. So I have already shared all the important points with you all. So basically Arvind-Chargaff's rule, right, Arvind-Chargaff's rule, that is what you have to keep in your mind, right. So as per this Arvind-Chargaff, you know that, right, you remember this Arvind-Chargaff's rule? Anybody here? Anybody here? So it's simple. The concentration of purine will be equals to pyrimidine. In simple words, if I have to tell you, purine concentration is equals to pyrimidine. So PUGA, cytosine or thymine. That is how it is going to be. That is how it is going to be. So when I will take the ratio, so that is how it will be. That is equals to 1. Right, it will be equals to 1. Yes or no? It will be equals to 1. Okay, and other than that, when you talk about the base ratio, right, so purine's concentration is always equals to pyrimidine, because you know that adenine will always form hydrogen bond with thymine. Thymine and cytosine will always form hydrogen bond with guanine. So that is why when you are talking about this base ratio, right, this is species specific, matlab constant for each species. It is constant for each species. That's the point. Okay, so this is your Chargaff's rule. So on the basis of that, if there is a question, let's suppose I will just solve one type of question here. You can even write down. So let's suppose if they are saying ki adenine amount is given, adenine is 30%, right, and they are asking what is C, what is G, what is T, right, the percentage of them. So it's simple. Adenine will always form the hydrogen bond with thymine. If A is equals to 30%, it clearly indicates your thymine is also 30%. It is also 30%. So 30 plus 30 will be 60. 30 plus 30 will be 60. So you are left with 40. So C will be 20%. G will be 20%. Right? So that is how we can check it in one way or another way. Anybody having any doubt? Anybody having any doubt? Anybody having any doubt? Done? So structures done, right? So anyone having any doubt here in the diagram as well? So 5' end, 3' end. 5' end, 3' end. That is how the bonding is. Adenine and thymine 2 hydrogen bond, cytosine and guanine 3 hydrogen bonds. That's it. Now this is what you have to consider. The diameter is 2 nanometer. The distance between two bases is 0.34 nanometer. Do revise it in this way. Okay, so there are different types of DNA, right? Like but here only Z DNA is the one that is left-handed, right? I'm just focused on the thing which can be asked in the question. It is the only DNA which is left-handed. Rest all are, right? Rest all are what, bache? Rest all are your right-handed. Okay, so now from this part I have picked up one very important line, that if I have to check the length of the DNA double helix, if I have to check the length of the DNA double helix in a typical mammalian cell, if I have to check the length of the DNA double helix in a typical mammalian cell, we have to multiply total number of base pairs with distance between two consecutive base pairs. Total number of base pair with distance between two consecutive base pair. So it is 6.6 into 10 raised to power 9 into 0.34. That was nanometer. We have to convert it into meter. And here the result is 2.2 meter. Again, PYQ. So from this chapter, from each and every single line, previous year question is there from each and every single line, and it can come. Okay, so let's suppose if in the paper there is a question, they have given you 20. Total number of base pairs 20. The distance between two base pair is 10 meter. So obviously 20 into 10, 200. That is what you are going to say, 200 meter. Of course, not possible, just telling you. Okay, so here also, two consecutive base pairs, right? First of all, total number of base pair, multiply it with the distance between the base pairs. So we have converted it into meters. That is why we are multiplying 0.34 with the 10 raised to power minus 9 meter. Is that clear? Is that clear? But the nucleus dimension is 10 raised to power minus 6. So how will that fit in? How will that fit in? Of course, we have to pack the DNA. Of course, we have to pack the DNA. And when it comes to the packaging of DNA, it is there in prokaryotes, beta. It is also there in eukaryotes. Now in prokaryotes, we know that they have primitive. They are primitive cells, right? They are P for primitive, P for prokaryote. Karyon means nucleus. So their nucleus is primitive, and here we talk about what? We talk about the nucleoid. What do we discuss here? Nucleoid. What do we discuss here? Nucleoid, right? Which is also known as genophore. So here what's going to happen? We know it very well that it is also the double-stranded DNA and it is the naked DNA. It is directly there in the cytoplasm. It is directly there in the cytoplasm. This nucleoid, this double-stranded DNA, this naked DNA, it is directly there in the cytoplasm. So it doesn't mean that it is not coiled. It is also coiled. But here, right, no histone proteins are there. No histone proteins are there. But polyamines are present. So polyamines are also positively charged. So polyamines are present here. Are you getting my point? Are you getting my point? So no histone proteins, no histone protein, but polyamines are present. Done? Doubt? Doubt? Yes? Okay, so here polyamines are present, right? So DNA, right, that these positively charged proteins are going to help here. There are no histone proteins. So of course they will also form loops and they will get packed. That is what we need to understand in the case of prokaryotes. Now in the case of eukaryotes, as I said, the DNA in human is 2.2 meter long, but the nucleus is 10 raised to power minus 6. Right, it is very small, right, but the DNA is long, so we have to do the packaging. So when you talk about the packaging, here we discuss it in few steps. As if for an example, it will be having nucleosome model, right? Number 2, solenoid model. Number 3, radial loop model. So that is how it is. So what are they saying? First of all, nucleosome has to form. Now for that, we need to understand something known as histone proteins. When we talk about the histone proteins, what are they? They are the basic proteins, bache. So when I'm saying basic proteins, it simply means they are positively charged. Your DNA is negatively charged, and that, right, your DNA is negatively charged, it will coil over the positively charged proteins, right, that will help in its packaging. Just for an example, this is the what? Histone. It is positively charged. Your DNA, which is negatively charged, will coil over it, right? Because ultimately we have to pack it. Okay, so histone proteins, basic proteins, positively charged they are. And when you are talking about these basic proteins, they have lysine and arginine. You know that proteins are made up of what? Amino acids. So we have lysine and arginine here, right? And they are what kind of amino acid? They are basic, or I would say positively charged amino acid. Okay, so this is what you see in histones. But overall, we have a whole here, right? We have a whole here. So we have histidine, we have arginine, we have lysine. So these are actually the basic amino acid. There is a trick for basic amino acid, that is Hall. There is a trick for basic amino acid, that is Hall. There is a trick for basic amino acid, that is Hall. There is a trick for basic amino acid, that is Hall. Everybody repeat it. There is a trick for basic amino acid, that is Hall. Right? Histidine, arginine, lysine. Histidine, arginine, lysine. Okay, but when it comes to the histone, you will talk about the lysine and the arginine. They are basically basic proteins. So we have H1, we have H2A, we have H2B, we have H3, we have H4. These are the histone proteins that we have. Again, it's a most important question, right? H1, H2A, H2B, H3, H4, right? H1, H2A, H2B, H3, H4. Now what's going to happen? 2 copies of or 2 subunits of H2A, 2 subunits of H2B, 2 subunits of H3, 2 subunits of H4. So basically what is happening? Your H2A, H2B, H3, H4. These 4 histone proteins, my dear people, they are going to form an octamer, right? They will form an octamer and your DNA will coil over it. It's like that. It's like that. So what I just said, that your H2A, your H2B, your H3, your H4, right? All of it will form the octamer. All of it will form the octamer, right? They form the octamer and the DNA will coil over it. And here you have the H1 as the linker protein. Here you have the H1 as the linker protein, right? So this structure, when only histones are there, so histone proteins will form an octamer. Histone proteins will form an octamer. But when DNA will coil over it, then you will call it as nucleosome. What will you call it? You will call it as nucleosome. Done? Done? Done, बच्चे? Done? So between these two, this is your nucleus. So now, when histone proteins are covered, when the DNA is covered over the histone protein, this structure is nucleosome, and this is what we are getting. This is chromatin. And in between these two, this DNA beta, this loosely bound DNA, it is the linker DNA. It is the linker DNA, and it is the one which is transcriptionally active, which can give us the protein. It is euchromatin. So this part is clear. This part is clear. So imagine it in this way, my dear people. Imagine it in this way. Here you can see this is what your DNA is. This is what your DNA is. It is coiling over. It is coiling over the histone proteins, right? It is forming this chromatin. It is forming this chromatin. When we observe it under electromagnetic beads, when we observe it under EM beads, right, what will we see? what will we see? We will see, under electron microscope, we will see that they are just like the beads on a string. They are just like the beads on a string. Now, I told you this is the first model, nucleosome model. Now, what's going to happen next? Like, let's suppose this is what you have got. Imagine, these are the nucleosome, DNA is coiled over that, DNA is coiled over that. These are the beads on the string. They'll further coil. They'll further coil. Okay, just let's suppose this is the wire that I have. This is the wire that I have, right? I am coiling this wire, right, over this. So basically, right, basically what's that? Like, I am coiling my DNA over the nucleosome. So there will be so many units like that. Now, all of them will further coil, all of them will further coil. That's the main agenda here. So, when it comes to the solenoid model, further there is the coiling. Further there is the coiling. After that, further loops will form, right? So, this is the whole point here. So, we started with the 2 nanometer diameter. Then this nucleosome you are going to get. As I said, in between linker DNA, which is transcriptionally active, it is further coiling. Now these nucleosomes are further coiling. Look at the dia here. It is 30 nanometer. So nucleosome will also fold, right, right, into the form of the solenoid or in the fiber. The diameter is this. So it is further forming radial loops. Right. So this 30 nanometer fiber is thrown into irregular loops. They'll form a fiber which is 300 nanometer thick, and again this looped chromatin will coil further and they will form this. This is your chromatid having the diameter of 700 nanometer, and then you know that after S phase it will become 1400 nanometer, right? So when there is the further packaging, like let's suppose if your radial loop model, right, I told you na solenoid then radial loop, here you need some non-histone chromosomal proteins. Non-histone chromosomal proteins, and that proteins are acidic. That proteins are acidic. That proteins are acidic. Any doubt here? Any doubt here? Any doubt here? So just keep it in your mind. So this is what packaging is. Right, this is what packaging is. There are multiple diagrams that are showing this. So this is 11 nanometer, further 30 nanometer, further the loops are there, and further they will form the compact structure like this. Right, compact structure like this. Anybody having any doubt? Anybody having any doubt? So all the points, they say beads on string structure is formed, right, is packed to form chromatin fibers which will further coil and condense at metaphase stage. They will further condense at metaphase stage, and they will form what? They will form chromosomes. So packaging of chromosome at higher level, it also requires some additional set of proteins, and that proteins, as I said, they are NHC, non-histone chromosomal protein, and beta they are acidic as well. So diagram important hai. These are the EM picture which is showing beads on the string. So kindly keep it in your mind, right? Kindly keep it in your mind. Is that clear? Right, then what else do you have? You have, what else do you have? You have euchromatin. You have euchromatin. You have heterochromatin. You have euchromatin. You have heterochromatin. So euchromatin and heterochromatin mein, what do we get here? Right, remember it is transcriptionally active. What's that? What's that? I think it's not visible. Let me. So what's that? It is transcriptionally active. It is transcriptionally active. It is loosely packed. Right? But this one, tightly packed and transcriptionally inactive. Transcriptionally inactive. Keep it in your mind. Okay? Keep it in your mind. Now let's go for the experiment part, the search for the genetic material, right? The search for the genetic material. Let's discuss that, everybody. Ready? So when it comes to the search of the genetic material, of course, Griffith's experiment, then the unequivocal proof was given by Hershey and Chase experiment. We know it very well, but let's have a quick revision of that. So first of all, you will see that DNA acts as a genetic material. It took very long to be discovered and proven, because by 1926, that quest, right, that thinking that which one is actually responsible for genetic inheritance at the molecular level, right, scientists, they tried to figure that out. Okay, so basically they are saying that after the discovery of Mendel, Sutton, Morgan, and many other scientists, so they narrowed down their research. They were like, there is something which is responsible for the inheritance at molecular level, and that something is somewhere there in the chromosome, right? But they need to know what was that. So many experiments were conducted. So the very first thing that we have to discuss here is what? It is the transforming principle given by Griffith. Right, given by Griffith. So when you talk about Mr. Frederick Griffith in 1928, is year important? Yes. So Frederick Griffith in 1928, he conducted the experiment on Streptococcus. So if coccus is the word, what kind of bacteria it is? Spherical shaped bacteria. If coccus is the word, what kind of bacteria it is? Spherical shaped bacteria. Spherical shaped bacteria. So Streptococcus pneumoniae. Streptococcus pneumoniae, that is what we have, right? This is the bacteria. It causes the pneumonia. So it is also written as Diplococcus, pneumococcus, right? So don't get confused if the name is different. Streptococcus, Diplococcus, pneumococcus, spherical shaped bacteria, that is going to cause the pneumonia. This bacteria is having two strains, right? When you talk about these bacteria, you know that metabolic diversity is extreme in their case, right? So let's suppose if this is the bacteria, it is having two strains. S3 strain is there and R2 strain is there, right? These are two different strains of bacteria. So when I'm saying S3, I am talking about the smooth strain. What am I talking? Smooth strain. When I'm talking about the rough R strain, I am talking about the rough strain. Right? This strain is non-virulent one. This strain is non-virulent one. Sorry, this strain is virulent one. Means pathogenic. Means pathogenic. This one is non-virulent. This one is non-virulent. It means it is non-pathogenic. It is non-pathogenic. This one will cause pneumonia, this one will not. Why is it so? This is S strain. Bacteria is having in its surrounding, it is having that capsule, right? That polysaccharide coat is there. That capsule, that polysaccharide coat is there. So when we try to grow that bacteria, we'll see the smooth colony. But here there is no such coat, no such glycocalyx and no such capsule. So that is why it is not causing what? That is why it is not causing pneumonia here. Now when you look at this part, right? So what you people will see? You people will see rough strain. You people will see rough strain, right? You will inject it into the mice. Mice will live. Smooth strain, virulent. Mice will get pneumonia, will die. Then heat killed smooth strain. You are giving heat to that smooth strain. You are trying to destroy its capsule, or you have destroyed the capsule, right? Because of that heat, the capsule is destroyed. So because of that, what is happening, bache? When you inject that particular bacterial strain into this particular mouse, it is going to live. It is going to live, right? And then beta, he conducted the experiment where he injected rough strain and heat killed smooth strain. Rough strain and heat killed smooth strain. What he did? Rough strain and heat killed smooth strain. So when he tried this thing, what happened then? What happened then? So when he tried this thing, so again mouse died. So he was confused. That rough strain is non-virulent. Heat killed smooth strain is also non-virulent. How can it, right? How can it die? So he was confused about that. He was confused about that. So then he checked the things. Then he observed the things. So what he concluded after that, that somehow that R strain was transformed, was converted by that heat killed S strain bacteria. Something from heat killed S strain bacteria passed to this R strain, due to which R strain synthesized that capsule, and due to which it was able to cause the pneumonia to the mice. So some transforming principle, but they didn't mention that it is DNA. So some transforming principle, it transferred from that heat killed S strain and had enabled the R strain to synthesize a smooth polysaccharide coat. Okay, and that is why it became virulent. That is why it became virulent. That is why it became virulent. Okay. Okay, so however, the biochemical nature of genetic material was not defined. Then after that, we talk about this biochemical characterization of transforming principle. So here, Oswald Avery, Colin MacLeod, and McCarthy, MCQ. Scientist name is also important, right? Scientist name is also important. So Oswald Avery, Colin MacLeod, McCarthy, right? So before them, it was considered that the genetic material is protein, right? So they worked to determine the biochemical nature of that transforming principle. So basically what they did, they tried to get everything out of that heat killed S strain, the proteins, the carbohydrates, everything. Now they were trying to check, just let's suppose when they were trying to do that things, na, let's suppose there is a test tube, all the content of that heat killed S strain bacteria is there. They added the protease there. Protease means that will digest the protein. Then they checked whether that mixture is able to transform R strain. The answer was no. They tried with the carbohydrates. The answer was no. Then they tried it with DNAs. The transformation didn't occur. Right, that is the experiment that they conducted. So they say that they purified biochemicals from the heat killed SC cell to see which one could transfer live R cell into the S cell. So they also discovered that the protein digesting enzymes. They also discovered that the protein digesting enzyme, that is proteases, and RNA digesting enzyme, RNases, they will not affect the transformation. I told you why. Because this one will digest protein, this one will digest the RNA. So the transformation will for sure occur. But when there is the digestion with DNAs, so DNA, deoxyribonucleic acid, but it is DNAs. It is the one that will destroy the DNA, will digest the DNA. This is the one that inhibits the transformation. So they were suggesting that DNA caused the transformation, right? But many biologists, they were not convinced, right? They were not convinced. So you can have a look of this experiment as well, right? So they just recovered the S filtrate. Then they treated with deoxyribonuclease. So they were like transformation with live cell. They experimented it. No transformation was occurred. So just have a look of it. Just have a look of it. You will get to know everything about this experiment. Right, so you can even take the screenshot. You can even take the screenshot. Everything will be crystal clear from this part then. Done, beta? Okay, then the next experiment is of Alfred Hershey and Martha Chase, 1952. Year is also important. Beta, they provided the unequivocal proof, because this is also the exact line from the NCERT. They provided the unequivocal proof that the DNA is the genetic material, right? They were the one who provided the unequivocal proof that DNA is the genetic material. So now let's talk about their experiments. So do you know anything about their experiments? Do you know anything about their experiments? Anything about this? Sure. Beta, it is not that N15 heavy isotope, Mr. Kalesh Yadav. Okay, so basically in their experiment, this was their experiment, having 3 steps, right? Their experiment was having how many steps? 3 steps. infection, blending, infection, then blending, then centrifugation. 3 steps were there. They selected what? They selected the bacteriophage. What is this bacteriophage? It is a virus. It is a double-stranded DNA virus. This virus, we call it as bacteriophage. Okay, and this virus can infect bacteria. That is why its name is bacteriophage. This virus can infect bacteria. That is why its name is what? Its name is bacteriophage. Okay, so it is the double-stranded DNA virus. It is the double-stranded DNA virus. Now what's going to happen in this case? This virus is going to infect the bacteria. Whenever we use the virus for transferring the DNA from one bacterial cell to another, it is transduction. Now what's going to happen? Two different sets of experiments were there. In first test, they were testing on the protein. In another experiment, they were testing on the DNA. When they were testing it on the protein, what they did, they used the radio labeled sulfur. Right? Why sulfur? Because sulfur is the part of protein coat present in this particular virus. This virus, this surrounding part, this is the outer protein coat. So they radio labeled it. They radio labeled it with what? Sulfur, S35. This was the isotope that they used. In another experiment, DNA. For that, they use phosphorus P32. And now you know that phosphorus is the part of DNA. Now in the first experiment, first step was infection, right? Where they were trying to grow that bacteriophage near the E. coli so that it can infect it. Then there is the blending, right? Because in the first step, they wanted the infection, ki bacteriophage should infect the bacteria. Then in the next step, blending. They were trying to break the contact in between virus and E. coli. Why is it so? Because in the next step, right, so here basically they have to break the contact. So they agitated them in a blender. Because they need to break this contact. And then there comes the centrifugation, where what they have done, they need to separate the virus component from the bacterial component. So in first step, they wanted this virus to infect the bacteria. In second step, they were breaking the contact, B for breaking, B for blender, with the help of blender, because they agitated the bacteria and virus there. And in the third step, the centrifugation, where they are trying to separate the viral and the virus and the bacterial component. Now what is going to happen here? Right, now what is going to happen here? So virus is comparatively lighter. Virus is comparatively lighter than the bacteria. So its content will settle down at the base. Right, and bacterial component will go up. So in this experiment, they were like that radioactivity was there in the so virus, the component, like virus, its molecular weight is less than bacteria. So obviously the things which are heavy, they will settle down at the base. So bacterial component will settle down at the base, and the virus component will be there in the supernatant. It will be there in what? It will be there in the supernatant. So natant. So what's going to happen? So we are agitating it. We are doing centrifugation, and then they came down at the base. So radioactivity in the first experiment was in supernatant. Right? It was in supernatant. So this is the supernatant. Not in the palette it was there in supernatant. So radioactivity was in supernatant, means it was indicating that the radioactivity was with the viral component. Now the radioactivity was there in the palette. Palette means this is the bacterial component. So from this experiment, we got to know that it is the DNA that will pass from virus to bacteria. So that is how it is. So this is what we have. Okay, this is what we have. Okay. Any doubt? Any doubt? So infection, blending and centrifugation will be there, right? So this experiment proved that DNA is the genetic material. So whenever you talk about the genetic material, of course, it should be able to generate its replica. right? And you know that our DNA will do the same in the S phase. Structurally, it has to be stable, chemically and structurally. So that is why with the time, beta, DNA evolved from RNA. Why? What happened? Your DNA, RNA is single-stranded, right? And you know that RNA can also act like an enzyme, and we call it as ribozyme. We call it as ribozyme. So being an enzyme also, RNA is more reactive. Plus it is having the uracil as well, right? And that ribose sugar is also having that extra OH also, which was making it more stable, more reactive. But whenever you talk about the DNA, yeah, whenever you talk about the genetic material, it should be something which is more stable. So if it is an enzyme, your RNA, then it is more reactive. But we want something which is stable chemically and structurally. Right, so that is why with the time DNA evolved from RNA, right? And you know that DNA also provide the slow change or the scope, a slow change or the mutation that are basically important for the evolution. Okay, beta. So it should be able to express itself in the form of Mendelian characters. That's something we need to keep in our mind, and all the points related to that. जैसे ये देखो, pairing and complementarity is there. Both the nucleic acids are having ability to direct their duplication. RNA, DNA, both can duplicate. Both can duplicate, right? Right? But remember, genetic material should always be stable enough. It should provide only the slow changes, and it should be stable. It should not change with our body's physiology or with our aging or something. It comes under that, right? So that is what we have to remember. And from this Griffith's experiment, where the bacteria was heat killed, but still it has transformed the R strain, right, into the S strain. So it shows that the DNA, it is highly stable. It is highly stable. It shows that. Okay, it is highly stable. It shows that. Okay. Okay, Baki beta, you know that two strands being complementary, if separated by heating, they come together, and when appropriate conditions are provided. So further 2-OH group present at every nucleotide in RNA is a reactive group that will make RNA labile, right? Labile, okay? That its structure is not that stable. It is unstable and it is easily degradable. So RNA is also catalytic, right? Right, RNA is also catalytic, bache. So you know that because it is catalytic, it is more reactive, but we don't want that. Okay, and even the presence of thymine at the place of uracil will also provide additional stability to the DNA, beta. So both are able to mutate, both these nucleic acid, but RNA is unstable. It can mutate at a faster rate. So that is why the viruses which are having RNA genome, which are having RNA as the genetic material, in their case the mutations occur very fast, right? Be it your HIV, be it your coronavirus. So that is why it takes a lot of time for the scientists to study the structure and then to again make the medicine. So that is the point that we need to remember. So these viruses, they evolve very fast, right, bache? So that is what you have to remember. Baki, you know that RNA can directly code for the synthesis of protein, so they can express the characters. Keep it in your mind. So sometimes questions, they come from such lines where we don't pay attention, right? Where we don't pay attention. Okay, so they are saying that RNA can directly code for the synthesis of protein, hence can easily express the characters. RNA, because it is giving the protein, it can easily express the character. And DNA, you know that, is dependent on RNA for the synthesis of protein. DNA cannot make the protein directly. DNA is dependent on that RNA. Okay, so they are saying that protein synthesis machinery, it is evolved around RNA. It tells us about the dominance of RNA, right? So it tells us about the RNA word concept, my dear people. So remember one thing, this point is again important, and I believe the question can come from such point. So where they are saying that DNA being more stable, it is preferred for storage of genetic information, right? Because DNA is stable, so all the genetic information is stored here. All the genetic information is stored here. So ultimately, students, the story is like this. You have the DNA. DNA will form mRNA. mRNA will form protein. This is the central dogma of information. This is the central dogma of information. This is the flow of information given by Francis Crick. Given by Francis Crick. So DNA to mRNA. Transcription. mRNA to protein. Translation is there. Okay, and this DNA, it can self-replicate. So DNA to mRNA, it is transcription, beta. DNA to mRNA, it is transcription, right? It is transcription, and mRNA to protein, it is translation. So if you will look at it, so you know that DNA needs, right? DNA, it needs the RNA for making protein. It cannot directly make it. It is dependent on this. But DNA being more stable than RNA, so the genetic information, all the information is stored here. But when it comes to the transmission of that information, mRNA is preferred. So read this line carefully, and do not make the mistake here. Do not make the mistake here, guys, quick. Do not make the mistake here. Done? Done? Chaliye, next. RNA word concept, I have discussed it. First genetic material it is. All the essential life processes evolved around it. It is also a catalyst. We know that we use the word ribozyme for that. So why it is unstable and all, we have covered it. Now next come to the DNA replication. Okay? So we have to start with the experimental proof here. So whenever we talk about the DNA replication, see, it is Watson and Crick model for semi-conservative DNA replication. It is Watson and Crick model for, right, semi-conservative mode of DNA replication. Remember one thing, when it comes to this thing, right, when they ask who proposed the, keep it in your mind, people, keep it in your mind. When they ask who proposed the mode of DNA replication, जैसे ये देखो, right, here it is clearly mentioned, right, that Watson and Crick, they had immediately proposed a scheme for the replication of DNA. They were the one who proposed a scheme for the replication of DNA. That is how DNA can be replicated. They proposed that semi-conservative mode. When I'm saying semi-conservative mode, ultimately what is happening? Semi-half. Conservative, you need to conserve something, save something, protect something, whatever you want to say. Now here what happened? This is the semi-conservative mode. So this is the parental DNA strand. Yeah, parental DNA. These two are the daughter DNA. These two are the daughter DNA. Daughter DNA. Parental DNA is there, daughter DNA is there. Now here in the daughter DNA, one strand is as such, it is parental, and the another strand is newly synthesized one, and the another strand is newly synthesized one, right, and the another strand is newly synthesized one. Take it, right? So this is semi-conservative mode of DNA replication, where in the daughter strand, one strand is ditto like the parent strand and one is newly synthesized. So how this newly synthesized DNA strand will form? Of course, complementarity. The principle of complementarity is there. It's simple. Parental strands will open. If parental strand is having A, C, G, T, A, imagine. So of course students, because complementarity is there, complement, the rule of the bases are complement. If it is A, T will form. If it is C, G will form. If it is G, C will form. If it is A will form. If it is A, T will form. So if you open the parental strand, that will act like what? That will act like a template, right? It is having, right, this single, when we open the parental DNA or DNA strand, each strand will act like a template because it is having certain bases. We just need to add bases complementary to that, and this is what we are going to get. So who proposed it? When it comes to who proposed it, then you will go for what? Watson and Crick. But when it comes to the first experimental proof, you will go for Michelson and Stahl. Michelson and Stahl's experiment. Whose experiment I'm talking about? Michelson's and Stahl's experiment. Whose experiment I'm talking about? Michelson and Stahl's experiment. Michelson and Stahl's experiment. So when I'm talking about this experiment, they experimented on E. coli. What they used? Heavy nitrogen isotope. If you will write here the radioactive isotope, that is incorrect. It is the heavier one. It is the heavier one. And if, right, because it is on E. coli, if in the question they are asking eukaryotes, ki first such experimental proof for semi-conservative nature of DNA replication in eukaryotes, then you talk about the experiment of Tyler and colleagues, right? Tyler and colleagues. And that experiment is on plant, that is Vicia faba beans, right? Vicia faba beans. Okay, and here they used radioactive thymidine. So initially I'm just drawing the comparison here, right? I'm just drawing the comparison here. Keep it in your mind. They are simply asking first experiment, Michelson and Stahl, E. coli, heavy isotope of nitrogen. It is not radio labeled. So it is not radioactive. It is heavy, just heavy. Here on the basis of weight, you are separating the thing. When they ask the experimental proof, in the case of eukaryotes, you'll go for Tyler and colleagues. You'll go for Tyler and colleagues, right? Vicia faba beans, radioactive thymidine was used. From this topic, if you ask me the question, that is, right, these are the general points, right? These are the general points. Done? Done? So Michelson and Franklin Stahl, they performed the following experiment in 1958. So when we talk about this bacteria, its generation time. Its generation time. The generation time means that in how long it will divide into the and will form the new generation. So we have to consider it 20 minutes. I know at places it is 18 minutes also. You go for the 20 minutes here. And why 20 minutes? Because in their case, the circular DNA will replicate, will show bidirectional mode, in bidirection, right? It is working like that, okay? So they grew E. coli. They grew E. coli in a medium containing 15 NH4Cl. So by default, if you talk about our DNA, okay, one second, students. So by default, right, normally, naturally, if you talk about our DNA, it is having N, but that N is 14, that N is 14N. What's that? That N is 14N, right? Normally, this is what we have. This is what we have. Now the point here is, right, E. coli is normally having 14N DNA. Now we will shift it to a medium where heavy isotope of nitrogen is there. And we will allow it to grow there for many generations, because we want the cells having 15N isotope of nitrogen in their DNA. This is what we need. This is what we need. Right, bache? Right? So 15N is the heavy isotope of nitrogen, and it is the only nitrogen source for many generations. Right? Heavy isotope. So what they have used ammonium chloride. Can we get question from this part? 110%. So they used ammonium chloride, and this N is the heavy isotope of nitrogen. It is the heavy isotope of nitrogen. So result was that 15N, it became the part of all the daughter cells. Now, why are they doing it? Because they will separate the heavy DNA molecule, right, from the normal DNA molecule. And how are they going to do it? Again, it's a PYQ, cesium chloride density gradient. Cesium chloride density gradient. Matlab from each and every line, you can get the question here. This chapter is that important. Okay. This chapter is that important. Right. But what is the issue with the students? What is the issue with the students? When we start, let's suppose I'm talking about genetics. They'll ask about respiration. Right. They are asking about the respiration. Now, when I'm teaching molecular, they'll ask about evolution. When I'll teach evolution, they'll ask about ecology. So why can't you focus on one topic at a time? Why can't? Huh? Nahi? Chaliye. So they use cesium chloride density gradient method, bache. Then they transferred the cells into a medium. Matlab, now they have the E. coli, which is having 15N, right, as a part of its DNA. Now they again shifted it to the normal ammonium chloride, where the normal or the lighter, comparatively lighter isotope of nitrogen is used, right, bache? So at times, what will they do? They'll grow the E. coli for one generation, then they'll take out the DNA, then they'll go for another generation, then they'll take out the DNA. So they are taking out that they took that samples, they extracted the DNA, right? Because, and what they did, they independently separated them on cesium chloride gradient, because they have to measure the density. They have to measure the density. So that is what we have. 15N DNA. That is what we have here. 15N DNA. After 20 minutes, this is what you will get, intermediate. This is what you will get, intermediate, also known as hybrid, right? So after 20 minutes, I told you why 20 minutes. The two daughters DNA, right, they extracted that E. coli's DNA, right, when they did the centrifugation. So they were like this. Let's suppose this is what we have, right? This is the control that we have. So here the bands for heavy isotope is there. Here the bands for lighter isotope is there. So this is something that they had already measured. Now what they did, they are like that after first generation, when they check this DNA under that cesium chloride density gradient method, so they were like, that band is not at this place, the band is not at this place, but it is somewhere in between, right? So that is why they called it as hybrid. That is why they called it as hybrid, because it is somewhere in between. Now after another 20 minutes, they checked, okay, initially just like starting, that same, because two daughter DNAs were there having hybrid, and now because they are growing in 14N medium, so all were having the lighter isotope. So that's what they got, and that is how they proved the semi-conservative nature of DNA replication. So question can come like this as well, my dear people, right? That let's suppose you started with one DNA strand. So after 4 generations, right, they can give you the same condition that initially they shifted it in 15 and then in 14. And so after 8 generation, how many DNA strands will have the hybrid? How many DNA strands will not have the hybrid? So the question like this can come. The question like this can come for sure, right? So this is about the experiment, right? This is all about the experiment. Now we will talk about the process of DNA replication, and that too in a very simple way. Guys, I will not write anything regarding this process. I'll make it easy for you. I'll make it simple for you. Then you have to read it from the NCERT. Are you all aligned? Are you all aligned? You know that it's a very lengthy chapter. We took 8 to 9 hours or more than that if we have to explain it in detail. But now we have to move fast because we have the other chapters as well. We have to target the biotech today as well, and we have to target the evolution as well. So if you want me to do that, you have to be a support system. So quickly tell me in the chat section. Quickly tell me in the chat section. Are you ready? Are you ready? Are you sure? Want to see some fire in the chat section, guys? Quick. Right? Okay, chali. So the next topic that we have to discuss is DNA replication. So when we talk about this DNA replication, in the case of, so it will occur in S phase, remember, interphase, S phase. Remember this DNA replication. And in our syllabus, in detail, we read about the prokaryotic DNA replication, not the eukaryotic DNA replication. Okay? We discuss the prokaryotic DNA replication of E. coli, not the eukaryotic DNA replication. So just imagine this is your DNA strand, right? We know that it will follow the complementary base pairing rule, but before that you have to understand that we have to open the DNA helix as well. If we need to copy the information, right, present on that DNA, we have to open it first. We have to make sure that the two strands, they separate and they can tell us about what? They can act like template and we can synthesize new strand. This is something we need to keep in our mind. Are you getting my point? So we have prokaryotes, we have E. coli. So obviously here the DNA that we have is circular. Am I right here? The DNA that we have here is circular. Am I right here, people? Am I right here? So here you are talking about the circular DNA. Right? You are talking about the circular DNA. So of course the replication has to start. So it has to start from a point. I'll be fast here. So that point from where the replication will start is your origin of replication, my dear people. What's that? That is origin of replication, which is also known as ORI site. It is the place from where the replication will start. It's not like that. The replication can start from any point. It is the point from where the replication will start. It is origin of replication. And when you talk about that, in the case of prokaryotes, because circular DNA is there, they have one ORIC site. They have one origin of replication. No replicant, because they have single replication point and they are poly or multi-replicant. Right? They are poly or multi-replicant. So they have, because in eukaryotes, what do you have? Linear DNA. So in the linear DNA, there are many sites from where the replication can start. They are multi-replicant. Any doubt here? Guys? Please tell me. Do you have any doubt? I am talking about the DNA replication. I am talking about the S phase here. I am talking about the prokaryotes here. So we have the circular DNA. So we have one origin of replication, one ORIC site, and in eukaryotes we have many origin of replication. We have multi-replicant site. Is that clear? Are you sure? So it is the point from where the replication will start. So if I talk about the history here, that how is it possible, how can this happen, right? How it is possible for them to open the DNA, you have to recognize that site. Again, enzymes and proteins are involved. But of course, we cannot focus on that part at this point of time. So this is the point from where the replication is starting. So it will be there in both the directions. So that's why in E. coli we say bidirectional mode is there. It's not like that your enzyme will start from this part, will copy the information slowly from one strand, then will come back, will copy the information from other strand. No, this thing will take place simultaneously. So bidirectional mode is there. So firstly tell me this part is clear. If anybody is having any doubt here, do let me know, people. So after all this, what is required? Unzipping of DNA or unwinding of DNA. We have to open this DNA double helix. And for that, there is the requirement of an enzyme, that is helicase. I'll stick to the topic, right? I'll provide you all the essential information and points, but you try to understand that. So helicase is required. So this enzyme, it needs the energy from ATP hydrolysis, plus it needs magnesium ion. So it is the one that help in opening the DNA double helix. So what do you think that how is it going to do the same? It's simple. Just imagine, right? Ye dekho. Imagine this is your DNA, right? Imagine this is the origin of replication. The point from where the replication will start. So your helicase, it has to open the DNA helix. It cannot do it in one go. No, because it takes a lot of energy. It cannot do it in one go because it takes a lot of energy. It will do it slowly. Okay, it will slowly break the hydrogen bonds. This helicase, right? So imagine if this is your DNA, that is how it is going to be 3' to 5', 5' to 3'. That is how it is going to be. That is how it is going to be. Am I right? Am I right? Slowly. It will not be in a one go. It will not be very fast. It will go slow, right? This is the fun thing. So because of that, you are getting what? You are getting a Y-shaped replication fork. And here, you are getting the template strand. One template strand having polarity 3' to 5'. Another template strand having polarity 5' to 3'. Look at it. You are opening your DNA. Your helicase is doing the same. So you are doing it slowly because you have to break the hydrogen bond. So one template strand is having polarity 3' to 5'. Another template strand is having polarity 5' to 3'. So this is the template strand. This is the template strand having polarity 3' to 5'. This is the template strand having polarity 5' to 3'. That is how the things are. So now your helicase, it is trying to open the DNA, right? Just imagine this is your helical DNA structure. My bad. This is your helical DNA structure, right? You are coiling it. You are doing what? You are coiling it. You are trying to open it. Enzyme is there. That enzyme, with the help of that enzyme, you are trying to open it. There comes a point when supercoilings will come. These are the supercoilings. I am not creating them. They will not allow me to open it. Okay, we have to remove that. Plus, these strands, they will again try to join, right? We have separated them. They have the complementary basis. They will again try to join. We don't want that. So here, there is the role of some other proteins. We call them as single-stranded binding proteins. These proteins, my dear people, these single-stranded binding proteins, what will they do? These single-stranded binding proteins, what will they do? Yes, what will they do? These single-stranded binding proteins, my dear people, they will, right? They will not allow, right? Or I would say that they prevent re-annealing here. They will not allow the hydrogen bond to form again. So that's why I'm saying they'll prevent re-annealing here. This is the first aspect. Plus here, right? Here, one more thing is there. Just imagine this is the DNA double helix. This is the DNA double helix, right? Now, beta, here, right? You have the origin of replication. I'm talking about prokaryotes. Now, helicase has to open it. Bidirectional DNA replication is there. So, it will open to both the sides. It has to open to both the sides like this. So, here, in this format, you'll see the replication bubble is there. Right? So, imagine it like this. This is your double helical DNA, right? That replication is in both the directions. So it will be something like this. Okay. So this side is also having, it is also showing that, if you will observe carefully, Y-shaped replication fork. This side is also showing that Y-shaped replication fork. Okay. Y-shaped replication fork here. Y-shaped replication fork here. So overall collectively it is what? It is your replication bubble. What's that, people? It is your replication bubble. What's that, people? It is your replication bubble. Are you getting my point? Are you getting my point? So this is what we have here. Okay. Now the another thing that you have to remember is we have one more category of enzyme, that is topoisomerase. Right. What is the role of that? It removes the supercoiling. What is the role of that? It removes the supercoiling. Right. So now when I talk about topoisomerases, beta, I have two classes, class 1, I have class 2. Right, matlab this class 1 will cut the DNA, single strand of DNA will, fold the other part, will try to remove the coiling. Class 2 topoisomerase can cut both the strands, right? Matlab it will make a cut in both the strands, then it will try to uncoil it. So this is what we have. So now here, remember, DNA gyrase. It is what? It's a type of topoisomerase only. It is the one that is discovered in prokaryotes. It is the type of topoisomerase only. It is the type of topoisomerase only. Where was it discovered? In prokaryotes, DNA gyrase. So this gyrase also needs the energy from ATP hydrolysis. It also needs the energy from ATP hydrolysis. Okay. Achha, this part is not visible, right? So it also needs the energy from ATP hydrolysis, right? ATP hydrolysis. Are you getting my point? DNA gyrase. So it will get the energy from what? It will get the energy from ATP hydrolysis. So it will do what? It removes the supercoiling, right? In prokaryotes, we have DNA gyrase. That is the type of topoisomerase only. So it removes the supercoilings, right? But it also introduced some supercoilings. Let's not talk about it here, right? So it releases the tension which arises due to what? Which arises due to supercoilings. Is that clear? Is that clear? So prokaryotes mein DNA replication will take few minutes, but in eukaryotes it will take hours. It will take hours. Am I clear? Now, other than that, we have to form new chain. So this, even if you are talking about the prokaryotes, this is your DNA here. Right, here it is the cytoplasm. So beta, all the bases are present here only, right? All the bases are present here only. That will help in the formation of new chain, and that is something we need to understand. Just for an example, here in cytoplasm, right? Here in cytoplasm, DATPs, DGTPs, DCTPs, DTTPs, they all are present, right? They all are present. Just DATP. DATP means deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytidine triphosphate, cytidine triphosphate, thymidine triphosphate. Now, if you remember, when I taught you nucleotide, I clearly mentioned monophosphate. I told you adenosine monophosphate will be there, guanosine monophosphate, cytidine monophosphate, thymidine monophosphate will be there. That is what I told you. Right, now here you have the triphosphates. So they are acting like substrate. They have dual purpose here actually. They have dual purpose here. Right, they act as substrate. They act as substrate and provide energy for polymerization as well. Provide energy for polymerization as well. So it will provide the, yes, it will act like the substrate, plus it will provide energy for polymerization as well. It acts like the substrate, plus it also provides the energy for polymerization. Am I clear here? Yes, am I clear here? Right. So basically, all I'm trying to say, like let's suppose if this is your DNA, here let's suppose you have G, here let's suppose you have A, you have T. So complementary to G, C will come, T will come, A will come. So initially they are triphosphates. So now they are acting like the substrate as well. So when they will join, right here, so their two extra phosphates will release. They are high energy phosphate bond. They'll release the energy that will be used for polymerization. This is what I'm trying to tell you here. Okay, so now other than that, we have to discuss about certain enzymes, right? When it comes to the DNA synthesis, especially in prokaryotes, we will talk about DNA polymerase. Now I'll use the short form here, DNA polymerase 1. We have three types actually. We have DNA polymerase 2, and we have DNA polymerase 3, my dear people. We have DNA polymerase 1, 2, and 3. We have DNA polymerase 1, 2, and 3. We have DNA polymerase 1, 2, and 3. So, main replicating enzyme and the main replicase is this DNA polymerase 3. And it is in the case of prokaryotes. Right, it is in the case of prokaryotes. Why? Because bache, in eukaryotes there are different types of DNA polymerase. Just they will have DNA polymerase alpha, beta, delta, epsilon, etc., etc. So they have different types of, right? They have different types of DNA polymerase. But here you will have, in prokaryotes you will have 1, 2, and 3. Right? You will have 1, 2, and 3. Okay, so this one, it is on the name of the scientist. So it is also known as Kornberg enzyme, bache. It is also known as Kornberg enzyme. So now when you talk about this, it is the main replicating enzyme. Why is it so? Why is it so? Why it is the main replicating enzyme? Because in DNA replication, you need accuracy plus speed. You don't just need speed. You don't just need accuracy. This process has to be fast plus accurate. Like in your NEET examination, you have to give the responses fast, but that too in an accurate manner. You cannot say that I'll go very quick, I'll do this, do that. No, no, that's not going to happen. That's not going to happen at all. So keep it in your mind, guys. Okay? Keep it in your mind. So DNA polymerase alpha, beta, delta in eukaryotes, etc., etc. Here you have this. So this one is having more fidelity. This one is the one which is more accurate. This one is the one which is very fast as well. Now, in DNA polymerase 1, dekho, it is having 3' to 5' exonuclease activity. Even this one is also having 5' to 3' polymerase activity. It is also having 5' to 3' exonuclease activity. If you have studied it earlier, you will be able to relate. If not, pay attention. So this one is unique. It is required for gap filling, my dear people. It is required for gap filling. ठीक है? ठीक है? So here in DNA polymerase 2, 3' to 5' exonuclease activity, 5' to 3' polymerase activity. Here also 3' to 5' exonuclease activity, 5' to 3' polymerase activity. So if you people observe this thing, all the 3 enzymes can synthesize the DNA strand. But we go for DNA polymerase 3 because it is fast, it is accurate, number 1. Okay. Now the another point is, here in this, and now the another point is, what about this 3' to 5' exonuclease activity? It's simple, bache. Right? It's very simple here. Just keep it in your mind. Ye dekho. Always remember, whether it is your RNA or it is your DNA, synthesis is always in 3' to 5'. Synthesis is always in 5' to 3' direction. Doesn't matter what's that, whether it is DNA or it is RNA, synthesis will always be in. New strand will always be formed in 5' to 3' direction. When I was explaining you the structure of DNA, I have explained you that. Right? I have explained you that why is it so? What is the need for that? Right? What is the need for that? I have already told you this. Take now, you have synthesized the strand, let's say in 5' to 3' direction. Of course, we have to make sure that there is no error. Of course, we have to make sure there is no error. Now, from 3' to 5' side, just it's like that now. Let's suppose you are arranging people in a sequence. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Now you have to check them back. You'll move in the same direction and you'll be like 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. Again, I'm repeating. Let's say you are making a queue. Yeah, you are checking a queue. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Then you're coming back and you're checking it. 10, 9, 8, 7, 6, 5, 4, 3, 2, 1. That is how it is. Okay, that is how it is. That is how it is in a queue. So here, this is the role of this 3' to 5' exonuclease activity. It is a part of that DNA damage and repair. It will make sure the errors are not there. Now, when it comes to the unique activity, MCQ can come from this part. I'll explain it. When I'll explain you the DNA replication ka complete process, I'll explain it. Now, students, this is your Y-shaped replication fork. 3' to 5', 5' to 3', right? 5' to 3', okay? 3' to 5', right? This is 5' to 3'. That is what we have. Now, bache, whenever you talk about your DNA polymerase 3, no doubt it is the main replicating enzyme. No doubt it is the main replicating enzyme. What kind of enzyme it is, bache? It is the main replicating enzyme. What type of enzyme it is? What type of enzyme it is? It is the main replicating enzyme, but it is having a drawback. It needs 3'OH to start DNA strand synthesis. So it is not capable of doing the synthesis by its own, right? It needs a 3'OH. It needs a 3'OH to start the DNA strand synthesis. So that is why we talk about one more enzyme here, which is known as primase. It is also known as DNA dependent RNA polymerase. It is also known as DNA dependent RNA polymerase. Why it is DNA dependent? Because it will use the DNA strand as a template, number one. Why it is RNA polymerase? Because this primase, it synthesizes the short RNA primer, right? It is having 10 to 15 ribonucleotides, right? So because it will synthesize this RNA primer, this DNA polymerase 3 will get 3'OH. Then further, DNA polymerase 3 will further start adding deoxyribonucleoside monophosphate. So that is the drawback here. That is the solution here. So first of all, right, as I said, the synthesis is always in 5' to 3' direction. The synthesis is always in 5' to 3' direction. So first of all, RNA primer will form here. Here the direction is this, right? The template strand here is what? The template here is what? 3' to 5'. The template here is what? 5' to 3'. So of course, when this is the template strand, right, the direction, the reaction of this synthesis will be this. Jaise ye dekho 5', 3', further bases will add it, right? Further bases will add it. It will go like this, right? It will go like this. It will go like this. Number one. Then here 5' to 3', here also strand will synthesize, but in this direction, opposite, right? 5' to 3', 5' to 3', 5' to 3'. Now you must be thinking, why am I making this? It's very simple, bache. It's very simple, right? Here, no doubt, the DNA replication is semi-conservative. The DNA replication is semi-conservative, but it is also semi-continuous or semi-discontinuous. Now, what's the meaning of discontinuous? Semi-continuous or semi-discontinuous? Semi-continuous or semi-discontinuous. That is the word that we have. Now, it's simple. It's simple. You can see this. This is the direction the helicase is moving, right? And it is opening the DNA helix. This is the direction of that. This is the direction of replication fork as well. This is the direction of replication fork as well. So in this direction, when DNA polymerase 3 is moving, right? Because it can synthesize the strand in 5' to 3' direction, right? So it will easily synthesize the strand. But it's not like that. Polymerase will go, first add bases here and will make this daughter DNA, then will come back and make this daughter DNA. No, it has to do this activity simultaneously, parallelly. It has to go all together. Now here, when this DNA polymerase 3 is synthesizing the DNA strand, It is moving in the direction of replication fork. It is synthesizing in the direction of replication fork. And here also simultaneously it has to synthesize. But the direction of synthesis is opposite. Here 5 prime to 3 prime. Here 5 prime to 3 prime is like this. So it has to work in the opposite direction. It has to work in the, जैसे देखो here 5 dash to, see, 5 dash to 3 dash, 5 dash to 3 dash, 5 dash to 3 dash. So one enzyme. It's like one enzyme. Like let's suppose you are here. You have to do one thing like this. But same thing, this. That will be, and that too parallelly, that too in opposite direction. That is a tedious task. Right? That is a tedious task. Right? That is a tedious task. So here what's going to happen? Now, when it comes to that template strand 3 dash to 5 dash, then because polymerase is also moving in this direction only, then continuous strand synthesis will be there. It is leading strand. One time RNA primer will form, it will further start adding the bases. But when this is the template strand, there is the discontinuous synthesis. There is the discontinuous synthesis. So what type of strand will form? It is the lagging strand. What type of strand will form? Lagging strand. What type of strand will form? Lagging. Why? Why, bache? Because again and again, just let's say here it has started, it will make the primer here, it will synthesize, the connection will break, then again. Oh, right? Then again. Okay, can you see that? Can you see that? ये देखो, again it will add. Again it will add. So like this. So here you will see this is the lagging or discontinuous strand, and these fragments are known as Okazaki fragments on the basis of name of the scientist. Right? On the basis of name of the scientist. Right? Right, bache? This is something you have to remember. So question can come from template strand, that which template strand, right, will have the continuous strand synthesis, which one will have discontinuous strand synthesis, right? It will be like that. Okay? 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 So that is how the synthesis is going to be. That is how the synthesis is going to be. Is that clear? Is that clear? So one is leading, one is lagging. One is leading, one is lagging. I told you why. Now, here in this DNA strand, bache, these are the, right? If this is the DNA, imagine these are the RNA primers. We cannot let them like this only. We cannot say if it is my DNA, right? In between RNA primers can be there. No, of course not. So now here comes the role of 5 dash to 3 dash exonuclease activity. Here comes the role of 5 dash to 3 dash exonuclease activity of DNA polymerase 1. Right? Here comes the role of 5 dash to 3 dash exonuclease activity of DNA polymerase 1. Right? It will remove the primer here. Right? So it helps in primer removal and gap filling. It helps in primer removal and gap filling. It helps in primer removal and gap filling. So it will remove the primer from this part, right? And then here it will add the DNA bases here, right? It will remove the primer, will add the DNA bases, and finally the molecular glue known as DNA ligase will join sugar phosphate backbone here. That is how it is. That is how it is. Are you getting my point? That is how it is. So this is all about the DNA replication. So what you have to do, read it from this side, look at the diagram here. If there is any point, tell me then. If there is any point, tell me then. Done? Any doubt? Sure? Sure? So leading, lagging, stand, and here if you will see polymerase delta has mentioned there. It means they are talking about the eukaryotes, right? So for your quick revision, the diagrams and this table is also there that you will get after, right? That you people will get after what? That you people will get after the lecture. Okay? So next topic that we need to understand is, I already told you, central dogma of information. Now we will understand what transcription is. Okay? We will understand what transcription is. So again, before going forward, let me tell you, your DNA is making mRNA. That is transcription. mRNA is making protein. That is translation. Right. So this DNA, it shows what? Self-replication. It shows what? Self-replication. Are you getting my point? Are you getting my point? So DNA to mRNA will show what? Transcription. mRNA to protein will show translation. So transcription site in prokaryotes, it is cytoplasm. In eukaryotes, because in prokaryotes there is no nucleus, in eukaryotes it is nucleus. It is nucleus. Then here it is the translation. Where will it occur? It will be there in cytoplasm. So who gave the central dogma of information? Your Francis Crick. Who gave the central dogma of information? Francis Crick. Right? Who gave it? Francis Crick. Now, one more thing that you have to remember, that RNA can also give us DNA, right? So this is the central dogma reverse. This is the central dogma reverse, also known as teminism. How is it possible? With the help of enzyme, reverse transcriptase. With the help of enzyme, reverse transcriptase. Is it clear? Everybody here in the class? Is it clear? Everybody here in the class? Done. So next is the transcription, right? Next is the transcription. Now बच्चे we have to speed up and we have to focus on the important points as well. जैसे देखो HGP है, question will come from HGP. That is totally theory. I'll highlight the important points, just read it. Your DNA fingerprinting in biotech, we will cover it. Operon, genetic code, there are short videos that I have, posted multiple times. Do check that out. Right? Here also I'll explain, but I'm telling you the way outs as well. So let's focus on that main point. So are you ready? Let's focus on transcription. So when we talk about the transcription, right? See, when we talk about the transcription, we need to discuss about the transcription unit. We need to discuss about the transcription unit, right? But even before that, I want to tell you something, that we need to remember something that is known as cistron. We need to remember something that is known as cistron, right? Just say here, let me show you the NCERT part as well. I'll start with the cistron first, then we'll come to this part. Okay? I'll start with the cistron. I'm not telling you anything extra, anything tough or something, right? I am just picking up the important points or the direct point here, right? I'm just picking up what? The important point and the direct point here. So what am I talking? I am talking about the cistron. जैसे ये देखो. Okay? Okay. So when you talk about this, I'll discuss the transcription unit, but firstly I'm talking about something known as cistron. When I talk about the DNA, but my DNA, right? It is not, initially the concept was one gene, one enzyme. One gene, one enzyme. Concept was there, right? एक gene, एक enzyme. But later on it was one gene, one protein. Later on it was one gene, one polypeptide, because some enzymes, they are made up of one polypeptide, some are made up of many. Now when you talk about the gene, yes, gene is the functional unit of inheritance. Indeed, we know it very well that gene is what? Gene is the functional unit of inheritance. We know it very well. We know it very well. So there is no ambiguity, no doubt that genes are located on DNA. But it is difficult to define it in terms of DNA sequence. Gene is the unit of inheritance, right? It is true. That is obvious, right? But in terms of DNA sequence, when we have to define it, then it is different, because the DNA sequence is also coding for your tRNA, also coding for rRNA, also coding for, right, also acting like a unit of crossing over, yeah, mutation, whatever it is. There are multiple things here. But the main point is, when we have to say that DNA is giving us the polypeptide, then what are we doing? So by defining a cistron as a segment of DNA, we are saying cistron is the segment of DNA which is coding for polypeptide, right? It is the code of, it is the, right? So cistron is that segment of DNA which is coding for polypeptide. Cistron is that segment of DNA which is coding for polypeptide. So here, when it is the transcription, there will be a gene which will give us the protein. So we will call it a structural gene. We'll discuss it now. If that structural gene, it is monocistronic, it is in the case of beta eukaryotes, mostly eukaryotes. Mono means single, cistron. Like that structural gene will give you single polypeptide. But in prokaryotes, P for prokaryotes, P for poly. So they will, right, they will have, that's one structural gene will give you many polypeptides. That is the concept. Again, don't get confused. Prokaryotes are polycistronic, but they are monoreplicant. Eukaryotes are monocistronic, but they are multireplicant, yeah, polyreplicant. So keep it in your mind. Okay, keep it in your mind. So this is something you have to keep in your mind. So this is my transcription unit is, right? So here 3' to 5' polarity, 5' to 3' polarity. This is the transcription unit having promoter, having terminator. In between the structural gene is there, right? And here in this structural gene, this strand having polarity 3' to 5' will act like the template strand. Okay, beta. And the opposite one having polarity 5' to 3' will be coding strand. It will be your non-template strand. It will be your sense strand or positive strand, a plus strand. This will be your non-coding strand. It will also be your anti-sense strand. So don't get confused. If I'm saying anti-sense, I'm talking about the template strand only. If I'm saying anti-sense, I am talking about the template strand only. Is that clear? Is that clear? Is that clear? Right? So in the transcription unit, you have the promoter, of course, that helps in starting the transcription or promoting it. Terminator will help in stopping it. Here you have the structural gene that will give you the polypeptide. Okay. Now, bache, when you are using the word template strand, see here in the case of transcription, both the strands cannot behave like like template, because the RNA that they are going to give us is a single stranded, right? The mRNA, the messenger RNA that they are going to give us is single stranded. And let me tell you, messenger RNA is just making 1 to 5% of total RNA. It is not making much. It is just making 1 to 5% of the total mRNA. Yeah, sorry, RNA. Okay? 1 to 5% of the total RNA, right, bache? And it is the least stable RNA. What type of RNA it is? It is the least stable RNA. Now, this RNA, it is having uracil. We know it very well. Now, if let's suppose if both the strands of the DNA will start making the mRNAs, they will be complementary to each other. They will just join with each other. They will become double-stranded. Then how are we supposed to form the protein? These are the kind of things we need to take care. Now, imagine. So here only one strand will act like a template, and that template strand, my dear students, that template strand will have polarity 3' to 5'. 3' to 5'. It will be 3 dash to 5 dash. So no doubt the word coding strand is given, right? But that doesn't mean it is the one that will, give you the coded information. Nahin. It's not like that. So it is the template strand having polarity 3 dash to 5 dash. Then there is the coding strand having polarity 5 dash to 3 dash. So template strand will give you the RNA. That is the point. That is the point. So just imagine this is your DNA that we have, right? So here let's suppose you have adenine, thymine, cytosine, guanine, adenine, right? Again adenine, thymine, guanine, cytosine, again adenine, right? Again thymine. So of course it is having polarity 3 dash to 5 dash. It is having polarity 5 dash to 3 dash. So here you will have, because they are to complementary na, T, right? They are to complementary na. This is, I'm talking about a DNA. So it will have T, A, G, C, T, T, A, C, G, T, A. That is the sequence here. That is the sequence here. Now, because this one is my template strand, so I will, and synthesis is always in 5' to 3' direction. So my mRNA, what my mRNA will do? My mRNA here, because this template strand has to be transcribed, right? So here it will have U, A, G, C, U, A, C, G, U and A. Just check it. Just check it. A complementary to A, U will come. Don't make this mistake. You are talking about the mRNA. It doesn't have thymine. So at the place of thymine, if adenine is there on the DNA, mRNA will show what? mRNA will show uracil, right? Not thymine. So that is what it is. That is what it is. Now, if you will pay attention on the sequence of mRNA and the coding strand, if you will pay attention on the sequence of mRNA and coding strand, you guys will see one thing. What? That this mRNA is exactly having the sequence like this coding strand. Only at the place of thymine, uracil is there. The sequence on mRNA and the coding strand will be same. Only at the place of thymine, uracil will be there. That is the only difference. So if in the paper they'll give you the sequence, ki hanji, this is the sequence on template, what should be there on the coding strand. So of course, what should be there on the mRNA, just check the coding strand, that's it. So this is one point that you have to understand. And let's suppose if both the DNA strands, if they will start forming the RNA, beta, they will be double bonded then. If it will be double bonded, then how are you going to do the translation? So that is something you have to keep in your mind. Now, somewhere here, you have a start point actually, from where the transcription will start. Okay? So imagine it like this. This is the start point. When you go to this side, this is upstream. This is downstream. You're going downward. Keep it in your mind like this. So start point, something is this side. Something is at this side of the start point. You'll call it as upstream. Something is at this side of the start point. You will call it as downstream. You will call it as downstream. Keep it in your mind. Okay? Keep it in your mind. Okay? Done? 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 So start point, upstream, downstream, upstream, downstream. Keep it in your mind. So if I take the reference of coding, okay, so this is my start point. My promoter is upstream to that. My terminator is downstream to that. Focus on this language. I'm just picking up the important topic. Okay. Now the next point here is that you need to keep in your mind. It's like some teachers or even some students, they get confused. And I don't know why. It is such a simple topic. So please pay attention. If guys, I'm taking the reference of coding strand, that is also a previous year question. If I take the reference of coding strand, so coding strand is here, right? If I'll ask, where is promoter? You will say at 5' end. If I take the reference of coding strand, you will say terminator is at 3' end, right? So in the question, sometimes they ask it that where is terminator located at 3' end or 5' end if we take the reference of coding strand or the template strand. So read such questions carefully. Read such questions carefully. So if you look at this part, if you look at this part, right? If you look at this part, what you have to keep in your mind, right? Just have a look, my dear people. Just have a look, my dear people. Right? Please have a look, my dear people. So what are we right doing here? If we are taking the reference of coding strand, promoter is at 5' end, terminator is at 3' end. If we take the reference of template strand, promoter is at 3' end and terminator is at 5' end. So it's like that. I'm facing the camera. I'm saying my right side is having wall, my left side is having gate. Okay, right side is having wall, left side is having gate. Now I am saying my right side is having gate, my left side is having wall. So both the things are that particular place only. It's just that I have changed the reference, right? Firstly, I was facing the camera. Now I'm not. Accordingly, I'm telling you. This is the point here. Kindly keep it in your mind, right? Kindly keep it in your mind. Okay. Okay. So in the case of promoters, right, I'll discuss few points directly here only. Okay, so promoter is a sequence which is upstream to the start point, and it helps, of course. How does it help? It helps in what? It helps in the transcription, definitely, for sure. Now in the eukaryotes, we also discuss one more concept that is not there in prokaryotes. Beta, in eukaryotes, I told you the transcription will be there in nucleus. Transcription will be there in nucleus. Okay, right? Here in the eukaryotes, we will talk about 3 types of RNA polymerases. Just in prokaryotes also, we need to open the DNA. We will have one RNA polymerase. Here we have RNA polymerase 1, 2 and 3. That also I will discuss, but now this is not my major concern. Actually, I want to tell you something here. In the case of eukaryotes, the mRNA that forms, that is actually an immature mRNA. What type of RNA it is? Immature. It is also known as heterogeneous nuclear RNA. It is immature RNA, heterogeneous nuclear RNA, right? Now, why is it so? Because this mRNA is not having the coded, like it cannot give us the information for synthesizing a protein. Why? Because here it is having certain sequences. It is having certain sequences, right? जैसे कि it will have exon, intron. It will have exon, intron. It will have exon, intron. So basically in eukaryotes, their mRNA is initially immature, right? They have exon and introns in them. And same thing. Why is it so? Because in DNA we have the same thing. Now when you are talking about the exon, it is the coding sequence. It is having the information for coding the DNA. Now when you're talking about the introns, so exons are having what? They are having the information for coding, right? For coding a protein. They are having the information for coding a protein. And introns, they doesn't have that, right? They are the non-coding sequences. And why is it so? Because in eukaryotes, right, you discuss about the concept of split genes. These split genes, they have evolutionary significance as well, right? In eukaryotes, that is what we see. We see that these genes, right? We see that these genes, because see, eukaryotes are monocystronic. One structural gene will give you one polypeptide, right? But prokaryotes are polycystronic. So in eukaryotes, there is a concept of what? There is a concept of split genes. There is a concept of what? There is a concept of split genes, right? Now in these split genes, it's not like that gene will give you the complete information for the protein synthesis. No, just like this mRNA, of course. That's why mRNA is like this. They are also having exon, intron, exon, intron, exon, intron like this, right? And it is having the evolutionary significance, right? It is having what? It is having the evolutionary significance indeed. Indeed, beta, it is having the evolutionary significance. Are you getting my point? Are you getting my point? Right? Right. So, mostly eukaryotic genes are the split genes. They will form the mRNA, which will have coded and non-coded sequences. So, of course, if we need the functional mRNA, we have to remove the introns. That is the point. That is something you need to keep in your mind. And right, we don't discuss it here at the class 12th level, but yeah, we do discuss it when in our higher studies, that this is also of benefit. Just say for an example, I have exon 1, 2, 3, 4. Let's suppose sometimes It is as per the requirement of the body. Sometimes we can join 1, 2 and 4 exon, we'll get different protein. We can join 2, 3 and 4, we can join 2, 3 and 1, then we'll get different protein. So that is the concept here, that is the point here. But we don't touch that part. So split genes are there in eukaryotes, not in prokaryotes. Just keep it in your mind. This is the story of eukaryotes only. Okay. So mostly eukaryotic genes are like that, except genes for histones and interferons. Except genes for histones and interferons. They are not split genes. They are not split genes. Genes for what? Genes for histones and interferons. Genes for histones and interferons. Are you getting my point? Are you getting my point? So that is something you have to keep in your mind, number one. Now, other than that, see, these are the different, diagrams that I have. Now, whenever we will talk about this, we'll discuss the enzymes. One thing that I want to tell you, just say, let's suppose if this is the start point. Upstream to that, you have the regulatory region, you have the Tata box. What is this Tata box, bache? What is this Tata box? It is the point where these RNA polymerase will join, will help the DNA helix to open and then to synthesize the mRNA. Then this Tata box is known as Priv-now box in prokaryotes. These are not the prokaryotes, right? In prokaryotes. And this Tata box is known as Goldberg-Hugeness box. In both the cases, you'll see the Tata box, but a P for prokaryote, P for Priv-now. Here in eukaryotes, it will be your Goldberg-Hugeness box. It will be your Goldberg-Hugeness box. Am I clear here? Now, let me come to the topic directly. Okay? So, I told you what transcription is. I told you what promoter, structural gene and terminator is. And even I told you that which reference do we need to take. But here, we need to talk about the enzyme. So, come to this part. So, when you are talking about the prokaryotes, in the case of prokaryotes, which enzyme are we talking here? We are talking about the RNA polymerase, which is also known as DNA dependent RNA polymerase, which is also known as DNA dependent RNA polymerase, which is also known as DNA dependent RNA polymerase. Am I clear here? Am I clear? Am I clear here? Which is known as DNA dependent RNA polymerase, right? Which is known as DNA dependent RNA polymerase, because it will use DNA as template and it will form RNA. Now in prokaryotes only, right? You have some factors. Just say it is the sigma factor. S for sigma, S for start. It helps in initiation of the process. When it is termination, it will be rho factor, right? Rho factor. And the sigma and rho factor, they are in prokaryotes only. In eukaryotes, you are not talking about them. In eukaryotes, you are not talking about them. In eukaryotes, you will not consider them. We have different factors. As a termination, transcription factor, the, right? A, transcription factor D, A, right? B, right? G, right? E and H. That are the factors. But here we have sigma and rho. Here we have sigma and rho factor. Now this sigma factor will bring your RNA polymerase, as I said, to the promoter region, having Tata box, right? Having what? Tata box. So this Tata box here is, right? This Tata box here is TATAAT. These are the 1, 2, 3, 4, 5, 6. 6 bases are there. And it is easy to break the hydrogen, 2 hydrogen bonds in comparison to 3. So that is why we have the Tata box here, not some CGVG box here. Okay? So sigma factor will come, will tell RNA polymerase, ki bro, please come, please try to open. Firstly, it will tell, because here it is the RNA polymerase only. So remember, this RNA polymerase will also help in opening of DNA helix and RNA strand synthesis. Both will be done by this. Okay? So in 2020, NEET 2021 question was there. Which enzyme helps in opening of DNA double helix in transcription? So even when I was solving the paper in hurry, I was like, opening the DNA double helix, it should be helicase, but it was not, because in the question they specified that in transcription they are talking about the things in the transcription. So that is something you need to keep in your mind. Okay, that is something you need to keep in your mind. Is that clear? Is that clear? So sigma factor will bring this RNA polymerase. So remember, it will only help in initiation. Once RNA polymerase starts the mRNA synthesis, it will leave. So sigma factor is there in initiation but not in elongation. It is there in initiation but not in elongation. It will help that RNA polymerase to start that mRNA formation, but like open the DNA helix and start. Once it starts that, it will leave. It will not help in elongation. That's it. And in elongation also the same story is there. Your ribonucleotides, triphosphates, right? Your ribonucleotides, triphosphates. Okay, they all will act like substrate as well as provide energy for polymerization. So once the stop sequences will be recognized, the termination will occur. And it's not mandatory that termination will always be dependent on raw factor. Sometimes it is raw independent as well. Next diagram. It is in the case of eukaryotes. So in eukaryotes, number one thing that I just told you, right? They have the concept of split genes, right? So because of that, their mRNA is going to be immature at first place, right? It is heterogeneous nuclear mRNA. It is immature, number one. Secondly, here you have different RNA polymerases, 1, 2 and 3. So when it comes to the mRNA, it is mainly the 2, right? It is mainly the 2. So this is the information that we need to keep in our mind, that RNA polymerase 1 will transcribe RNAs like your 28S, 18S, 5.8S, right? Not 5S. And RNA polymerase 3, from 3, remember 3, THRE3. So remember tRNA. So it helps in tRNA synthesis plus msRNA. So your 5S ribosomal RNA will be synthesized not in nucleolus but in the surrounding, right? In the nucleoplasm there, it will be done by RNA polymerase 3. Am I clear? It will be done by RNA polymerase 3. Am I clear? Am I clear? Right? So the RNA polymerase 3 will synthesize tRNA, will synthesize 5S ribosomal RNA, will synthesize snRNA, small nuclear RNA it is, right? Will synthesize small nuclear RNA as well. Small nuclear RNA as well. Keep it in your mind. It will synthesize small nuclear RNA as well. And it is RNA polymerase 2 that will give you what? That will give you your mRNA. So 1 ka role I told you, 1 ka role I told you, 3 ka role I told you, 2 ka role I told you, that it is basically will form immature mRNA, heterogeneous nuclear mRNA, precursor mRNA. That's it. Now here, right here, if you will see, you people will observe it, right? See, exon introns are there. So we have to go through one process, which is again unique to your eukaryotes, not to your prokaryotes. That is RNA processing. Now we have to process this RNA so that we can send a functional mRNA in the cytoplasm for protein synthesis. So two things will occur, beta. First is capping, matlab at 5' end. At 5' end, an unusual base will be added. Why unusual? Because it is 7-methyl guanosine triphosphate. Normally in the strands here, right, monophosphates are added. 7-methyl guanosine triphosphate will be added here. 7-methyl guanosine triphosphate will be added here. Which enzyme will do the needful here? Guanyl transferase. Which enzyme will do the needful here? Guanyl transferase. Now, it is the 3' end. It is the 3' end. Here, tailing will be done. When it comes to tailing, adenylate residues will be added. adenylate residues will be added. Means many A will be added. And that's how in a template independent manner. How will they be added, bache? In a template independent manner. In a template independent manner. How will they be added? In a template independent manner. They don't need any template. Simply they will just add poly. So this is the PAP, polyadenylate polymerase. Polyadenylate polymerase, that will do the needful here, right? So here capping at 5' end, 7-methyl guanosine triphosphate, enzyme will be guanyl transferase. Tailing, adenylate residues will be added, and that too in template independent manner. Matlab so many A will be added. We don't need any template here. Simply they'll be placed with the help of this enzyme. So this capping and tailing will help the mRNA from the nucleases present in the cytoplasm. This capping and tailing will help the mRNA to protect it from the nucleases present in the cytoplasm. Any doubt here? Any doubt here? Next, we have to remove the introns. So removal of introns is known as splicing. Splicing. For that splicing, spliceosome is required. So it is unique to eukaryotes. So remember, you will only see it in eukaryotic cell, not in the prokaryotic cell. This is the spliceosome, right? This is the spliceosome. They remove the intron. So this spliceosome, it is having some small nuclear, right? It is having some what? It is having some SNRPs. What will it contain? It will contain some SNRPs, right? What am I saying? I'm talking about I'm giving the word, SNRPs, right? What am I saying? I am saying some SNRPs are there. What are they? SNRPs. SNRNPs. They are pronounced as SNRPs. They are pronounced as SNRPs. SNRNPs, right? So SNRNPs, how do we pronounce them? We call them as SNRPs. SNRPs. SNURPs. SNRPs. What is the full form here? They are small nuclear. They are small nuclear ribonucleoproteins. They are small nuclear ribonucleoproteins. What are they, people? They are small nuclear ribonucleoproteins. SNRPs. So SNRPs along with some other proteins, they form these structures known as spliceosome, which help in the removal of intron. That is it. Right, that is it. This is something you need to remember here. Okay, so this is all about the spliceosome as well. I'm just looking for any other diagram if I have added, because chalo, koi baat nahi. Right, so this is all about this process. So again, take the screenshot here. Remember the spliceosome. You will only see that spliceosome in the case of your eukaryotes. Okay? So spliceosome are nothing. They have SNRNPs combined with some other proteins. They have SNRNPs combined with some other proteins. Am I clear? So take the screenshot, please. Take the screenshot, please. Yes, please. Done? So just keep it in your mind. Then next is genetic code, right? Which is triplet given by George Gamow. Who gave it, bache? The term genetic code was given by George Gamow, right? It is triplet. Okay, so chemical method developed by Hargobind Khurana. Okay, Marshall Nirenberg, right? So chemically, matlab who developed it? Chemical method to synthesize that. So basically, right? Hargobind Khurana. Then Marshall Nirenberg's cell-free system for protein synthesis, helped in deciphering the code, right? And insertion or deletion of one, chalo ye to mutations hai. These are the quick revision points, but you will only be able to understand them once you will do what? Once you will understand this genetic code wala funda. So any doubt? Any doubt till this point? Any doubt till this point? Dekho beta, it is very simple, right? Your DNA is giving you the mRNA. It's very simple. Trust me, it's simple. Your DNA is giving you the mRNA. That's simple. DNA is also polynucleotide. RNA is also polynucleotide. It is making the sense. But mRNA is giving me the protein. Protein is made up of amino acid. Protein is a polypeptide made up of so many amino acid. RNA is made up of so many nucleotide. What type of relation do they have? How a nucleotide chain can give me the amino acid chain, polypeptide? That is the question. That is the relationship we see here. And this relationship is explained with the help of what? This relationship is explained with the help of your genetic code, right? Right? This will be explained with the help of genetic code. So it was George Gamow, a physicist, who argued that since there are only 4 bases, right? Which bases do we have? You can arrest gangster, right? Which bases do we have? You can arrest gangster. Which bases do we have, bache? You can arrest gangster. Am I right? Am I right? These are the bases that I have. So you can arrest gangster, right? You can arrest gangster. So when I'm saying it is you can arrest gangster, right? When I'm saying that you can arrest gangster, right, beta? So 4 bases are there. So he said if we need to get 20 amino acid, because this is the magic amino acid, they'll give us all the protein, 20 amino acid. Remember? Alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, proline, phenylalanine, serine is there, tyrosine, threonine, tryptophan, valine, right? 20 amino acids are there. But yeah, now the number is 22, but in NCERT we discuss about this only. So 21st is your selenocysteine, 22nd is your pyrolysine. 21st is your selenocysteine. Please write it down. 21st is your selenocysteine. 22nd is pyrolysine. 21st is your selenocysteine. 22nd is your pyrolysine. 21st is your selenocysteine. 22nd is your pyrolysine. 22 amino acids are there. So he said that there should be combination. They say here that mathematics ka combination permutation thing will come. Let's suppose if you will take 2 bases. Out of 4, any base can come. Out of 4, any base can come. Only 16 amino acid. But if I take the combination of 3 bases, at first place, out of 4, any base can come. At another place, out of 4, any base can come. At another place, out of 4, any base can come. So it will be total 64 codons. How many codons will be there? 64 codons. How many codons will be there? 64 codons. So 64 codons. Right? That too triplet. Why triplet? Because each codon is having 3 bases. So here 20 codons are directly giving me my 20 amino acids for sure. Right? 3 codons here are the stop codons. So when I am talking about the stop codons, the trick is UUU, AG, AGA. That's how I remember. UUU, AG, AGA. In this way only you have to write it. UUU, AG, AGA. UUU, AG, AGA. So this UUU, AG, AGA, matlab UAA, it is your ochre, it is your amber, it is your opal. So is it important? Yes, madam. Because these 3 codons are the stop codons. They are the nonsense codons. Because they'll stop translation. Do we need to remember? Yes. So 20 plus 3, 23. So we are left with how many codons, beta? 41 codons. Isn't it? 41 plus 3. 44? Huh. 44 plus 20. 41 codons. So what about these 41 codons? मतलब 20 ठीक है, 20 will give me 20 amino acid, 3 are the stop. What about rest 41? So this is what we need to understand here. This is what we need to understand here. So when it comes to the scientific contributions here, yeah, they are very, very, very important. Just here they have simply mentioned the name of Indian scientist Hargovind Khorana, right? So he was instrumental in synthesizing RNA molecule. He took some defined combination of bases. Homopolymer मतलब सब same, जैसे U like this. Copolymer, just giving you some lame example. Let's say if it is Cu like this. So first 3 bases, next 3. First 3, next 3. That is how it is. Right, so Hargovind Khorana's contribution. Then Marshall Nirenberg's cell-free system for protein synthesis finally helped the code to be deciphered. So Severo-Ochoa enzyme, which enzyme? Polynucleotide phosphorylase. It helped in polymerizing RNA with a defined sequence in a template independent manner. Right? With the defined sequences in a template independent manner, बच्चे मतलब with the help of enzymes, without using any template, we can just make our RNA. Is that clear? Is that clear? Done. So this checkerboard will be formed, beta. I'll be quick here. This side, so the trick is you can arrest gangster. The trick is you can arrest gangster. The trick is you can arrest gangster. Right here. So this side is showing first base, second base, third base. Matlab, I'll be quick. Please pay attention, guys. Please pay attention. So you can arrest gangster. You can arrest gangster. So you can arrest gangster. So just for an example here, the first base will be C. This side is telling me C. Second base will be A. And here it is like this. You can arrest gangster only. So CA. Just let's suppose it is A. First base A. Second base C. Then you can arrest gangster, or AC. Now let's suppose this. First base U. Second is all first base U. Second base U. You can arrest gangster. Right? Same way. Let's suppose first base G. Second base G. G. You can arrest gangster. So that is how we can make this checkerboard. So because it is a marathon, I really don't want to skip anything. So that's why I'm telling you this as well, but along with the topic. So beta, here we have the trick, right? We have the trick. Just here for this column, we have the trick. Sar pro thar ala. What is the trick that we have? Sar pro thar ala. Right? Sar pro thar ala. Done, बच्चे? Right? Done? Done? Done. So serine, proline, threonine, alanine. And there are other tricks. So just put genetic code tricks by Ambika ma'am. You will get a very short video of less than 10 minutes. Open it, check it. Now here, remember, जैसे ये देखो, your stop codon, stop codon, stop codon. Other than that, it is only your tryptophan and here your methionine. जैसे methionine is also known as initiator codon. ठीक है, it will start, it is also known as start codon. It helps in starting the translation. Now tryptophan and methionine, beta, they are the one which are just having one codon, rest all are having more than one. Jैसे your leucine is having 6, right? Your leucine is having 6, your serine is also having 6, right? Other than that, your arginine, if you will see your valine done, so the serine and lysine, do not forget about it. Serine and lysine, do not forget about it. Even your arginine, ye dekho 4 here, 2 here. So they are, this amino acid is coded by 6 codon. 6 different codons are coding same amino acid. So these are the points which we need to revise. So that is why just focus on this. So codon is triplet, done, right? One codon codes for only one amino acid, so it is unambiguous. Matlab it is specific. So it doesn't matter which species are you talking about. It doesn't matter which cell are you talking about. Prokaryote, eukaryote, whatever it is, right? Doesn't matter the species, doesn't matter the cell. In all the individuals, right? These codons will give you same amino acid. That is why we say the genetic code is universal. Okay, so it is unambiguous, yeah, non-ambiguous, matlab it is not doubtful, it is specific, right? So ambiguity is not there. If in the paper, if they have said genetic code is ambiguous, then it is incorrect. So it is unambiguous and specific, right? Unambiguous and specific. Matlab in all the possible situations, triple U will give you phenylalanine. It will not be like, today I'm not in the mood, I will give you something else. No. In all the possible situations, it will give you phenylalanine only. Now the degeneracy. Matlab more than one codon can code for same amino acid. जैसे more than one teacher can same student, because the subjects are different, but here actually the point is that more than one codon can code for same amino acid. So many times students, they get confused in unambiguous nature and degeneracy. Unambiguous mein we are saying only one amino acid will be coded by one codon, no matter what. Thus a particular codon will give particular amino acid is. But here we are saying that more than one codon can code for same amino acid. Jaise UUC, both are giving us phenylalanine, right? Both are giving us phenylalanine. That is the point, right? That is the point. Baki, when you read the codon on the mRNA, first of all, it has to be in 5' to 3' direction, right? This is first 3 bases, next 3, then next 3, then next 3, then next 3, then next 3, like this. Continuity should be there. No comma, no punctuation. It cannot be like that. Ki firstly, you are reading 3 bases, then you are reading like this. No, continuity. First 3, next 3, next 3, next 3. First 3, next 3, next 3, next 3. Then, as I said, it is universal, but right, but some exceptions are there. That is in mitochondrial codons and in some protozoans. Few exceptions are there. Baki beta, AUG is having dual function. It is coding for methionine as well as the start codon, right? But sometimes GUG can also act like the start codon. Remember that. Sometimes it is the GUG that can also act like the start codon. That's it. Any doubt? Any doubt? Right? So sometimes GUG. So when I'm saying GUG gives me valine. But sometimes it can also give me methionine. That is the exception. Take it? That is the exception. And Hanji. Okay, so this is all about the genetic code. And beta, whenever you talk about the degeneracy, it is due to the last base, right? Which is at 3' end. That is also known as vowel base, right? Matlab, whenever you will read the translation, you will talk about the tRNA, right? Then we see this is the anticodon loop, which has to form the bond, yeah, which has the codon complementary to mRNA. complementary to mRNA. So this first two bases, they show the Watson-Crick pairing. Third base will show non-Watson-Crick pairing, right? So this third base is that wobbly base that is responsible for degeneracy. That's it, that's all. That's it, that's all. ठीक है, that's it, that's all. बाकी बेटा mutations में obviously बात है, as I said, right? So 1 2 3 1 2 3 1 2 3 1 2 3 1 2 3. It's like that, right? Now let's suppose if somebody will add one nitrogenous base here, one nitrogenous base here, one nitrogenous base here, the reading frame will disturbed. Yeah, somebody will delete one nitrogenous base here, reading frame will disturbed. You will add two, again the disturbance is there. Let's suppose if you have added three bases, or if you have added deleted three bases, that reading frame will not be disturbed. So because if there is such type of mutations that can result, these are the point mutation if there is a change in single base pair, right? And let's suppose if there is addition or deletion of one or two, they can even change the reading frame. So when it comes to this mutation, you have to focus on single cell anemia that we have discussed in the disorder part as well. Remember that what is going to happen, that at sixth position in beta chain, at sixth position in beta chain, what's going to happen? Your GAG will become GUG. Your GAG will become GUG. Matlab GAG will become GUG. So at the place of glutamic acid, valine will come. Right? Valine will come. So that is the exception that we have here. So frame shift mutation can occur. Now beta, let's discuss about the tRNA structure. Let's discuss about the tRNA structure, and then let's start what? Let's start this particular part. So when you talk about the tRNA, it is also making 10 to 15%. So 3' end will have OH, and always remember mostly 5' end is having G. And when you are talking about this 3' end, the reading frame is always 5' to 3'. Reading frame is always 5' to 3'. So that's why we say CCA is there. That is why we say CCA is there. We are not saying ACC, we are saying CCA. So this is the tRNA that we have. When it is the 2D structure, then it is clover leaf-like. Then it is clover leaf-like. And when it is the 3D structure, it is your inverted L-shaped structure. It is your inverted L-shaped structure. Okay? Inverted L-shaped structure. So this side is having OH, which will form bond with the amino acid. Amino acyl bond will form. So this is your amino acid acceptor arm. This is your anticodon arm, and this is your loop. So in the case of arm, obviously bases are paired. This is loop, single bases are there. They are not paired. They are not forming hydrogen bonding. So amino acid acceptor arm, where OH, this OH will form bond with amino acid. Will form bond with amino acid. And remember beta, this amino acid, COH group of amino acid will form the bond here. So it is the amino acyl bond, right? That is also PYQ. It is the amino acyl bond number 1. Then here it is the anticodon arm. This one is variable. Its function is not so clear, but it helps this tRNA to get categorized. Okay? This one, this arm is your T-Cy-C loop. Right? This part is loop, this part is arm. When it is arm, the bases are paired. This is loop. T-Cy-C. Cy means pseudo-uridine. Uridine, unusual base it is. Right? Here, this arm is your DHU loop. DHU stands for dihydrouridine. Dihydrouridine. DHU stands for dihydrouridine. Dihydrouridine. So this, that is how you have to write. D for down. When we are down, we need energy. We need enzyme. So this side will bind with the enzyme. Right? DHU loop. DHU loop. This side will bind with enzyme. Which enzyme are we talking about here? tRNA. Hanji. It is amino acyl tRNA. synthetase. Which enzyme it is, beta? It is amino acyl tRNA synthetase. Which enzyme it is? Amino acyl tRNA synthetase. So DHU loop clear. Anticodon arm, this will bind with the mRNA, as I said, right? So remember your tRNA, right? This tRNA is just making 10 to 15%, 10 to 15% of your, right? It is just making 10 to 15% of total RNA, beta. It is also known as adapter. It is also known as a soluble. Right, so it is also known as adapter, it is also known as soluble, and it is also known as the smallest RNA. Smallest RNA. Keep it in your mind. Okay? So that is how it is. That is how it is. So this THU loop will help to get attached to the ribosome. So this function has to be clear, number one. Secondly, here, they say these are the diagrams of NCERT. Here they have mentioned serine. Here they have mentioned tyrosine. Try to remember their codons and anticodons because sometimes they ask the question from this part. Okay? Sometimes they ask the question from this part. So this is what we have, inverted L-shaped, clover leaf-like. This is what the translation is. So let's have a break for 20 minutes, right? We have to speed it up. If we are interested in finishing the topic, we have to pace it up. Okay, we have to pace it up. Okay, done. We have to pace it up. So let's talk about it. So let's have a break. Then we will revise translation, operon. Human Genome Project, completely theoretical, discussed multiple times. Just will tell you the question that will come in paper. That's it. Then we will move to the biotechnology. Then we have to finish the most important chapters and topics from biotechnology. Then we will further go for the evolution and ecology. And let's suppose if you think that, ma'am, we will not be able to complete the chapters, all the chapters today. I am not here to give you the false hope of syllabus completion. Ki I will touch this, touch that. No. Whatever is important, right, from these chapters, because they are very scoring chapters, bache, they are very scoring chapters. Molecular basis of inheritance, biotechnology principles and processes, applications, your ecology part, very, very scoring. So I have to pick that up, right? I have to pick that up. Are you getting my point? I have to pick that up. I want, if you are reading these chapters, at least you should be able to attempt all the questions. Am I right? We should be able to attempt all the questions from these chapters. So rather than, giving you the feel, ki, theek hai, we have completed everything. No, whatever we are going to complete, that will be the most important one, and that we have to discuss sincerely. And after this marathon, right, as I said, we have next marathon of class 11th revision. This is not going to be like regular marathon ki again madam will come, students have to sit for 10 hours, 11 hours, 12 hours. Nine. Only the NCERT part will be taken. NCERT lines will be taken. NCERT questions will be taken. I'll give you the lines, you have to fill the blanks. I'll give you the lines, you have to fill the blanks. That's how it will go. That is how it will go, right? So this is one revision session for us. Then Tuesday, I'll again come up, right? I'm not going to leave you guys ever. Okay? Again I'll come up Tuesday. I'll tell you ki beta, chalo, get up. Let's revise the biology, right? 12th in the same format. We'll start with the ecology then. Then after that, we will have one final revision with all the MCQs. Okay? All the MCQs. Then we will just focus on MCQs. So MCQs, all the diagrams. Already we have covered the disorders part, and already I am putting these type of sessions for all of you, right? They are so good sessions, right? Even Yava sir, Wasim sir, we all are putting sessions like that. So ye dekho, complete microbes, right? In 10 minutes, the entire chapter is done and dusted. No question will come from outside, right? After that, morphology, all the families of morphology in 15 minutes. All the families of morphology in 15 minutes, done and dusted, right? Then the cell cycle, which will directly give you 24 marks, is done and dusted. Sorry, this is plant growth and development. Here we have your cell cycle and cell division. So who's going to revise it? Yeah, you, right? I have done my work, right? I have posted everything required, right? I have even completed the morphology ka this part, families ka part, which you need to remember. So it's you who has to wake up, who has to, pick up the things, who has to revise, right? So if you will write na genetic code by Ambika ma'am, you'll get the tricks. Crab cycle by Ambika ma'am, get the tricks, right? ETC by Ambika ma'am, you will get the tricks. So this is what you have to do. Operon by Ambika ma'am, you'll get the tricks, right? Morphology, plant kingdom, all the tricks are there. Okay, so let's have a break. Okay, so do it. So let's have a break till 11 p.m. Then we'll see you. Okay, then we'll see you. Yeah, biotech to we have to complete na, very scoring. Very scoring it is. What's up, people? All good? Well, Hannah? All good? Well? Okay, so let's talk about the translation, right? Hannah, madam, you just started the class late and now you are directly coming to the point. Yes, yes, right. I was just having my meal and of course it took some time to, right? So that is why. Huh. Chaliye. So let's start the topic, and the topic that we need to discuss now is translation. Which topic do we need to discuss now? We need to discuss the topic translation. Am I clear here? Which topic do I need to discuss, beta? I need to discuss the topic of translation. So translation, the number one thing is it will there in the cytoplasm. Be it a prokaryotic cell, be it a eukaryotic cell, where will it occur? Where will it occur? It will be there in the cytoplasm. Am I right? Done. So it is the translation, right? It is the translation, right, bacha? So what do we have here? We are talking about the translation. The very first thing that we need to understand here is that there has to be the activation of amino acid, because ultimately these are the amino acid that we have to put in that polypeptide chain, and that will be added as per the sequence on the mRNA, right, bache? So if I have to tell you about the activation of amino acid, see, we talk about the 20 amino acids. We will have 20 particular tRNAs for that 20 amino acids, and we will have 20 particular enzymes for that tRNAs. Okay? Okay? And where is it occurring? It is taking place in the cytoplasm. Okay? So what's going to happen that your amino acid, right, with ATP, right? And now, as I said, the 20 amino acid, or we will have 20 tRNA, 20 enzyme. So which enzyme do we have? We have amino acyl tRNA synthetase. We have amino acyl tRNA synthetase. So this enzyme will help in the activation of amino acid. So amino acid AMP complex will form. Amino acid AMP complex will form, and of course these phosphates, right, these phosphates will get released. Number one. So after this activation of amino acid, there will be charging or loading of tRNA. What will be there, bache? There will be the charging or loading of tRNA. That's something you have to keep in your mind. Okay? Charging or loading of tRNA. Now, beta, this is your amino acyl, right? So this is your amino acyl. I'm writing it here. Do not get confused here. I am talking about amino acyl AMP complex. Now what's going to happen? This charged amino acid, right? This charged amino acid, amino acyl AMP complex, right? Now we have to load it on the tRNA. Again, the same enzyme will be used here for this process as well, right, bache? So tRNA we have. Now what we have to do? We have to put this charged amino acid on this tRNA. We have to put this charged amino acid on this tRNA. So it will form amino acyl tRNA complex, right? And the AMP will be released here. So you have to understand it in this way. You have one amino acid, ATP is like when ATP will come, right? AMP is joined here. So this is what you people are going to get. This is what you people are going to get, charged tRNA. And how is it possible? With the help of an enzyme. Just imagine if this is that particular enzyme. Just imagine if this is that particular enzyme. So this amino acid, this charged amino acid is attached to this part. Let's suppose this is the charged amino acid. This is attached to this part. Now then what's going to happen? You have the tRNA, right? You remember tRNA. So this enzyme will be like, yeah, I'll bring tRNA. And which loop of tRNA? Your DHU loop. This is the one that will bind with this tRNA. Now the role of this enzyme is to put this charged amino acid here on this tRNA, and that is actually the loading of tRNA. That is actually the loading of tRNA, right? That is actually what, beta? That is actually the loading of tRNA. That is actually what? That is actually the loading of tRNA. So that is how it is happening, bache. So basically the enzyme is same, right? So 20 amino acid, 20 tRNA, and that 20, right? That 20 enzymes we have. 20 amino acid, 20 tRNA, and that 20 enzymes we have, right? Done? Done? Sure? So these are the two steps here initially. These are the two steps here initially. Is that clear, beta? Is that clear? Now you have to synthesize the polypeptide. Now again, the same step, initiation is there. So here for the translation, when I'm talking about the translation of prokaryotes, which is the part of our syllabus actually in detail, so we will have initiation factors 1, 2, 3. Eukaryotes are having different initiation factor. But before that, we have to see that how the binding will occur. Just we have the mRNA, 5' and 3' end. Beta, no doubt it will have the start codon, and that is AUG, methionine. And here at 5' end before start codon, after stop codon, some UTRs are also there. Untranslated regions are also there. Some UTRs are also there. Some untranslated regions are also there. So in the very first step, beta, what is going to happen? Your 30S subunit has to bind with the mRNA. Right? Initially, what's going to happen? Your 30S subunit, it has to bind with the, right? So your 30S subunit, you know, na, it is the small subunit of ribosome. So 30S subunit of ribosome, it has to bind with mRNA. What it needs to do? It has to bind with mRNA. So 30S subunit, it has to bind with mRNA, and initiation factor 3 and magnesium ions will play the role there, right? So then you will get 30S subunit mRNA complex, right? This is what you are going to get. So how is it possible, beta? It's very simple, right? This mRNA, this is 30S. Beta, it is having 16S ribosomal RNA. You know na, ribosomal RNA makes maximum amount of total RNA. It is the maximum of total RNA. So basically, right, with the help of a sequence, mRNA will recognize the smaller subunit of, right? Yes, bache. Right, the mRNA will recognize the smaller subunit of ribosome. So mRNA is having a sequence. That sequence is known as SD sequence. What is the meaning of SD sequence? Shine-Dalgarno sequence. Shine-Dalgarno sequence. Shine-Dalgarno sequence. And here in the 16S ribosomal RNA, you will have anti-Shine-Dalgarno sequence. You will have anti-Shine-Dalgarno sequence, right? So this is the sequence that will help in this binding. Okay, this is the sequence that will help in this binding. But my dear students, in the case of eukaryotes, you will not talk about Shine-Dalgarno or anti-Shine-Dalgarno, right? In the case of eukaryotes, you are not going to talk about Shine-Dalgarno and anti-Shine-Dalgarno sequence, not at all, right? Not at all. In that case, what you people will see in eukaryotes, that 5' cap of the mRNA will help it to bind with the 40S subunit, right? Will help that to bind with the 40S subunit. Am I clear here? Am I clear here? So now, what's going to happen, right? This 30S subunit, right now, this 30S subunit mRNA complex, 30S subunit mRNA complex will bind with tRNA. Which tRNA are we talking about? Formyl methylated, right? Formyl methionine tRNA. formyl methionine tRNA. So you have initiation factor 2, initiation factor 3 and magnesium ion here. So with the help of that, matlab next step what's going to happen? Next step mein what is going to happen? Because you know that it is the methionine which is the start codon. It is the methionine which is the start codon beta. But here in prokaryotes, you will talk about, right, you will talk about formyl methionine. You will talk about formyl methionine. So basically what is going to happen beta, right, after this thing, your 30S subunit mRNA tRNA complex will form. Matlab that tRNA will bind here, right? And for this particular step, energy is required. GTP is required here. GTP is required here. Okay? So ultimately, this is the sequence that we have now. So now your tRNA will come, and after that, now your larger ribosome will come. So now when you talk about the larger ribosome, so it is E, it is P, it is A. It is E, it is P, it is A. These are the three sites that we have to discuss here. So E stands for beta exit site. P stands for peptidal site. A stands for amino acyl site. A stands for amino acyl site, right? Amino acyl site, right? And then there is a groove. Then there is a groove here from where the newly formed polypeptide will come. So exit peptidal or amino acyl site. So after this, the larger subunit has to come. And remember, the first tRNA will always come to the P site, right? Just remember this process. The first tRNA will always, right, come to the P site, right, bache? Right, bache? So once this structure will form, initiation factors will release. Matlab now they will not, no, they are not attached here, they will release, right? They will not stay attached here, they will release. So the first tRNA will come to the P site. The next tRNA, as per the sequence on amino acid, will come here. Now understand one thing. This is your mRNA having 5' to 3'. Start codon is AUG. So obviously, complementary to it, you will have UAC. On tRNA, you will have UAC. But always remember, this is your mRNA 5' to 3' having AUG. So complementary to it, you will have UAC. So remember, this is at 5' end, this is at 3' end. This is at 5' end, this is at 3' end. So that is something you need to keep in your mind. Okay? That is something, right? Done? So it is tRNA. What's that? It is tRNA. What's that? It is tRNA. What's that? Right? It is tRNA. Right? So mRNA, tRNA, it is like that. Now, one point is this that I really want to explain you. Next point that I want to explain you is about the bonding here. Okay, beta? Just say here you have one amino acid, here you have another. Of course, they have to join. They have to form the peptide bond. Just say I'll give you one example. Let's suppose this is your amino acid. This is the basic structure of amino acid, right? This is the basic structure of amino acid. What is this? This is the basic structure of amino acid. Just keep it in your mind, people. So here what's going to happen? The water will come out, minus H2O will there. So after this minus H2O, after the release of this water, my dear people, after this minus H2O, after the release of this water, my dear people, these two will join. These two will join, CONH, right? They have to satisfy the valency, and this CONH is actually your peptide bond. So here, who's going to catalyze that? Number one. Who's going to catalyze that? Number one. That is my direct previous year question. Who's going to catalyze that? That will be catalyzed by peptidal transferase. That will be catalyzed by peptidal transferase. And this peptidal transferase, students, it is a ribozyme here. Actually here, right here, it is a ribozyme. RNA is acting like an enzyme. Your 23 ribosomal RNA, 23S ribosomal RNA is acting like an enzyme here. Am I clear? Am I clear here? Your 20th mRNA will do it. Right? Done? Done? Any doubt? Any doubt? ठीक. अब what is going to happen next? Next is when this next tRNA will come. Beta, all of a sudden something will happen. This amino acid will be like कि यार I want to, मतलब it will break its connection with this tRNA. It will break this connection with this tRNA. Now it will form the bond here with this amino acid. It will form the bond here with this amino acid. I told you how the bond will form, right? I told you how the bond will form. So what is going to happen because of that? Because of that, just see, we have E site, we have P site, we have T site. So because of that, this tRNA will not have amino acid and this tRNA will get dipeptide. This tRNA will get dipeptide. That is going to happen here. Now, what will happen? There is an enzyme known as translocase. Right, it will help in sliding of ribosome. Right, it will help in sliding of ribosome. How will it help, beta? Sliding of ribosome. That sliding of ribosome will be there in 5' to 3' direction. That sliding of ribosome will be there in 5' to 3' direction. Will be there in 5' to 3' direction. Now, when there is the sliding, right, obviously, even parallelly, you know that elongation is going on here. First step here, that the step that I have discussed here, it is regarding the elongation. It is regarding the elongation, right, bache? So one thing is that ki translocase will help the ribosome to slide, but along with that, in prokaryotes, you have elongation factor 2TUSG. 2TUSG means TUTSGH. So they are the elongation factors. They are the elongation factors in prokaryotes, not in eukaryotes, but yeah, in prokaryotes. Baki translocase will slide. So when translocate will slide, will help in the sliding of mRNA, right? When translocase will help in the sliding of mRNA, my dear people, then what's going to happen? What's going to happen? Because ribosome, my bad. Translocase will help in the sliding of ribosome. In which direction? In 5' to 3' direction. So when that ribosome will slide, right, your tRNA will come to, the tRNA which was on P will come to E, the tRNA which was on A will come to P, and the A site will be empty again. A site will be empty again. Where as per the mRNA, a new charged tRNA will come. So this new charged tRNA will come here. Same procedure will be repeated again. Again, these two peptides will move here. Again, your ribosome will translocate. Again, the same procedure will be repeated. So elongation factor 2TUSG will help here like this. Any doubt? Any doubt? And this enzyme also needs GTP for its activity. It also needs GTP for its activity. Is that clear? Is that clear? So how this translation will stop? When it will encounter nonsense codon. So that I told you already, UUU, AG or AGA. Right? So these are the three stop codons. So if anyone will appear, the translation will stop, and there are release factors, beta. RF1, RF2, RF3. They are also known as dissociation factors. So with the help of GTP again, they'll help in this process. Right? With the help of GTP again, they will help here in this particular process. Am I clear? Am I clear? Am I clear? Hanji, doubt? Done? Done? Are you sure about it? So this is all about your the translation. Next topic that we have to discuss and we have to quickly finish it is, right, operants, right? Next topic is operand. This is all about the translation, trust me, right? So now there is one more concept that we have to discuss, but that is not there in your eukaryotes. It is in prokaryotes only. Operon. So here we talk about the year 1961 and we talk about two French scientists. What do we need to discuss, beta? We need to talk about the two French microbiologists here. Right, two French microbiologists here. So that is Francis Jacob and Monod. Right, simple is Jacob. Right, you remember na Jacob and Monod, Jacob's Monod, Jacob and Monod. This is what we discuss. So this is Francis Jacob and if I am not wrong, that is Jacob's Monod. Right, this first name I forgot. So they were the one. Right, in the case of E. coli, they studied the E. coli. They gave that concept of operon here. Right, so basically in the operon, what are we discussing? Dekho, your prokaryotes are polycistronic. Your prokaryotes are what? They are polycistronic. Right, just so these prokaryotes, they are what? They are so prokaryotes are polycistronic. Okay? So basically they studied the E. coli. They got to know about the operon. They studied the regulation of genes in E. coli. Just say we have two types of genes when we talk about the genes here. Right. One will be your constitutive gene. One will be your constitutive gene. One will be your constitutive gene. Right. Another are your non-constitutive gene. Right. So first of all, write it properly. I need to write it properly. Constitutive genes. And the another word is non-constitutive genes. One is constitutive genes. Another is non-constitutive genes. So when you are talking about them, you are talking about the housekeeping genes, my dear people. Here you are talking about the luxury genes. Just for an example, they are always, how a gene expresses itself. Dekho, you know there are different levels of regulation of gene expression. First of all, how a gene expresses itself by making protein. In the case of eukaryotes, we have different levels where we can control it. At the level of transcription, at the level of RNA processing, at the level of translation and all. Okay? Now here we are saying that how a gene will express itself by making protein. How a gene will express itself by making protein. How a gene will express itself by making proteins. I told you already. Now here I am saying that these genes, they are always expressing itself. Our cell cannot survive without them. Right? They are always expressing. Our cells cannot survive without them. Our cells cannot survive without them. Right? जैसे कि gene for glycolysis. Right? Gene for ATPase. Always your cell needs that. Okay? Now they will only express when required. Right? Let's suppose if a gene, right, if a cell doesn't need that particular protein, these genes will not express itself. These genes will not express itself. It will be like that only. Okay? Okay? So now when you are talking, so this is the main concept. So basically Jacob and Monod, they were studying the E. coli. They were studying the regulation of gene expression in E. coli. And then they got to know about this operon. Operon is nothing, beta. It is just like, you can say that, of course, segment of DNA it is, where you will see one regulatory gene that will control the expression of many structural genes. It's a part of DNA which is the single regulated gene, right? It is the part of DNA. What's that, beta? part of DNA or genetic material. It is the part of DNA or genetic material which acts as a single regulated unit, which acts as a single regulated unit, which acts as a single regulated unit, right? Having one or more structural genes, having one or more structural genes, an operator, a promoter, right, and a regulatory gene. Okay, and a regulatory gene. So what do we have here? What do we have here? We have, right, so here there is a single regulated unit that is having one or more structural genes, beta, an operator, a promoter, and a regulator. And what? And a regulator. Are you getting my point? And a regulator gene. And a regulator gene. Is that clear? Is that clear? So scientist name is important. And this part is important. Now, here we have one thing. We have Pipo ZYA या ZYA, Pipo ZYA या Pipo ZYA. देखो बेटा, here when we talk about the operon, right, we just talk about two concepts. One is inducible, another is repressible. So it has a very simple meaning that when you are talking about inducible, by default it's off. By default it's off. Here we have to switch it on. By default it is off. By default it is off. We just need to switch it off. By default it is off. We just need to switch it off. Switch it on, right? By default it is off. This is for an example, lights here in a room, if they are right, if they are, जैसे ये देखो, by default they are off. ठीक है? Wow, even I am not visible. By default they are off. So I have to switch that on. I have to switch that on. Again I'm repeating, it's fun, huh? So by default it's off. I have entered in the room. Wow, if this light is not available, I'm not even visible. Oh my God. So by default it is off. By default it is off. I have to switch it on. I have to switch it on. I have to switch it on. So by default in inducible operon, the genes are not expressing itself, right? And I want them to express inducible, right? Once more, huh? Once more, it's fun. By default they are, by default they are off. Okay, they are off. So I have to switch that on. It is inducible. It is inducible. Now repressible may they are on. I have to switch it off. Okay, I have to switch it off. So to induce something, to start something, it's like that. है ना, HSP sir? Long time no see, sir ji. Right? Okay, so now here, this is the first thing that we need to keep in our mind. Second thing is, right, second thing here is that in our syllabus, we have the negative control or negative regulation of inducible operon. I am again repeating the same point, my dear people. I am again repeating the same point, my dear people. And that same point is what? That same point is that in our syllabus, we have inducible operon and negative control, yeah, negative regulation of inducible operon, right? That is our, right? That is our syllabus. Okay. So basically we talk about the lac operon there. So lac here stands for lactose. Lac here stands for lactose. Lac here stands for lactose. What this lactose is? It's a disaccharide, right? What this lactose is? It's a disaccharide. It is made up of glucose and galactose. It is made up of glucose and galactose. Now, what is happening? We have a cell. This is our E. coli, Escherichia coli. This is our E. coli, Escherichia coli. Now this cell is in a medium, beta, where lactose is present as a food, but not glucose. Now this E. coli is like, I have to survive. Glucose is not there, but lactose is there. I have to make glucose out of it. So then it is the requirement to induce the expression of those genes which can digest the lac operon. Which can digest the lac operon. Okay? Sorry, which can digest the lactose. Now what's going to happen by default, right? First of all, this sequence needs to be clear, right? So by default, what's the scenario? It's PPO-ZY. It's PPO-ZY. I here is not some inhibitor, beta. I here is regulator. I here is what? It is regulator. P here is promoter. O here is operator. Are you getting my point? Are you getting my point? Z here, this is ZYA. These three are the structural genes here. Z is beta galacto. It will give me the enzyme beta galactosidase that will digest your lactose. Y will give me permease that will help in entry of lactose in the cell. A will give me trans-acetylase. Somewhere its role is in detoxification and transferring the acetyl group. These are the three genes. These are the three genes and these are the three enzymes that we will get from this part. Now by default, what is happening? Your I, regulatory gene, is forming regulatory mRNA that is forming a protein. Beta, actually here this protein is a repressor. Here this protein is a repressor, right? It is a repressor. It binds with the operator here. This protein binds with the operator here. So you have to remember few things here. And what is that? First of all, the sequence is PPO-ZYA. The sequence is PPO-ZYA. What is the sequence here? It is PPO-ZYA. It is PPO-ZYA. It is PPO-ZYA. PPO-ZYA. Promoter and operator, they are overlapping. When it comes to promoter, right? P for promoter, P for polymerase. RNA polymerase bind with it. Number one. O for operator. O stands for operator. So the product of regulatory gene binds here. Product of regulatory gene. Now this I, regulatory gene, is having, it has formed an mRNA and a protein. This protein, my dear students, is a repressor, right? And where will it bind? As I said, the product of regulatory gene, it always bind with operator. Product of regulatory gene will always bind with operator. Product of regulatory gene will always bind with operator. Promoter and operator, they are overlapping. Now, operator is having a protein. Because of that, the polymerase is not able to bind with the promoter. The transcription is not there. Now, if I have to switch that O on, if I have to switch that on, then what am I supposed to do? जैसे मेरे पास है यहाँ पे P, I, P, O, Z, Y, A. So, A, right? So, this I is forming mRNA. This I is forming protein. This protein is attached here. But I want to switch on the expression. So I want this, right? I want this repressor protein to not bind with the operator. So for that, I need something. Matlab, if I want them to have breakup, I need to implant something. If I want the promoter and repressor, sorry, operator and repressor to have breakup, I need to bring a third person here. So here, the role of inducer will be there. Inducer itself is lactose, bache, and it is also known as allolactose. It is also known as allolactose. Matlab, more correctly, it is allolactose, right? It is basically regarding the isomers. So more correctly, it is allolactose. So this lactose is the inducer. This lactose will bind with this repressor. So now the repressor is distracted. Repressor will not be able to bind with operator. So your RNA polymerase will come, right? Your RNA polymerase will come and that will bind here with the promoter and will help in the formation of this polycistronic mRNA, and due to which you will get Y will give you beta galactosidase, Z will give you beta galactosidase, Y will give you permease, A will give you transacetylase, right? So that is what the operon system is. So this lactose is digested by beta galactosidases that will give me glucose and galactose. That is going to give me glucose and galactose. Clear? And as I said, permease allow the entry of lactose in cell. Okay, so now if you are thinking that if permease will allow the entry of lactose in the cell, and if lactose is an inducer, right, then how the things will work out here, है ना? How the things will work out here. So it's simple, my dear students. It's simple. Some amount of permease will always be there. Some expression of permease will always be there, and due to which some amount of lactose will enter here, will act like an inducer, and then will start the switching on of these genes and their respective products, right? Now here, because your regulator is forming the repressor, because here regulator is forming repressor. So that is why, right? Why here? Because here regulator is forming repressor. So that is why it is the negative regulation of inducible operon, या negative control of inducible operon. Yeah, it is negative control of inducible operon. So that is it. This is all about the topic. This is all about the translation. Of course, the notes will be shared with all of you. So I hope after that notes, you will be able to understand the things properly. Okay? Okay? Done, beta? ठीक है. बाकी next topic is HGP. HGP is a theoretical topic. One question is for sure expected from HGP. So first of all, तो these points which are written there ना, in the case of HGP, देखो, question will come from methodologies of HGP. So EST stands for expressed sequence tagging, because in HGP you are basically sequencing the whole genome, sequencing the DNA sequence. So one way is expressed sequence tag, one way is expressed sequence tag. So when I'm saying expressed sequence tag, I am talking about the gene which has expressed itself, which has formed the genes which have formed the mRNAs. So here we focused on identifying all the genes that are expressed as RNA. Second is sequence annotation, where you have to pick up the entire genome, where the sequencing of the whole genome will be there. And of course, that is again a previous year question. Which vectors will be used here? Bacterial artificial chromosome and yeast artificial chromosomes, because they can accommodate the genes of, yes, large, the big genes basically, right, having, you know, that number of base pairs, more number of base pairs. Now this information is important, right? Baki, that's it, right? So HGP mein, this is the main topic that you have to cover. So bache, we have completed the molecular basis of inheritance, right? We have completed all the essential things there, right? We have completed all the essential things here. And now, right? If I have to, right, no doubt, evolution is a time-taking chapter, and I really love to teach this topic because this topic is amazing. And this is what we don't have, that is time. So I am directly coming to the most important topic, जैसे we discuss about the operon and Haldane theory, right? Operon and Haldane theory, right? It is the theory of evolution of life. It is a theory of chemical evolution, naturalistic theory, whatever you want to call it. Operon and Haldane theory. Right, Operon and Haldane theory. If you remember, it is HERO. Haldane was from England, Operon was from Russia, right? Russian scientist. So hero is the trick here. So when you talk about this Operon and Haldane hypothesis or Operon and Haldane ki theory, right, bache, naturalistic theory, right, chemical theory, right, bache, theory of origin of life it is. So there is an experiment that supports that, right? It is Urey and Miller's experiment. right? It's ore. So this hypothesis, matlab, as per, right, I'll directly pick up the important questions here, that if I talk about operon and Haldane's theory, right, operon and Haldane's theory, right, so here they discuss the evolution in two ways. Firstly, they talk about the chemogeny, then they talk about the biogeny. When they say chemogeny, right, they are talking about the chemical evolution. As per this theory, on planet Earth, earlier conditions were like that. It is possible that the chemicals, they came first and then they developed into the cell, right? That chemogeny is possible. Chemogeny is possible. Chemical evolution, right? Bagi that factors are common. Ki life will start in the water, right? Obviously, firstly atoms will form molecules. That is the complete story. And obviously, earlier atmosphere pe, primitive atmosphere pe, reducing conditions were there. Oxygen was less, hydrogen was more. So reducing conditions were there. Number one, the temperature was very high there, volcanic eruptions and all of that was going on. So they say that firstly, of course, with the time, atoms, they formed molecules, they formed, elements, compounds, inorganic, organic compounds. So when it comes to the water and ammonia, they are the first inorganic compounds that we consider. Methane is the first organic compound. So inorganic compound came, then organic compound came, simple, complex, slowly together they form some protobionts, protocells, which are not properly the living cell, but they started growing. They then started acquiring the division ability as well. So that is what we discuss. And to support that, we talk about the Fox-Kim microsphere. And operon has given the co-servates, if you remember, right? So they are the kind of protocells, protobionts. They are not the living cells. They are somewhere synthesized in lab, right? So they are just showing that aggregation ki these, these polymers, they came together and they started showing the ability of growth and all. This is what we discuss. So then comes the biogeny. So now here, what is important? This. So who supported it? Urey and Miller's experiment. Urey and Miller's experiment. Right? So they supported it, they checked it, and they were like, yeah, it is possible on early Earth to have the chemical evolution first. So we talk about this particular diagram here, my dear people. So you know that it is their experiment. They started boiling the water here. This is the chamber. So this experiment is having this gas chamber, condenser. Here it is the one from where they will trap the liquid water. Now they are boiling the water. Electrodes are there. They are maintaining the temperature around 800 degrees Celsius. Now here in the gas chamber, what have they done? They have your, methane. They have your ammonia. They have the hydrogen as well. They have the H2O as well, because water is also getting boiled. So these are the three gases that we need to keep in our mind. Ki methane, ammonia and H2O is there. Which three gases do we have here, beta? Which three gases do we have here? Right, which three gases do we have here? We talked about the methane, ammonia and H2. Now here, remember one thing, right? Remember one thing, right? That A for ammonia, that's how you have to remember. A for ammonia. Ammonia is the first letter. So ammonia is in one part, methane and hydrogen is in two parts. That is the story. That was the composition that they took. That was the composition that they took. Are you getting my point? So what they just said, let's suppose if I'm writing it like this, ammonia, CH4, H2, then you will write 1:2:2, right? So one thing that we need to keep in our mind, that when it is the ammonia, one part of ammonia, two part of methane, two part of hydrogen will be there. One part of ammonia, right? Two parts of methane, two parts of hydrogen will be there. So this is something that we have here in this composition. We have the gases here, right, bache? Then after that, what is happening? Because of this discharge electrodes, the temperature is also very high, right, bache? So what is going to happen right here? Then after this, it will all, because water vapors are also there, pass through this condenser, right? And here the water will cool down. So basically this experiment, it took 18 days, right? And here, right, they obtained some amino acids. What they obtained here? Amino acid. Just aspartic acid, right? Your aspartic acid was there, your alanine was there, one more amino acid was there. I'm not able to recall the name right now. So one more amino acid was there. Okay? One more amino acid was there. So one is alanine, another is aspartic acid, third one ka I am not able to recall the name. ठीक, okay, done. Done. So aspartic acid and that alanine is there in their mind, in my mind. So maybe there is glycine also, right? This is the something that I'm forgetting right now, glycine. So what is happening? What happened here? So when they, like, after this experiment, as I said, 18 days, 800 degrees Celsius, the composition of gases is important. So when they checked the water, then they observed that here in this water, amino acids are there, organic compounds are there. So organic compounds, they developed from that inorganic compounds, developed from that gases wala part. So it means, it is clear that if here in the lab, if we can create such conditions and we can get the organic compounds, so definitely it is possible on the earth that, you know, at that time, because volcanic eruptions and all, lightning was also there, so source of energy was there. So yeah, it is expected, right? It is expected to have what? To have this chemogeny and then biogeny. The next important question is HDAC. HDAC, homologous divergent evolution. Homologous organs will show divergent evolution. Analogous will show convergent, to converge, to meet. This is, let's suppose if we have the same ancestor and two different species evolved. Two different species evolved because they, you know, settle down in some different conditions. So they evolved as two different species. So that is the example of divergent evolution. So here what is the scene? In the homologous organs, their fundamentally a basic structure is same. Their fundamentally a basic structure is same, but functions are different, right? But functions are different. Remember this thing, right? What is different then? Sorry. So what happened here? So here HD it is. So basic structure is same. Basic structure same, but functions are different. Basic structure same, but functions are different. That is homologous organ. ठीक है? Analogous में basic structure is different. So structure different, function same. I'm writing it in a simple way now. Structure different, but function is the same. Structure different, but function is the same. Are you getting my point? So we have homologous organ, we have analogous organ. We have homologous organ, we have analogous organ. We have homologous organ, we have analogous organ. So basic structure same, but functions are different. Structure different, function same, right? So here ancestry is different, but they are converging at one point. But they are meeting, they are surviving in the same conditions. They are surviving in the same conditions, right? So they are living in the same conditions. So that's why adaptations are according to that. So that is why when we talk about homology, right? Homology, divergent evolution, ancestry is common, basic structure is common. You can compare yourself with a dog, our forearm, humerus, radial, this and that, they are also having the same, right? You can even compare that with the amphibians, right? So basically I'm saying the ancestry is same here, but here ancestry is different. So they are similar in basic structure, but they have, right, adapted to different things. So their functions are different. Here their functions are similar, right? So homologous, analogous organ mein examples are very, very, very important, beta. Examples are very, very, very important. Do not miss that, right? Do not miss that. So jaise ye dekho, you have thorns of bougainvillea, right? It is thorn. Here the tendril of cucurbita. Do not say that bougainvillea is having tendril. So it is like, remember it like this, TB. So thorn in bougainvillea, right? Thorn in bougainvillea. So cucurbita is having tendril. Basic structure is same, function is different. So man, cheetah, whale, bat, humerus, radioulna, it's all same. So vertebrate ka heart, vertebrate ka brain, right? So basic structure is same, function is different. Basic structure is same, function is different. Ab analogous mein wings of butterfly and birds, they'll help in flying, but birds mein that wings will have feathers. In butterfly, that is just the membranous wing. Eyes of octopus, right, and mammal, they'll help to see the object, but the basic structure is different. Flippers of penguin and dolphin, and then sweet potato and potato, right? They all are, the question will come from the examples, right? The question will come from the examples. So sweet potato is beta root modification, and potato is the stem modification. So structure is different in both the cases, right? The structure is different in both the cases, but their function is same, that is storage of food. So that is how you have to remember this part, right? Next comes the adaptive radiation. So we somewhere consider this adaptive radiation and divergent evolution same. But here in adaptive radiation, you have to focus on the geography as well. So when it is the adaptive radiation, yes, so basically there is the evolution of different species, but in a given geographical area, that's the main point, right? But in a given geographical area. So there is one ancestor, different species will evolve, right? In the given geographical area, starting from a point, and then they radiate to other habitats. So basic ancestor is common, the geographical area is common, but different species will form, then they will move to the different habitats, right? Then they will move to the different habitats. Is that clear? Is that clear? So famous example is Darwin finches, right? So they all evolved from seed-eating birds in Galapagos Island. So their beaks, they were altered, right? They were altered. So some become insectivorous, some became vegetarian, like this, right? Baki, this adaptive radiation in marsupials, matlab their area. You are talking about the marsupials, right? Right? We are talking about the marsupials here. Marsupials. I'm not comparing them as of now. I'm simply saying when you talk about the marsupials, right? We know that where are they present. In the case of marsupial, also in the given geographical area from these ancestors, they radiated in different directions. Just say for an example, Tasmanian wolf, tiger cat, banded anteater, marsupial rat, kangaroo, wombat, bandicoot, koala, marsupial mole, sugar glider. So sometimes they ask the question like this, that sugar glider, is it a marsupial or is it placental? Question like that can come. So this is important for NEET. This is important for NEET. Okay? So other than that, so this is adaptive radiation. Placental mammals itself is the example of adaptive radiation. Your marsupials are also the example of adaptive radiation. Now what are we doing? We are just talking about the convergent evolution. Yeah, adaptive convergence it is. Adaptive convergence it is. Now, in the adaptive radiation, you were simply saying that in a given geographical area, the species were arising in different direction from the common ancestor. Now here when more than one adaptive radiation, when more than one adaptive radiation, when what beta? When more than one adaptive radiation, it occurs in different, matlab in adaptive radiation, you were talking about a given geographical area, different species are evolving. But here what are you doing? You are saying that two areas, they are totally different. Two habitats, they are totally different. What are you saying here? You are saying that two habitats, they are entirely different, right? But we call their convergent evolution, right? Just placental mammals you have, Australian marsupials you have. Individually, if you talk about them, placental mammals, in a particular area, in a given geographical area, they radiated. Then it is also adaptive radiation. Marsupials, adaptive radiation. But now you are comparing them, right? Individually, they are the examples of adaptive radiation. But now you are comparing them because what has happened? Their geographical areas are different. Their habitat is basically different. But still, right, when they evolve, right? So when more than one adaptive radiation appeared to have occurred in isolated geographical area. So just say placental mammals ka mole, simple to marsupial, anteater, numbat, mouse, marsupial mouse, lemur, spotted cuscus. So you need to compare that. MCQ can come from this part. MCQ can come from this part, right? MCQ can come from this part. बाकी बेटा Lamarckism में nothing much, use and disuse of organs and inheritance of acquired character was there. He was saying there is a will that because of that organisms they evolve and the organs that we use more, right, that will be better developed, that will pass to the next generation. Baki Darwin ne, you know that he gave the concept of natural selection, right, that nature is going to select the best. So here I will pick up 2 points directly from the NCERT. I will not discuss these things in detail, to be very honest, right, because we'll just pick up the most important points here because then we have to move to the biotechnology. So Darwinism. So yes, right? Even the theory that we accept today, it is also neo-Darwinism, new Darwin. So that contains natural selection, that contains mutation, that consider everything. But here when we talk about the Darwinism, yes, right, he talks about he has discussed the natural selection, that nature is going to select the best. Whosoever is going to survive in the changing condition, nature is going to select that. Nature is going to select that, right? That is true. So now here when you are talking about the Darwinism, so Darwinism is based on two key concepts. Darwinism is based on two key concepts, branching descent and natural selection. That is purely NCERT, my dear people. So it is like, it explains that all the organisms are modified descendants of previous life form, branching descent. But this is the ancestor, new organisms are coming with modifications, then new, then new, then new, then new, like this. Right, like this. So it is saying what? It is saying that all the organisms are modified descendants of the previous life forms, and nature will select the one which will survive. They will reproduce, they'll pass their genes to the next generation, and they will survive. So nature will select, and it depends upon the fitness. Okay? Now, this natural selection, direct MCQ will come from this part, right? So natural selection mein beta heritable variations which will pass to the next generation, they enable the better survival, right? The organism will reproduce, they'll pass that to next generation. So here what do we have? What we have to discuss, right? We have three types of natural selection. What do we have, beta? We have three types of natural selection here. Hanji, anybody having any doubt? Anybody having any doubt here? Anybody having any problem here in this topic? I am beta picking up the most important topics, right? I am literally picking up the most important topics. So you tell me, do you have any trouble here? Do you have any trouble here? I will just pick up the most important points, and trust me, they are needed for you right now, right? Right now it is needed for you, because otherwise so this topic we have a lot in this topic, but we have to stick to certain important topics only as we are running out of time. Chaliye. So we are talking about the natural selection here. We have to talk about the types. Jaise we have stabilizing, we have directional, we have disruptive. Stabilizing, directional, and disruptive. Stabilizing, directional, and disruptive. These are the three types of selection, and we will have to discuss the examples as well. So when you are talking about the stabilizing, right? Stabilizing is kind of balancing also, but directional is kind of progressive also, right? Right. So that's what we have to discuss. First of all, you have to see a kind of diagram here, kind of graph here. So if you will see, it is something like that. This your x-axis, this is your y-axis. It is showing number of individuals with a particular phenotype. It is showing number of individuals with a particular phenotype. Number of individuals with a particular phenotype. Right? It is showing number of individual with a particular phenotype. Okay? This is a graph that we have. Now when you look at its corners, this sides, beta, we consider them as extremes. I'm trying to explain it, but in a very simple language. Jaise ye dekho, that's something that you have. So here you have number of individual having that particular phenotype. What do you have there? Number of individuals, right? Number of individuals with a particular phenotype. Now, whenever you talk about it, just remember, when you talk about these sides, they are telling us about extremes. What kind of extremes are we talking about here? Just say for an example here, you are talking about the homozygous dominant, right? You are talking about the homozygous dominant. Here you are talking about the homozygous recessive. Here you are talking about the homozygous dominant. Here you are talking about the homozygous recessive. And in between, what will you discuss? You will discuss about the heterozygous. What will you discuss in between, beta? You will discuss about the heterozygous individual. So दोनों side homozygous dominant, recessive, heterozygous. मतलब this one if I have to express AA, this one if I have to express AA, and this one if I have to express, it will be this. It will be this. Now we have three types of evolution. How many types? Three types. So number one is stabilizing, which is balancing as well. In this stabilizing one, you will see, matlab, if you will look at that graph, the peak is getting narrower. The peak is getting narrower. Why, beta? Why peak will get narrower here? If your extremes will be eliminated, right? Matlab, here in stabilizing, it will prefer average phenotype. Average phenotype is preferred here, right? It prefers average or normal phenotype. Then extremes. Extremes will not be preferred. Matlab you can take the example of height in our case. Not too tall, not too short. Average height is preferred here. Right, bache? Right, bache? Average height is preferred here. Right. So can I say that peak, see, as I said, peak will get narrower. Peak will get narrower. है ना? Mean value. Mean. Here you are talking about what? You are talking about the mean value. It is NCERT language. So mean value, या mean character value is acquired by more individual. Mean value, या mean character value is acquired by more individuals. It is acquired by more individuals. So mean is preferred here. Average phenotype is preferred here, right? Not the extremes. Average is preferred here. So constant environment is required here. So as I said, you can even discuss the example of human height here, right? We can discuss the example of human height here, that average human height is preferred than the extreme. The weight of babies, that example we used to discuss, right? So here you will see that environmental conditions are constant. So remember, peak here will be narrower. So if somewhere you are talking about the narrower peak, you are picking up the stabilizing selection. You are picking up the stabilizing selection. Then there is the directional, as the name is suggesting. Matlab the evolution is towards a particular direction. It is the progressive one, right? So it will favor one extreme value. देखो ना, it favors one extreme value. It will favor extreme, but one, and eliminates. And here only average will be preferred, and eliminates other extreme and average value. So it will eliminate extreme and average, because the evolution is there in one direction only. The evolution is there in one direction only. So obviously after this selection, the mean value will always change. So peak is shifted to one direction. More individual will acquire value more than mean character value. So you can write down कि after this selection, mean value change. Mean value change. These are the diagrams that we have studied a lot. Sorry, these are the examples that we have studied a lot. So mean value change. Mean value change. Mean character value या mean value always change. ठीक है? So peak shifts in one direction. So more individual will acquire value other than mean character value, right? So I will write it here. So peak shifts in one direction. So one character will acquire. So one character will acquire more value, right, than the mean, because mean value will change here. So one individual, more individuals here, you can say that, right? More individuals will acquire value other than mean character value. So one character will acquire value other than mean character value. So other than mean here. So here mean is preferred other than mean character value other will be preferred. So peak will shift in one direction. So organisms will acquire value other than mean character value, right? So they will not acquire the mean, they will acquire, right? They will not acquire the mean character value, rather they will acquire the other value. Now here also you can see the same story, right? Here also you can see the same story, beta. You can see this is from directly from the NCERT. So here they have clearly mentioned peak is getting narrower and higher. So it's simple. We are talking about the stabilizing selection here. We are talking about the stabilizing selection here, right? Then peak shifts in one direction. As the name is suggesting, directional value, natural selection. So individuals will acquire value other than, here mean is preferred, here other than mean character value, here other than mean character value, and here basically in disruptive, how many peaks are we going to get? 2, right? How many peaks are we going to get? 2. Why? Because here the average will be eliminated, extremes are preferred. Extremes are preferred. Matlab, basically what will be preferred, beta? Either homozygous dominant or homozygous recessive, right? Either homozygous dominant or homozygous recessive, but heterozygous will not be preferred. So directional may your industrial melanism is the example. Evolution of horse is also the example of this directional one. Evolution of horse. So industrial melanism, evolution of horse, these are the examples here, right? These are the examples here. And here what will you see? That extremes are preferred, right beta? So average will get rejected. So more individual will acquire peripheral character value, right? So average is eliminated. So of course this is periphery. Periphery always show, I told you, periphery will show you the extremes. So peripheral characters value will be preferred. Peripheral character values will be preferred, right? So here the example that we have is the shell pattern in limpets. Shell patterns in limpets. What is the example that we have? Shell pattern in limpets. Matlab limpets, what are they? It is a mollusca, beta. It's a sea mollusca. It's a marine mollusca. Okay. So in this case, what will you see? Right? They have, right, white to dark tan. Matlab they are white to dark tan. This type of shell pattern we will see. Right? White to dark tan. So white wala will be camouflage. Matlab white wala will be show camouflage with white barnacles. It will show camouflage with white barnacles, right beta? And when you talk about the tan one or the dark one, right, they are also protected with tan colored rocks. But now if there is something in middle, if there is something which is showing the average phenotype, if there is something which will show the average phenotype, that will not be preferred. Right, bache? They'll be conspicuous. Prey are going to recognize them. They will destroy them. That's it. So, one important question from this part. Okay, I have picked up the important here only. Rest, the notes will be shared. Next is Hardy-Wingwall law and genetic drift. Chalo. So Hardy-Wingwall law, first of all, it is applicable on large population. If you think that we can apply it on the small population, no, the population has to be very large and there are certain assumptions here, number one. Secondly, in this case, when we read about this Hardy-Wingwall equilibrium, na beta, it is the population genetics. It is the part of population genetics, right, bache? It is the part of what? It is the part of population genetics. And here you will see that we have to assume certain things because we have to see the deviation. We have to see the evolution. Just say, let's suppose there is an area. I have applied certain conditions. Right? And that conditions are telling me that no evolution is there. Accordingly, let's say I'm getting a value. Now, I have applied all the conditions, right? Where I'm saying ki evolution should not occur, right? I have applied certain conditions where I am believing that evolution should not be there. Yeah, evolution should not occur, something like that, right? I have applied certain conditions. Okay. Now, but still, when I check the actual value, But theoretically, I have applied certain conditions which are saying the evolution should not occur. But when I check that practically, so I'll see that some deviation is there from my value. That will tell me the extent of evolution. It is like that, right? It is like that. So when you talk about this Hardy-Weinberg equilibrium, right? So this is what we have to believe, that there is no gene migration, right? This is what we assume, that gene migration in any format is not there, right? Right? So here too they are explaining the gene migration, ki migration of section of population to another place, right? Right. So gene frequency will change in original because some people they move to the different place. So basically when it is Hardy-Weinberg law, there are certain exceptions, right? There are certain exceptions. There are certain, I would say, assumptions, not exceptions. I would say there are certain assumptions that are followed here, right? That we need to believe in. Okay, that we need to believe in. So out of that, first one is of course this one, ki gene migration is not there. No gene migration, no gene flow. You know that naturally it is not possible. They are saying no genetic recombination. Matlab we are not considering any sort of variation. So we are saying there is no genetic recombination, right bache? Then we are saying no mutation. Matlab these are the assumptions that we have to take, right? These are the assumptions that we have to take. There is no migration, there is no recombination, there is no mutation, and there is no change, no genetic drift is there. Genetic drift is basically applicable in small population, right? Genetic drift is basically seawall rate effect. You talk about the founders effect, you talk about the bottleneck effect there. But here it is applicable in large population, and we are saying no gene migration, no genetic drift, no mutation, no genetic recombination, and even, right, the natural selection is not there. So even we are saying there is no natural selection. So this is under assumption. This is under assumption, right? This is something we have to assume. So imagine if a population is not migrating, not showing genetic recombination, right? Not showing any sort of variation, not showing natural selection. It simply means the population is not at all evolving. It is not growing. It is not evolving. That is the meaning here. Okay? That is the meaning here, right, bache? So here, what do you consider? Ki allelic frequencies in a population are stable, constant from generation to generation. ठीक है जी. Gene pool, मतलब total genes and their alleles, they remain constant. Constant. Matlab these are some condition. Here you talk about this. P square plus 2pq plus q square is equals to 1, or p plus q is equals to 1 as well. So when you are talking about this, you are talking about the genotypic frequency. What are you considering, people, here? You are considering genotypic frequency. You are considering the genotype here. Here, what are you considering? You are considering allelic frequency. You are considering alleles here. So genotypic frequency, allelic frequency. Just let's suppose in a population there is a gene A and A. So P will be my frequency of dominant allele. A is the, sorry, then Q is the frequency of recessive allele. So of course, here you are talking about a particular gene, or I would say allele. Here you are talking about allele, right? So जैसे मैंने कहा, as I said, P is showing the frequency of dominant allele, Q is showing the frequency of recessive allele. When you are applying this formula, you are talking about the genotype. मतलब P2 is showing AA, 2PQ is showing this, Q square is showing this. Are you getting my point? So this is the genotype. Because you know that in an individual, right, there will be the pair of alleles. Am I right? Yes. You are talking about the genotype here. Here you are talking about the allele. So as I said, P is the frequency of dominant allele A. Yeah, any dominant allele. Q is the frequency of recessive allele. That is what we need to understand. And if you are not able to understand 2pq, let me tell you, जैसे मान लो here you have pq, here you have pq, so it will be p2, it will be pq, it will be pq, it will be q2. So p2 means aa, pq मतलब one is a, one is a, one is a, one is a, right? So that is how we consider that. That is how we consider that. So it is p2, right? It is p2, 2pq, q2. So p2 is the frequency of individuals with the genotype this, right? So frequency of individuals with genotype AA is P2. So frequency of individuals with genotype AA, frequency of individuals with genotype this, right? That is how we check it, right? This is the meaning here, okay, right? So we will, whenever you will measure the frequency, the values will be different, and of course when values will be different, it will indicate the extent of evolutionary change. It will indicate the extent of evolutionary change, right? So you can solve the questions. I have already shared some PDF in the Telegram group. Now for the human evolution, from so long you are not getting the question out of it. One question can come from the evolution, one question can come from the cranial capacity. One second, student. One question, right? One question comes from the cranial capacity. One question can also come about their nature, right? Jैसे Homo habilis, tool maker they are, erectors, erect man they are. They can ask you which one is vegetarian, which one is non. So it will be all as per NCERT, right? Don't worry about that. So if I talk about the sequence, there is a trick, beta. Bolo dara H2HENFS. Please repeat. Dara H2HENFS. Dara H2HENFS. What do we have there? Dara H2HENFS. Dara H2HENFS. What is the meaning here? D stands for Dryopithecus. Dara H2HENFS. R stands for Ramapithecus. A stands for Australopithecus. Before this Australopithecus, prior to ape man. Australopithecus ape man. H2 matlab 2 times H, Homo habilis, tool maker. HE, Homo erectus. There are a lot of things that we have here in human evolution. We have to stick to the NCERT. So NFS mein kya hai beta? You have to remember ourselves, that is Homo sapiens. So it will be Homo sapiens, and stands for Neanderthalensis. ठीक है? So it is Homo sapiens fossilis. Homo sapiens sapiens. So that is the sequence that you can remember. जैसे we are Homo sapiens sapiens. ठीक है? So Dara H2HENFS. Dara H2HENFS. Right? Repeat it with me. Dara H2HENFS. Everybody. Dara H2HENFS. Dara H2HENFS. Yes, please. Dara H2HENFS, everybody. Right? Everybody. So Dryopithecus, Ramapithecus, Australopithecus, Homo habilis, Homo erectus, Homo sapiens neanderthalensis, Homo sapiens fossilis, Homo sapiens sapiens. So this is the sequence of NCERT. Right? This is the sequence of NCERT. Right now focus on that part only. That's it. Right now focus on that part only. That's it. Okay? That's it. Chalo, let's move to biotech. So what do you want me to start? Biotech or or ecology, whatever majority will stay, I'll go for that. Biotech or ecology, what you want me to start? Whatever you people will say, I'll suggest, I'll start. Whatever you people will say, I'll start. It's all up to you guys. It's all up to you. And but whatever we have discussed in evolution part now, we have covered the maximum important topics, right? And if we'll get time, I'll discuss the other one too, because evolution is my favorite chapter. What do you want me to start first, ecology or biotech? बायोटेक, अरे वाह यार, क्यों इकोलॉजी नहीं पढ़नी क्या? यू डोंट वांट टू स्टडी इकोलॉजी? चलो, इट्स ऑल राइट. Okay, so let's start the biotechnology. Let's start the biotechnology, okay? Doubt? Any doubt? Any doubt so far from molecular basis of inheritance, from evolution? Chaliye. So beta, when it comes to the biotechnology, you are simply talking about two things, right? We will see that how do we mix the technology and the living organism. Why do we need to do that? Of course, for our benefits. So whenever you talk about the biotechnology, right, whenever you talk about the biotechnology, you will go for old biotechnology and the new biotechnology. We will go for old biotechnology and we will go for new biotechnology, right? Old biotechnology and we will go for new biotechnology. So when it is the old biotechnology, when it is the old biotechnology, so here you are talking about the natural capabilities. You are simply talking about the natural capabilities of organisms, right? Jaise, let's suppose Aspergillus niger is giving you citric acid and all. When it is the new biotechnology, we are considering the genetic engineering here. We are considering the rDNA technology here. So genetic engineering and rDNA technology, that is what we consider here, right? That is what we consider here. This is the point that we have. So now when you talk about the right definition here, will it be there in the paper? Yeah, it can come. So European Federation of Biotech, that is your EFB, right? European Federation of Biotech, that is your EFB. EFB. Can we get the question from this part? Definitely we can. So when you talk about the European Federation of Biotechnology, that is EFB, so it is saying that it has encompasses both traditional as well as modern molecular biotech, right? Both are considered. Just I said, old biotechnology and the new biotechnology is there. So here they are saying that we have to integrate the natural sciences, physics, chemistry, biology, everything. We have to integrate that natural sciences, physics, chemistry, biology. This is what we are talking. And then, right, when we integrate that with the living organism. So when you are talking about the living organism, you will have to consider everything, right? You are basically integrating the natural sciences with the living organism, right? And of course, we are doing it for their right products and all, whatever products we can get from that, right? We will be able to develop it with the help of natural. When we combine these natural sciences and living organism, we'll be able to get that products also, right? So this is what we have when we talk about the EFB. So what exactly is going to happen in this particular chapter? You have to talk about the genetic engineering or rDNA technology. Let's suppose there is a DNA. There is a gene which you guys have targeted. This is the gene that I need, right? So restriction, this is the gene that I need. So with the help of some enzymes, we'll cut that gene. We have to insert it with the other, right? We have to insert it with the other DNA, and this is nothing but the plasmid, the extra chromosomal DNA of bacteria. When you join it, you get the recombinant DNA, you introduce that recombinant DNA in a host cell, there it will multiply, there it will express, and now my host cell is having that new or that particular gene. This is what I have to study in the rDNA technology. So when I talk about the rDNA technology, what do I consider? Two things: genetic engineering and chemical engineering or bioprocess technology. What exactly it is? These are the principles of biotech. These are the principles of biotech. So when you talk about the principles of biotech, right, genetic engineering is there, right, and bioprocess engineering, which is also known as maintenance of sterile, contamination free ambience and chemical engineering process, right? This is what we have to maintain here. So in simple words, if I have to tell you that when I talk about the genetic engineering, basically I have to alter my genes. I have to alter the DNA or RNA. I have to alter the genes, I have to alter the DNA and RNA. So it is a technique by which we'll be able to alter the DNA and RNA, right? And once they are altered, of course their alteration will only be fruitful if we will get the desired gene, if we will get the desired product. So for that, what we have done, right, for that what we have done, just have a look. So techniques will alter the chemistry of genetic material, my dear students, to introduce these into host organism and to change the phenotype of the host organism, and to change the phenotype of the host organism. What we have to do here, we have to change the phenotype. So when it comes to the bioprocess engineering, chemical engineering, which is also the maintenance, we have to maintain certain contamination free condition, right? Why? Because see, if we have altered the DNA, if we have inserted it into a host, then we want this host cell to survive. We want this host cell to express itself. We want this host cell to make that desired protein. So that is why bioprocess engineering is also important. Okay? Now if I talk about this part, they have generally given us the basic things. Sexual reproduction is better than the asexual reproduction, because sexual reproduction will result in the variations, and that variations are very important for our survival. That variations will help us to adapt, right? That variations will help us to adapt to the change. That variations will help us to live. Okay, so asexual reproduction is very good. I believe from this line, if question will come, students can get confused. That's why I'm targeting this. If it is the asexual reproduction, it preserves the genetic information, but sexual reproduction will permit the variation. So if in a paper their question is which reproduction will preserve genetic information, don't be like DNA will preserve genetic information. Asexual reproduction will also preserve genetic engineering, genetic information, because there will be no change, there will be no variation. Whatever is the DNA, it will pass as such to the next generation. It will pass as such to the next generation. So that is the point that we have. And when it is the sexual reproduction, the variations are there. The variations are there. Are you getting my point? Right? So in genetic engineering, we have to discuss about the rDNA. We have to talk about the gene cloning and the gene transfer. Okay, beta? And the gene transfer. Right, bache? Kyunki why? Why this genetic engineering is important? Why can't we rely on hybrids? Let's say if there is a superior male or there is a superior female, they will give us the hybrid, let's suppose. So that hybrid will have the characters of both the parents, right? So why are we focused on the genetic engineering? Because bache, this hybrid, no doubt it will have the character of both the parents. No doubt it can show us that desirable character of both the parents. But along with that, some undesirable characters will also be there. Along with that, there will be certain characters which we don't want that will still be there in the hybrid. But in genetic engineering, this is the benefit that we have. Okay, this is the benefit that we have, that we can manipulate the genes. If you want something, you can introduce, but if you don't want something, right? But if you don't want something, you can just simply delete that gene as well. So in one go, you will get a new combination, entirely new combination will be there with you. So that is the point here. That is the point here. Take it, beta. Now, we have to understand the discoveries here. Foundations of biotechnology. Next topic that we have to discuss is what? It is the foundation of biotechnology. Right now, whatever information I'm going to give you, it is there in NCERT. Like when you start this chapter, you will see some scientists and their description. है ना? Some scientists and their description, right? मतलब their discoveries and everything, na? So basically, whatever information now I am going to share, that is basically from that part. ठीक? That is basically from that part. ठीक है? Right? That why, मतलब what are the foundations? What is the foundation of this? Genetic engineering. So this is what I am discussing, right? And this is what I'm about to discuss. So I don't think that I have added that from the NCERT, but don't worry, I'll show you that. I'll discuss that with you all, so you can just give it a reading. Okay? You can just give it a reading. So when you are talking about the foundation of biotechnology, you talk about two things. One is plasmid, another is restriction endonuclease. One is plasmid, another is restriction endonuclease. So one is the presence of plasmid in bacteria and the use of restriction enzymes in vitro. Number one is presence of plasmid in bacteria. Then second point is that restriction endonucleases can be used in vitro. In vitro means in lab. In vitro means in lab they can be used. This is the point that we have. So here beta, we talk about the scientist. Which scientist do we need to discuss? We need to talk about the Herbert Boyer and Stanley Cohen. And yeah, they were the one who developed the first RDNA as well. So they are going to tell us about it. Jaise, Again, do not forget, huh? Do not forget. Okay, not this one. My bad. Ye dekho, this one. Right? First of all, you have to love all the sessions here. Right? Just your yesterday, today, Jonathan sir will take the class 11 part 2. Your Yava sir will also take the session. Then my complete biology revision is there. So this complete biology revision, right? This complete biology revision, what will it include, beta? Here, the NCERT liners will be there. What will be there? NCERT liners. So I want you people to hit the like button. I want you people to quickly, right? I want you people to quickly join, quickly click on the notify me button. Which button do you need to click? Which button do you need to click? Notify me. Notify me. Just click there. Right? Go to this particular session and click there. Just मेरी notification on है. My notification is on. Notify me. Click there. You'll get the notification and hit the like button here. Hit the like button here. So this session is there on Sunday. That is 2 at 11 a.m. Right? 11 a.m. 11 a.m. morning where we will start. Evening may we have the predictor session as well, right? Evening may we have the predictor session as well. How can I forget to tell you about that? Ye dekho. So we will start complete class 11 biology revision. Then we will go for the most predictor paper, 180 out of 180. 180 out of 180. Okay? That is what we have to do. So ye dekho. Now I will give you the reference of Herbert Boyer and Stanley Cohen. So it is not something that I discuss extra, it is the reprinted NCERT na. Ye dekho. This one. Okay? This one. Jaise here. Nahin, here is also correct. You will see ki do we need to read the description? Nahin. Yahan, here, this is just for, right, this description is just for one discovery. So we are talking about which scientist here? Herbert Boyer. So when we see Herbert Boyer, he studied the restriction endonucleases. What he studied? He studied the restriction endonucleases. So there is no need to read all that. So by 1969, he performed studies on a couple of restriction enzymes. He performed studies on a couple of restriction enzymes of E. coli bacterium. He performed some studies on which bacteria? Escherichia coli. On which bacteria? Escherichia coli. And in that bacteria, what he studied? The restriction enzymes. What he studied, the restriction enzymes and their useful properties. So what he observed? He observed that these enzymes can cut the DNA in a particular fashion. Right? He said that these enzymes can cut the DNA strand in a particular fashion. And when we talk about that strands, when we look at that strands, they will give some sticky ends. What is the meaning of this sticky ends? Beta, these are some complementary strands. Complementary sequence, like complementary they are. Right? Just this one strand can join with another strand. Right? Right. So basically sticky ends. I'll discuss it, sticky and blunt ends. So they are saying that they are having the capability of cutting DNA strands in a particular fashion, which left what has become known as sticky ends. Which left what has become known as sticky ends on the strands. Right, so these clipped ends made pasting together the piece of DNA. So basically he studied restriction enzymes. When he studied restriction enzymes, he got to know that they can cut the DNA in a particular fashion. When I'm saying particular fashion, matlab in a proper way. And because of that, some ends were generated, they are sticky ends. Sticky ends means they are kind of complementary to each other. This is one strand, this is another. If I will bring these strands together, they can form the pairing, right? So these sticky ends, they were very useful because let's suppose if you are using the enzyme, you are cutting one DNA will generate sticky end, another DNA that will also generate sticky end, then we can join them. This is one thing. Then the next scientist and its contribution is of Stanley Cohen. Next thing that we have to discuss is of Stanley Cohen. Which scientist? Cohen. He worked on plasmids, that ringlets, which are present in the cytoplasm of bacteria. Ye dekho. He was studying the small ringlets of DNA. What that ringlets are? Plasmid, right? They were there in the cytoplasm. They are freely floating in the cytoplasm. He developed the method by which he can take out that plasmids. So he saw that plasmids can replicate independently. This is what we know about them. This is what we know about them, that these plasmids, they can replicate independently, isn't it? They are not dependent on host. They are not dependent on host. They are not dependent on host. Right? So they are freely, right? So which float about freely in the cytoplasm of certain bacterial cells, and they can replicate independently from the coding strands of DNA. They can replicate independently from the coding strands of DNA. Right, bache? Done. So Cohen had developed a method of removing these plasmids from the cell and reinserting them in another cell. So when we combine these two discoveries, one enzyme is giving us what? DNA with a particular strand, and here we have some type of DNA which we can isolate, we can put in different cell and it can replicate by its own. So when they combine these two things, right? So combining these two processes with that of DNA splicing enabled Boyer and Cohen to recombine segments of DNA in a desired configuration and to insert the DNA in bacterial cell. Right? And that bacterial cell will act like a manufacturing plant for a particular protein. Matlab that bacterial cell will give us that particular protein in which we will insert our desired gene. Okay. So these two things are the foundations of biotechnology, and the two scientists that we have discussed here is Herbert Boyer and Stanley Cohen. Okay. So when they combine these processes, right, we got our first recombinant DNA. ये देखो, जैसे बाकी see, if we want a DNA to accept outside DNA to express in a particular host, of course that DNA needs to insert near to the origin of replication, because if it will get inserted there, then only it will be able to replicate. That is a very basic thing. That is, I believe my students, they know it very well. And of course, if a piece of DNA will get incorporated into the host DNA, right, then obviously baat hai beta, if it will get incorporated into the host DNA, then of course it is obvious, right? Indeed, it is obvious that what is going to happen, my dear students? What is going to happen, my dear students? It is obvious beta, it is obvious that I just forgot about it, what I was telling you. Dekha? Sometimes it happen. So if this DNA will get incorporated into the host DNA, right, into the host cell with its origin of replication, it will also replicate. It will also make its multiple copy. It will also express itself. This is what gene cloning is. Slowly, we will read about it. Now we have to focus on the construction of first recombinant DNA. And what type of DNA it is? It is an artificial recombinant DNA. Right? It is the first construction of an artificial recombinant DNA. Artificial recombinant DNA. Right, beta? So when it is the construction of the first recombinant DNA, right, how is it possible? A gene which is encoding resistance with a native plasmid. Matlab, we will pick up a gene, right, which is having antibiotic resistance. Now we need a plasmid, because this plasmid here, right, we need a plasmid because this plasmid here will act like what? It will act like a gene taxi. This plasmid here will act like what? Will act like a gene taxi. It will act like a gene taxi, right? It will act like a gene taxi. So this plasmid, it is autonomously replicating circular extra chromosomal DNA. It is autonomously, matlab it is self-replicating, right? Extra chromosomal DNA. It is self-replicating extra chromosomal DNA, right? 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 Okay? So, the gene is for antibiotic resistance. The gene is for antibiotic resistance. It is for antibiotic resistance. Right, beta? Right, beta? And the plasmid is of Salmonella typhimurium. Right? This question can come in the paper. So Stanley Cohen and Herbert Boyer, you know that they accomplished this feat in 1972. Why? Because they isolated the antibiotic resistant gene by cutting out a piece of DNA from plasmid, and you know that it is this gene is for antibiotic resistant. Now at particular location they made the cut, right, and you know that that is possible with the help of restriction enzymes. How is it possible? With the help of restriction enzymes. We have already discussed that. Now this plasmid is acting like the gene taxi. It is also known as vector. Jaise vectors, right? Your insect vectors, they spread the disease. So this vector will help to pass the desired gene to the host. So this is what they have done. So what they did from Salmonella typhimurium, they took the plasmid, they made the cut with the help of restriction endonuclease. Now they want to attach it with the antibiotic resistant gene. Antibiotic resistant gene, right? So they will insert that they link that antibiotic resistant gene with the plasmid vector, and then will finally join it with DNA ligase, right? So again I'm telling you, when you are talking about the DNA ligase, it is the molecular glue, beta. It is the molecular glue, right? What's that? It is the molecular glue, right? It helps in joining, beta, sugar phosphate backbone. How will it help? It helps in joining sugar phosphate backbone. Is that clear? Is that clear? So it will act as a, then the new combination of this replicating DNA in vitro will form, right? And it is known as recombinant DNA. It is known as recombinant DNA. What is it? It is known as recombinant DNA. So when this DNA is transferred into E. coli, which is closely related to Salmonella, right? So in E. coli, this plasmid will replicate, bache, will make its multiple copies. So this ability of multiplying, of getting multiple copies of antibiotic resistant gene is called cloning. Is called cloning, right? Is called cloning, right? Done. It is called cloning. Okay? So this is what we have to remember. So gene antibiotic resistant plasmid was taken from Salmonella typhimurium, and then they inserted it in E. coli. That's it. Right? That's it. This is what you need to know. Okay, this is what you need to know. So these are the 3 steps here. Sometimes step-based questions are also there. So what you have to do first, beta? Identification of DNA with desirable genes. Identification of DNA with a desirable gene. Identification of DNA with a desirable gene. Then, after identifying the desirable gene, you have to introduce that identified DNA into the host. Then, you have to maintain that host, right? That DNA in the host, and we have to make sure it passes it to its progeny. So, we have to start the tools of biotechnology. Let's get started. So, up to this part, any doubt? Dekho, tools wala part is important, right? Then after that tools wala part, we don't have much here. We don't have much here. We'll quickly finish tools. We'll quickly finish restriction endonuclease, ECR and all, PCR and all. Sorry, electrophoresis and PCR. Then we'll go to the application part. Okay? We'll go to the application part. So it's 1. Let's have a break till 1:10. Okay, 10 minutes का break. Hi everyone, I hope I'm perfectly audible to all of you. Okay, so now the next thing is the tools. Okay, the tools of RDNA technology. So when we talk about the tools here, we will start with the enzymes, right? What are we going to start with? We are going to start with the enzymes here. Like this is the first tool. Not the only tool, but yeah, this is the first tool that we have to start with, right? And what is it? It is the enzyme. So when you talk about the enzymes, firstly beta, we will talk about the lysing enzymes here. Which enzymes are we going to talk about? Lysing enzymes here. So lysing means breakdown. Okay, let's suppose if you need your gene of interest, your DNA from bacterial cell, then we need lysozyme that will help to digest that cell wall of bacterial cell. If it is a bit of fungal cell, then chitinase is required. Chitinase is required. If there is a plant cell, then cellulose and pectinase is required. Cellulase and pectinase is required. Is that clear? Right? That is what you have to keep in your mind. After that, we have cleaving enzyme that are going to cleave DNA, right? So basically here, you are talking about the nucleases. You are talking about the nucleases. So now when you talk about the nucleases, so we have exonucleases and we have endonucleases. We have exonucleases and we have endonucleases. So exo, here you are talking about the terminal ends. And when you are using the word nucleases, you are digesting the nucleic acid and terminal ends. Ends you are breaking. Just let's suppose this is the DNA. Let's suppose this is the DNA. So here in this DNA, right, you are trying to cut the ends. You are trying to break the ends. When it is the endonucleases, right, it will make the cut other than ends. Okay, and here what we have to think about, where do we need to be specific? We need to be where do we need to focus specifically? We need to focus on the restriction endonucleases here, right? Where do we need to focus here, beta? We need to focus on restriction endonucleases here. Where do we need to focus? We need to focus on restriction endonucleases. Any doubt here? Yes, any doubt here? We need to focus on what? We need to focus on restriction endonucleases here. Is that clear? Is that clear? So you know that it is also known as molecular scissor or chemical scalpel. Molecular scissor or chemical scalpel. Now when you talk about this particular enzyme, beta, this restriction endonuclease, it recognizes some particular sequences known as restriction site and cloning site. Known as what? Known as restriction site or cloning site. Right? Restriction site, cloning site, and also known as recognition site. Recognition site. So restriction site, cloning site, and it is also known as recognition site that they are going to recognize. Okay? And then they will make the cut here. So is it directly going to recognize that? No. Firstly, it will inspect the length of DNA. Firstly, it will inspect the length of DNA. Here they are talking about the restriction endonucleases. We'll come to that part also. But right now the portion that I'm discussing right now is this. Restriction enzymes, they are belonging to a larger class of enzymes called nucleases. I told you exo and endo. It will make the cuts at the ends. It will make the cut inside. Okay? And endonuclease, when you are talking about, right, in the case of that endonuclease, beta, it can cleave one strand of DNA, even both. Right? It can make the cut here in the one strand of DNA or both the strands, and If we get that desirable fragment here, we can use it for the genetic engineering. Okay, now exonuclease ends done. So this is what I'm telling you, that when you talk about the restriction enzymes here, right, these restriction endonucleases, they will inspect the length of DNA sequence. What will they do first? They will check or they will inspect the length of DNA sequence. So once it finds its specific recognition sequence, it will bind to the DNA and cut each of the two strands of double helix at a specific point, right? So basically what are we saying? We are saying that when we talk about the restriction endonuclease, it will first inspect the length. That is also a PYQ. Right, that is also a PYQ. It firstly inspect the length. It firstly check the length of the DNA sequence. Once it gets that specific recognition sequence, that particular recognition sequence, it will bind there, it will cut. What is it going to break? It will break sugar phosphate backbone. जैसे ligase joins the sugar phosphate backbone, it will break sugar phosphate's backbone. Ligase joins sugar phosphate backbone, it breaks sugar phosphate backbone. That is what we have to discuss. Okay, right? That is what we have to discuss. Now here they have mentioned the word palindromic sequences. जैसे ये देखो, right? Here I just told you that what is going to happen, it recognizes some particular sequence known as restriction site, clonic site or recognition sites, right बच्चे? So what is going to happen? So this is what we have, है ना? This is what they are going to recognize. Now when it comes to the sequence, it will be palindromic sequences that these restriction enzymes, they recognize. Now when we say palindromic sequences, just let's suppose a word mom. If you are reading it from this direction, spellings are same. If you are reading it in this direction, spellings are same. Same way radar. This side, same. Same way mal-ilm. When you read the spellings from this side to this side, this side to this side, it's all same. Now, here I'll give you the example of one restriction endonuclease, EcoR1. So this EcoR1, it recognizes a sequence GAATTC. I call it as Gattus. So here also GAATTC. So here you have to remember the network 5G, 3G, right? 5G, 3G. That is how 5G, right? Just say we are currently having 5G, no? So remember 5G. It means 5 dash end will have G. 5 dash end will have G. 5 dash end will have G. So it is Gattus. That is GAATTC. GAATTC. What's that? GAATTC. Am I clear here? What's that, people? It is GAATTC. and C. It is GAATT&C. So that is something you need to keep in your mind. Okay. GAATTC. So now when you will see the sequence, here देखो. So if you are reading in this direction to this direction, sequence is same. This direction to this direction, sequence is same. Right? So remember 5 dash end will have G. Right? 5 dash end will have G. That's it. So basically it will break sugar phosphate backbone. It is going to break sugar phosphate backbone. The sugar phosphate backbone in between G and A. So because of that, what's going to happen? This will get separate from this, because what we have done, we have hydrolyzed this sugar phosphate backbone in between G and A, If here on this strand, we are breaking the bond in between G and A, here also you have to do the same, right? And when you are saying palindromic sequence, it's not mandatory that it will always be like that. Sometimes it can also be, just suppose random sequence ATCG, here ATCG is written. Right? So in this direction also, same. Sometimes that type of palindromic sequences are also there. Right? Sometimes that type of palindromic sequences are also there. Is that clear? Done. So now we have to hydrolyze the bond in between G and A, sugar phosphate backbone in between G and A, in between G and A. So see, now look at these strands here. Beta, if I will bring them closer, they are complementary to each other. You can see that. If I will bring them closer, they are complementary to each other. They can again form hydrogen bond with each other, and DNA ligase can join their sugar phosphate backbone. They can still form hydrogen bond with each other, and DNA ligase can join their sugar phosphate backbone. So these type of ends, these complementary overhangs, they are actually the sticky ends that we were talking about. They are actually the sticky ends that we were talking about. So your restriction endonuclease, it can give us two types of ends, sticky bhi and blunt bhi. And these sticky ends are very useful for genetic engineering, and we have to convert these blunt ends into the sticky one if we want to use them in the restriction, this in the genetic engineering. Okay, in the genetic engineering, if we have to use it, number one. Now what is number two here? This is number one. What is number two here? The number two here is very simple, beta. There are three types of restriction endonuclease. Restriction endonuclease 1, restriction endonuclease 2, restriction endonuclease 3. But it is the 2, right, that is used in genetic engineering. Why? Because it can inspect the length of DNA and it can cut the DNA strand. Right? It can inspect that length and even it can cut. The rest too, they can just inspect the length of DNA, but they cannot make the cut. Right? They cannot make the cut. Is that clear? Right? So that's what we have to remember. Now here they are talking about nothing but rather they are talking about your restriction modification system. Right? What are they talking about here? Restriction modification system. What is this restriction modification system? Beta, it was discovered in E. coli. Your this restriction modification system, right? Where do we study this restriction modification system? Of course, right, in 1963, right, it was basically discovered in E. coli. When? In 1963. What happened then? A bacteriophage, that is a double-stranded DNA virus. It was trying to infect E. coli, Escherichia coli, right? So two enzymes were discovered by then. Two enzymes were discovered by then, right? So one was your restriction enzyme, one was your modification enzyme, right? Right? So one was your what? One was your restriction enzyme and another one is modification enzyme. One is restriction, another one is modification enzyme. Okay, restriction and modification. Now, actually these two enzymes, they restricted the growth of this bacteriophage in the E. coli. They say it's simple. Your restriction enzyme, what will it do? When bacteriophage will insert its DNA into E. coli, right? So this enzyme will make a cut, right? It will recognize a specific sequence. It will make a cut, right? Number one. Now, so this will not allow this bacteriophage to grow within the E. coli. This will not allow this bacteriophage to grow within the E. coli. That is how the restriction enzymes will work. That is how the restriction enzymes will work. Now, what about the modification enzymes? Now, of course, let's suppose if this is the E. coli. In the E. coli, you will have that restriction enzyme. In the E. coli, you will have that restriction enzyme. But why that restriction enzyme is not restricting or cutting the E. coli's own DNA? Because it is having these modification enzymes here in the sequence of E. coli's, in E. coli's DNA, what these modification enzymes will do? What these modification enzymes will do at certain bases, right? At certain bases. Okay, there are some particular bases where they will add methyl group. What will they do? Where they will add methyl group. So because they will add methyl group there in the sequences at certain bases there, so restriction enzyme will not be able to recognize this and will not be able to digest it. That's it. Will not be able to recognize it or digest it. Is that clear? Done? Done? Okay. So, first restriction endonuclease discovered was your Hind2. And beta, it gives the blunt ends. Right here itself, I'm mentioning it. It is Hind2. It gives what type of end? Blunt ends. If you ever get confused, yaad rakhna Hind2, A, B, second letter. And if Hind3 is written, then definitely it will give you the sticky ends. Right? Hind2 is for blunt ends, Hind3 is for sticky ends. So, of course, it will also recognize special DNA nucleotide sequence. Right? So, if they are saying ki whose functioning dependent on a specific DNA nucleotide sequence, it was isolated and it was characterized 5 years later. It was isolated and it was characterized 5 years later. So here, right, obviously the sequence that it will recognize will have 6 base pair. How many base pairs will be there, beta? 6. So it is nothing, it is your GTCGAC. I'll write here GTCGAC. GTCGAC. This is what, right? This is what Hind2 will recognize. GTCGAC, right? GTCGAC. What is it? GTCGAC. GTCGAC. That will be recognized. That will be recognized by Hind2. Now, they will make the cut here in between, matlab in the center, in the middle. So of course, if they will make cut like this, so what will you get? You will get GTC, CAG here, GAC, CTG here. So of course, these are the blunt ends. These are not the sticky ends, right? They don't have any complementary overhang here. They do not have any complementary overhang here. That is the point. Okay. So now we know more than 900 restriction enzymes, right? They are isolated from 230 different strains of bacteria, right? So this is what we have. So now when it comes to their nomenclature, remember, like first letter of the name, it is coming from the genus. The first letter is coming from the genus. Second two is coming from the species. Please remember, first letter is coming from the genus and that has to be in capitals. Second is the next two are coming from species. Then prokaryotic cell from which they were isolated. Just eco R1 mein, this R is basically telling us about RY13, right? And this 1 is telling us about the number of enzyme that has been isolated from that particular strain, right? So that is the meaning here. Okay, that is the meaning here. Is that clear? So this Roman number following the names indicate the order in which the enzymes were isolated from that strain of bacteria. In which enzymes were isolated from that strain of bacteria. Is that clear? Right? Is that clear? Chaliye. Next part. So here you can see your eco R1 is making the cut, right? These are the sticky ends. So same restriction enzyme or endonuclease will use to make cut in host DNA, yeah, desired DNA, to make cut in host DNA or in plasmid. Same. Why same? Because then they will give us the same complementary overhangs. Then they will give us the same complementary overhangs. So that is how it is going to be. Is that clear? That is how it is going to be. Is that clear? Is that clear? So accordingly, cuts will be there and then finally they will be joined. So yeah, diagram based questions are also there in paper. Diagram based questions. Diagram based questions are also there in paper. So here it is that transformation. When you will add, just a ligase will join the foreign DNA to plasmid. So ligase, it will help in joining sugar phosphate backbone. It will help in joining sugar phosphate backbone, right? It will help in joining sugar phosphate backbone. So ligase will join the foreign DNA to plasmid. So recombinant DNA molecule, the transformation will occur, the cells will divide, and this is how the cloning will occur. Any doubt here? That is how the cloning will occur. Any doubt here? Yes, sure. Are you sure? Done? Chaliye. So after this, right? After this, what's going to happen? Ultimately, you have made a cut, right? You have made a cut. Okay? Alkaline phosphatases are also used. They'll remove the phosphate group from the 5' end, right? So and that is basically to stop the self-ligation, but that is extra. Now the next thing that we need to understand is gel electrophoresis. Gel electrophoresis. And when you are talking about this, here you discuss about two things: page age. Page or age. When it is page, you are talking about polyacrylamide gel electrophoresis. When it is age, you are talking about agarose gel electrophoresis. Here you are talking about polyacrylamide gel electrophoresis, agarose. So now here in our syllabus you talk about the agarose, and this agarose is actually a polysaccharide. And this polysaccharide, my dear students, it is obtained from a plant, of course, right? Now, electrophoresis. What exactly is this electrophoresis? Electricity. So basically, under the influence of electricity, the charged particles will be separated on the basis of, that is important, on the basis of size. Remember, not on charge. Let me write it so that you don't make mistake here. So what are they saying? They are saying that under the influence of electricity, under what? Under the influence of electricity, my dear people, under the influence of electricity, the charged particles will be separated on the basis of size, but not on the basis of charge. Right? So here in electrophoresis, you will see DNA. DNA is negatively charged. So under the influence of electricity, it will find its way towards what? Towards what? Anode. Because anode will be positively charged. Okay? Here the cathode is negatively charged. So DNA sample will be loaded after staining with ETBR. Then beta, this agarose, right? Firstly, it will be in the powder form. We have to place it in the hot water. It will get that jelly-like consistency. We'll allow it to cool. And as per the concentration, the pore size will be there, right? So this is what we need to discuss. So basically, what is happening? You have the restriction enzyme. It's not like that you can clearly see DNA. You'll hold the DNA in the hand. Then you are going to make the cuts. Of course not. That is not the scenario. That is not the case. It's like you are mixing one solution with another with third one, and then you will see certain results. That is it. Then you will see certain results. That is it. That is it that we have to do here. So here, basically the plasmid is also, right, treated with the same restriction enzyme like the host DNA. So plasmid is circular. Always remember this thing, it's a circular. You are making a cut. You are making a cut in a circle. You'll get one fragment. If it is a linear DNA, if in the linear DNA you are making the cut, you will get two fragments. So even for the plasmid, even for the eukaryotic host, you will use same restriction enzymes. You will obtain the fragments. Then you will try to join them. What will you do? You will try to join them. Okay, ji? But firstly, you have to separate these fragments. Then you will try to join them. Okay, ji? So the cutting of DNA by restriction endonuclease, as I told you, they'll give you the fragments. What will they give you? They'll give you the fragments. So these fragments will be separated with the help of gel electrophoresis, my dear people. So DNA fragments, you know that they are negatively charged molecules. They can be separated by forcing them move towards the anode. So do not get confused here. DNA is negatively charged and here the anode is positively charged, beta. So under the influence of electricity, it will move. Okay. So agarose, I told you, it's a polysaccharide extracted from seaweed. So DNA fragments will resolve according to their size. So the one which is smaller will reach quickly to the end. The one which is larger will take time, right? Because in the agarose gel, as per their concentration, the pore size, it is the pore size that is different. So that is what you have to keep in your mind. That is what you have to keep in your mind. Is that clear? Is that clear? So these are the wells. These are the wells where the sample will be loaded. These are the wells where the sample will be loaded. And before loading it, we have to color it. Right? Matlab we have to stain it. Otherwise, we will not be able to see that. So, beta ETBR, ethidium bromide will be used. Right? That is your NTA's favorite question. I don't know why, but they love this question. Right? So, ETBR, this stain is used. NTA's favorite question. This stain is used. It is NTA's favorite question. So, separated DNA fragments can be visualized only after staining the DNA with the compound known as ethidium bromide. Then we have to give it the exposure of UV radiation. So remember, it is the UV radiation that we are talking here, right, bache, that we are talking here. So you can see bright orange color bands. You can see bright orange color bands. ठीक है बेटा, when we expose them to the UV light. So these separated bands, they are cut from the agarose gel and they are extracted from gel piece. Matlab they, obviously the fragments are separated. These are the gels. You will cut, right? You will cut these, right? You will cut them, right? You will cut these fragments. Now you have to separate it, right? You want to separate it. You're like, I need them back. So because you need, you have to separate it, it is known as elution. So the DNA fragments purified in this way, they are used to form the recombinant DNA. Any doubt here? Yes. Anybody having any doubt? Anybody having any doubt? Please let me know, guys. Yes, beta. Done? Sure. So question can come from this part. So illusion, you need to be clear in your, you have to clear it in your mind. So this is that tray. In the wells, the sample will be loaded, but after staining. So it is attached to the source of electricity, because under the influence of electricity, the charged particles will move, and that's what we have here, right? That's what we have here. Is that clear? Is that clear? Are you sure? So the next topic that we need to discuss is this, right? The next topic that we need to discuss is what? Is the vectors, right? Is the vectors. That why they are serving as a gene taxi. And beta, there are different types of vector, even if you will see. The most commonly used one is indeed, it is plasmid. It is your bacteriophage. It is plasmid. It is your bacteriophage, the most commonly used one. But there are many other, right? There are many other vectors as well, right? There are many other vectors as well. But this is something that you can keep in your mind, right? That these are the thing. So cosmids, right? And bacterial artificial chromosome, everything is there. Now, if there is the word shuttle vector, if there is the word shuttle vector, it can be used in both. It can be used in both prokaryotes and eukaryotes. Prokaryotes and eukaryotes. The shuttle vector. So shuttle vector mein you will talk about TI, tumor inducing plasmid. What do we discuss there? TI plasmid. Tumor inducing plasmid there. So the very important thing for a cloning vector is the origin of replication. Let's discuss it quickly. So when you talk about the plasmid and bacteriophage, so I told you, right? Jaise bacteriophage hai, it is a virus. It will use the host machinery, it will multiply and make its multiple copy. Plasmid is also capable of self-replication. It can self-replicate. So it has having the ability to replicate within bacterial cells independent of the control of chromosomal DNA. So now what is happening? These bacteriophages, they will use, because of their high copy number per cell, because of their high copy number. Basically, copy number is a thing. Let it be. Firstly, I'll start with the copy number. As the name is suggesting, copy number. Matlab number of copies, या number of plasmids in a particular cell. Like let's suppose if there is a plasmid, let's suppose it can divide 2 times, yeah 3 times. Accordingly, its copies will be there in the bacterial cell. That is the copy number of that bacteria. That is the copy number of that bacteria. Right? That is the copy number of that bacteria. Are you getting my point? Are you getting my point? So they have, there are different types of vectors. Their copy number also varies. If we want, if we want a gene where we need the multiple copies, so we will go for the vector having what? We will go for the vectors having high copy number, right? So this is what we have to see here. So they are saying, so bacteriophage, because of their high copy number per cell, right? So they are saying they have very high copy number within the bacterial cell. Some plasmid, beta, they can have 1 or 2 copies per cell, but some can have 15 to 100 copies per cell. The numbers can even be higher. So if we are able to link an alien piece of DNA with bacteriophage or plasmid DNA, if we are able to link the alien piece of DNA with bacteriophage or plasmid DNA, with bacteriophage or plasmid DNA, we can multiply its number equal to the copy number of plasmid, right? Right? So whatever is the copy number of that particular vector in a cell, we will get that number of copies of our desired gene. If you need more, you have to go for the plasmid with high copy number. So this copy number thing, it also depends upon origin of replication. This copy number thing, yeah, it also depends upon origin of replication. Am I clear? It depends upon origin of replication as well. ठीक है बेटा? ठीक है? Okay, so the vectors used at present are engineered in such a way that they help easy linking of foreign DNA and selection of recombinant from non-recombinant. So here we will talk about the transformants as well. ठीक है बेटा? So origin of replication is very, very, very important. See, plasmid can self-replicate. They can make their own copies. So that is why they are used as vectors. So if something is vector, for sure it should be capable of replicating on its own, right? So as I said, right, the copy number of a plasmid, yeah, vector depends upon the origin of replication as well. So if we want to recover many copies, this is done and dusted. Then comes the selectable marker. Matlab, see, we need origin of replication, but along with that we need one more gene, right, which can give a unique property to our plasmid. Matlab, one origin of replication is required, right, but we have to consider some gene, right, which can give unique property to my particular, vector, right? Yeah, my particular plasmid. Just for an example, if this particular plasmid is having antibiotic resistant gene, right? Or let's suppose it is having antibiotic resistance against A. Let's suppose it is having antibiotic resistance against A, right? So this will help me to figure out transformants from non-transformants. Right? What is the meaning of that? But as I said, you cannot hold a cell in your hand and then you can see, yeah, I'll put the DNA there, yeah, I'll put this there, yeah, I'll put that there. That is not the case, right? That is not the case. Genuinely, that is not the case. Okay? That is not the case. So what is the case? Let's suppose this is a cell, right? Basically, you will either incubate them or you will mix some solutions. So what will you do? Let's suppose you are trying to give them the plasmids. 2 cells have picked up, but 2 cells they didn't. How can we check it? Of course, the one that has picked up a recombinant plasmid, that is the transformant. That is the transformant. And the one which didn't, that is the non-transformant. But how can I check it? It's simple. All these bacterial cells, let's suppose they were not showing the antibiotic resistance against A. Let's suppose they were not showing the antibiotic resistance against A. They were not showing what? They were not showing the antibiotic resistance against A, right? They were not showing that. So because they were not showing the resistance, right, the antibiotic resistance against A, now I have inserted the plasmid and I am saying my plasmid is having antibiotic resistance against A. So if these cells, if they have picked up my plasmid, they need to survive there. They need to survive in the culture having antibiotic A. So let's suppose this is the culture. Here in this culture, I have added antibiotic A. What have I added? I have added antibiotic A. Now I am growing my bacteria there. Some of them they died, but some of them they survived. So the one that are survived, they are the transformants because they have picked up my plasmid, and that plasmid is having this particular gene, right, which is providing them the antibiotic resistance. So that is how I got to know about my transformants, and this is the role of selectable marker here. So whenever you talk about any of the vector, any of the plasmid, we have to pick up a gene. We have to pick up a gene which will provide it the unique phenotypic character. So these days, because it is very cumbersome task to, grow antibiotic cultures against and again and again. So insertional inactivation is going on these days. That is preferred to check transformants from non-transformant. Just for an example, let's suppose this is your plasmid. Okay? This is your plasmid. Here you have the cloning site, the recognition site, which the restriction endonuclease will recognize. Now here itself you have a gene. You have a gene for something, right? Now I have used this restriction endonuclease. Of course, it will make the cut beta. So my gene will not be functional, right? Why? Because I have made a cut and I have inserted a gene of interest here. So I have inserted a new gene due to which my gene sequence was interrupted. It is not able to express itself. This is the insertional inactivation. So here in this case, right, we discuss about the lac Z gene, right? You know that it gives what? It gives beta galactosidase. It gives what? It gives beta galactosidase. Okay, so lac Z gene is there, it gives what? Beta galactosidase. So let's suppose if this beta galactosidase is present, right, then blue colonies will be there because it acts on chromogenic substrate. If it is absent, white colonies. So sometimes with the help of such kind of things, we can also check the transformants from the non-transformants. So that is how the selectable markers differ, work. Okay? So catkin. Now, in the case of E. coli, right, the world famous bacteria, right, now in the case of E. coli, that is your Escherichia coli, what do you have, beta? What do you have? You have cat-K. What do you have here? You have cat-K. What do you have here? You have cat-K, right, cat-K. So cat-K matlab C. C matlab chloramphenicol. A matlab ampicillin. T means tetracycline. Or K means kanamycin. So, kanamycin. So, E. coli is cat-K. E. coli is cat-K, right? That is what I'm trying to say. E. coli is what? E. coli is cat-K. E. coli is what, beta? E. coli is cat-K. So, what is the meaning of cat-K? What is the meaning of cat-K? So, it is chloramphenicol, ampicillin, tetracycline and kanamycin. So, your E. coli, it basically, right, ये देखो, normally the genes encoding resistant to these antibiotics are considered useful markers for E. coli because E. coli do not carry resistance against them, right? Against them. So in the case of E. coli, any plasmid which is having the resistance against chloramphenicol, ampicillin, tetracycline, kanamycin, it can be used. Okay, it can be used. ठीक है बेटा. Now cloning site I already told you. ठीक है. So PVR322. So here it is important. You know that P stands for anybody here, plasmid. BR. Remember this full form. So first of all, this topic is very important, right? This topic is very important. Do check it, right? Do revise this particular thing. So PBR322, let's talk about this part. So it is basically the artificial, right? It is basically the artificially synthesize the plasmid. So P for plasmid, B for Bolivar Rodriguez. So that is what we have. That is what we have. PBR322, right? So if you will see, it is having ampicillin resistance, tetracycline resistance, selectable marker. You have ROP here. This is the, they are coding for the proteins involved in replication of plasmid. Now, if you will see this, this is the origin of replication, which is much needed. I already told you about the cloning site. I already told you that cloning site is something very important. It is also known as a restriction site. It is also known as recognition site. It is the site that will be recognized by. It is the site that will be recognized by what? It is the site that will be recognized by what? That will be recognized by your restriction endonucleases. So what kind of information do we need to understand? See, 1, 2, 3, 4, 5, 6, 7, 8. Total 8 recognition sites are there, right? Now, let's suppose if I have used the enzyme BAMH1. Look, write down your homework. Whatever enzymes are written here, you should know which one is having sticky ends, which one is having blunt ends, which one will generate sticky ends, which one will generate blunt ends. That is your homework, right? You people are going to tell me this. Okay, you people are going to tell me this, that which one is having sticky ends and which one is having blunt ends. Will it be done? Yes. So here 8 restriction enzymes are there. So beta, see, let's suppose if I will use this enzyme will make the cut here, will insert its gene of interest here. So obviously your tetracycline 1, 2, 3, 4, 5, 6, 7, 8. Your tetracycline, this tetracycline gene will become inactive because you have used BAMH1 and then you are inserting your gene of interest there. So your tetracycline will not be able to show. That is, this gene will not make the functional protein. Insertional inactivation. Okay? So, but still, ampicillin resistance is there. This gene is there to act like a selectable marker. Now, let's suppose if out of these two, if I will use any one restriction enzyme, then this gene will not express. Then I can go for another one. So, there is a line here. There is a line here, right? That the vector needs to have very few, preferably single, right, recognition sites for the commonly used restriction enzymes. Matlab, whatever restriction enzymes we use, whatever are the commonly used restriction enzymes, right, one vector should have one site for that. Otherwise, there will be the formation of multiple fragments that will not support in the recombinant DNA formation. So that is what I am telling you. You can read this part, right, that basically different cultures are there. That's it. Baki insertional inactivation also I have discussed with you all, ki rather than this ki you focus on, antibiotic resistant gene as selectable marker, then you have to make different cultures for checking transformant and non-transformant. You simply go for the insertional inactivation. Right, just say I gave you the example of beta galactosidase, right? It is obtained from your LAC-Z gene. Just now we discussed it. So it acts on what? It acts on the chromogenic substrate, right? So it will give blue colored colonies if the plasmid in the bacteria does not have any. So basically this result. So basically we need this enzyme. So this enzyme, you know that it produces the color if it is present, if its substrate is present, right? So in case of recombinant DNA, you know that this enzyme is insertional inactivation of this enzyme is there, right bache, right bache. So if our bacteria will give the blue color, matlab that is not showing insertional inactivation, matlab that is not showing me the transformation. So that is how we select them. That's it, nothing else. Baki beta, there are different types of vectors, right? Different types of vectors. Even the viruses also act like vector. But here we will discuss about one example of Agrobacterium tumefaciens, right? One example of Agrobacterium tumefaciens. It is also known as soil-borne bacteria. It is also known as natural engineer. It is known as what? It is also known as natural engineer, without degree wala engineer. Okay? It doesn't need 4-year degree to be an engineer. Okay? Now this particular bacteria, in the case of dicots, bache, it cause crown gall tumor. Right? It calls crown gall tumor. ठीक है. So this is plasmid here. Now this bacteria, this plasmid is Ti plasmid. What is the meaning of Ti plasmid? Ti is tumor inducing. Ti is tumor inducing plasmid. Actually this plasmid is having DNA. Which DNA beta? This plasmid is having a tDNA. This plasmid with the help, matlab this bacteria with the help of this tDNA, it will infect a normal cell. Its tDNA will be inserted here in that cell and will convert that normal cell into the cancerous one, right? Into the cancerous one. So what is going to happen? We are talking about the bacteria here, Agrobacterium tumefaciens. It's a soil-borne bacteria. It is also known as natural engineer. In dicots, it will cause crown gall tumor. What will it cause? Crown gall tumor. Now here you have the plasmid. This plasmid is the extra chromosomal DNA beta. It is also known as Ti plasmid. And I told you it is a shuttle vector. Can be used in prokaryotes as well as in eukaryotes. Can be used in prokaryotes as well as in eukaryotes. Now, what is the next point here? What is the next point here? Next point here is that you have a tDNA here. Now, this particular bacteria, with the help of its Ti plasmid, with the help of its tDNA, it will convert a normal cell into the cancerous one. It is its natural ability. It is its natural capability. It is its natural capability. It is its natural ability. Now scientists will exploit that. They check this plasmid. They are like that tDNA is having some restriction sites. They check that here in this sequence which causes the tumor in a normal cell, the restriction sites are present. So scientists use that particular enzyme, right? They made a cut there. They inserted their gene of interest there. So now next time, whenever this bacteria will try to infect any plant, so it will not infect that plant, but it will give our desired protein to that plant. It will give that desired protein to that plant. Is that clear? Is that clear? Are you sure? Are you sure? So that is what we have here, right? So baki tumko pata hai, even the retroviruses beta, they are also used, right? We will make them harmless. We will also use their natural ability because they convert the normal cell into the cancerous one. So we will make them harmless, and then that is how they can be used. So this is all about the vectors in plants and animals. Now the next point is that we have to make the host competent, right? What we have to do? We have to make the host competent. So if we have to make the host competent, competent means the ability of a cell to accept the foreign DNA, the ability of the cell to accept the foreign DNA. Are you getting my point? Are you getting my point? Right? So DNA is hydrophilic molecule, right? DNA is hydrophilic molecule. Hydrophilic means water loving, or can I say that lipophobic then? है ना? So it cannot pass through the cell membrane directly. So we have to create some pores there if we want our host cell to accept the recombinant plasmid, yeah, to accept the DNA, right? To accept the DNA. So we have to make our cell competent. So very first thing is beta the treatment with divalent cation. The treatment with divalent cation, right? So calcium ion, it can be magnesium, it can be calcium. So we have to treat them. When we will treat them, some pores will be created, some temporary pores will be created in the outer covering, right? In the outer covering, this is the first step. This is the first step. So basically we have to increase the efficiency with which DNA can enter so that the pores will be created there in the cell wall. Now in the second step, what's going to happen beta? Right, we have to incubate that cells with rDNA on ice. We have to incubate that cells on rDNA with ice. Is that clear? Right? Is that clear? Done. So we have to incubate the cells. We have to incubate the cells with rDNA on ice, right? Second step. Then after that again beta, we'll give it heat shock. And then again we will put that on ice. So imagine, I'll do what? Firstly, I'll treat them with divalent cation. Because of that, my cell will show some pores, right? Then I will incubate that plasmid with it on ice, right? Then we will give it a heat shock. We will give it a heat shock at 42 degrees Celsius, right? Then again, On ice, we have to incubate it. So we will force our cell ki you have to accept the DNA. You have to accept the DNA. We will not leave you any choice. Is that clear? Is that clear? Any doubt here? Any doubt here? Done, beta. So other than that, you know that there are other ways, vectorless way, vectorless way to transfer the gene. जैसे micro injections है बेटा, DNA is directly injected into the nucleus of animal cell. जैसे in reproductive health we have studied ICSI, right? Intracytoplasmic sperm injection. Directly you are inserting the nucleus there. So that is the point. ठीक है, बाकी ballistic gun or gene gun commonly used in plants beta, right? So basically we will, right, do bombardment with high velocity microparticles of gold or tungsten, and that is having our DNA coiled on it, so it will further enter in the plant cell, right बच्चे? So disarmed pathogens जैसे कि retrovirus, जैसे कि that TI plasmid, right, we have converted them, right, we have converted them into the harmless one, so we can use that as well. Is that clear? We can use that as well. So ultimately, you know that we have to club all the steps together, and we will be able to get what? We will be able to get our desired gene. Okay? So from this part, what you have to learn? Rest everything we have discussed ki you need the nucleic acid. Okay? Nucleic acid, or you know that it can, majority of organism it is DNA. So we have to cut this DNA with the restriction enzymes. So if we have to cut this DNA with the restriction enzyme, we need to get this DNA in the pure state. We need it in pure form, right? We need it in pure form. We need it in pure state. Keep it in your mind. So for that, what we have to do? So you know that DNA is present within the covering. So firstly, we have to break the cell. We have to open the cell beta, and then after that, RNA, proteins, polysaccharides and lipids, right? RNA, proteins, polysaccharides and also the lipids, they are present there. So of course we have to add the suitable enzymes that will help in their digestion. जैसे कि suppose lysozyme will break the bacterial cell wall, cellulase will break the plant cell, chitinase will break the fungus, right beta? Right, bacha, fungal cell wall, and ultimately you need it in the pure form. So you will give the suitable treatment. You will treat it with the, you know, multiple enzymes. So when it comes to the DNA, right, we have to add the chilled ethanol here, right? The chilled ethanol will give us the precipitated form of DNA. Chilled ethanol will give us this DNA in the precipitated form. So can you see this diagram? This word is not there in the theory, but it is present here. And the question will come from this part. Okay, beta, and the question will come from this part. Ki this DNA that separates out can be removed by spooling. So spooling mein what you have done? We have added the chilled ethanol there finally, right? Because we have digested everything. Now we have to make the precipitates of DNA. We have to take out that precipitates. So we have added a glass rod or a wooden stick, and then we are trying to take that DNA. This is the spooling. So do not confuse spooling with elution. Okay, so that is how, right, after the addition of chilled ethanol, you will get the precipitated form of DNA. This is important. Again, PYQ. ठीक बेटा, बाकी you have to cut it at the specific location with the help of electrophoresis. You know that we can separate the fragment. But let's suppose if there is a gene of interest, but that gene of interest is not there, right? It's like we need its multiple copies. So we can also go for beta PCR, polymerase chain reaction. So the direct question is dye here. These are the three steps, dye. These are the three steps, dye. So it is denaturation, it is annealing, and it is extension. Denaturation, annealing, and it is extension, right? So who invented this technique? Carey Mullis. Right, here in this technique, the DNA will be synthesized, like multiple copies will be formed. So here, let me tell you, no lagging strand thing will be there, right? Primers will have oligonucleotides. Matlab, these are the deoxyribonucleotides only. Primers will have oligonucleotides only. So no lagging strand, primers will have oligonucleotides here. That is something you can keep in your mind. That is something you can keep in your mind. So Carey Mullis awarded the Nobel Prize for the development of PCR. So 3 steps are there: denaturation, annealing, and extension. Okay. So denaturation, and yes, the third is enzyme. The enzyme used here is Thermophilus, Thermus aquaticus. Actually, that is the bacteria. Enzyme used is Taq polymerase here. This enzyme is obtained from Thermus या Thermophilus aquaticus, archaebacteria which can survive under extreme conditions. So what am I talking about? Denaturation, annealing, and extension. Right? No lagging strand primer will not be RNA primer. Oligonucleotide primer will be there. Enzyme used is Taq polymerase because it can survive under extreme condition. So here the temperature will be 94 degree to 96 degree Celsius. Then you have to decrease it somewhere around 40 to 60, or you can roughly say 52 degree Celsius. Right, you can roughly say 52 degrees Celsius, or here it should be somewhere in between 40 degrees Celsius to 60 degrees Celsius, somewhere let's say 52 degrees Celsius. Yahan pe it will be around 72 degrees Celsius. So basically what is happening, 3 steps are there. Right, you have the piece of DNA. You want to make multiple copies out of that. Firstly, bache, you have to increase the temperature so that you can break the hydrogen bonds. So that is why in the very first step, we will break the hydrogen bonds. We will get single strands. Now we know about this DNA. Now it's not like we need some RNA primer here. RNA primase here. No. Here what's going to happen, you will separate these 2 DNA strands. You already know about this gene. You'll order the chemically synthesized primer. These primers will join. That is annealing. So annealing may basically there is a joining. We need to join these hydrogen bonds. So that is why we need to join these hydrogen bonds. That is why the temperature of this step is lower, because we want that bonds to form. Then beta, obviously the primers are not some RNA primer, they are made up of oligonucleotides only. Now we need the enzyme that can survive under extreme conditions. So we go for Taq polymerase, because thermal stability is very important. This enzyme can work even at very high temperature. So bus, this will happen. That's it. 2 strands will form. So we have to repeat these 3 steps again and again. Right, we have to repeat these 3 steps again and again, and we will get the multiple copies, or I would say in billion of, right, we can get these copies in billions. Theek? Done? So selectable marker, recipient, recombinant DNA, that's all done. So basically we are doing it so that we can get the foreign product, bache, right? So that we can get that gene product. So the large vessel where we will grow that culture, right? If it is for the experimental purpose, we'll do it in lab. Or bioreactors, they are the large curved vessels in which we will put our culture, we will put our bacteria or whatever the genetically modified organism is. Then we will allow it some time to grow so that we can get our culture at the end, right? So that we can get what? So that we can at the end we can get our desired product. So that is why we are doing all that things, right? That is why we are putting such efforts. Are you getting my point? That is why we are putting such efforts. That is why we are doing that things. Are you getting my point? So here, what are we doing? So basically, we have to grow that, right? We have to grow that bacteria. So they are saying that when you insert a piece of alien DNA into a cloning vector, and when you transfer it into a bacterial, plant or animal cell, you want it to multiply, you want it to increase the number so that you can get your desirable product. Isn't it? You need that recombinant product, right? So small scale ke liye, in the lab for some experiments. But when it comes to the large scale, we will go for the bioreactors. So there are different types of culture. But one way is that there is a culture, you'll put the culture medium there, microorganism there, right? That's it. After some time, you will get your product. This is the batch culture. Just say, let's suppose I have started the classes. I am saying I need 30 students in my batch. That's it. Only 30. I'll complete their syllabus, then I'll go for next 30. Something like that. Then fed batch, the modified form of batch culture, where, after some time, the culture medium, sorry, the nutrient medium will be incremented, it will be added. Then comes the continuous culture that they are talking here. Okay, from one side you will keep on adding the nutrient medium, from another side you will remove the waste. Now, because of that, what's going to happen? The genetically modified organism that you are growing there, that will continuously get the food. The food is having the continuous supply. So you know that S-shaped growth, lag phase, but then, log phase, then lag phase, then stationary phase. Now this genetically modified organism is getting, right? It is getting, what do we call it? The food continuously. It is getting the food continuously, right? It is getting that food continuously. So of course it is their lag phase. It will just keep on dividing. It will give us recombinant protein. So we will take that protein also. Its quality checking will be done, right? So basically you can see bioreactor mein optimal condition, best suitable condition for achieving the desired product by providing the optimum growth condition. Chahe temperature hai, pH hai, substrate hai, salt hai, vitamin hai, oxygen hai, it will be provided. So there are two types here, beta. This is the simple stirred tank bioreactor. It is the sparged stirred tank. So here we will see the gas entrainment. We will see the surface area is increased for oxygen transfer, and these bubbles, they increase the oxygen transfer area. The question on the basis of examples, on the basis of this picture is mostly there. Okay, it is mostly there. Is that clear? Is that clear, everybody? Are you sure? Are you sure? Any doubt from this part? So this is all about the bioreactors. So this stirred tank bioreactor is usually cylindrical, or it is having the curved base. That will help in facilitating the mixing of bioreactor content. So this stirrer will facilitate even mixing and oxygen availability throughout the bioreactors. Alternatively, air can be bubbled through the reactors. So they are saying मतलब कि बायोरिएक्टर में agitator system है, oxygen delivery system, form control system, temperature and pH control system. There is a sampling ports from where we can take out the samples periodically so that we can do their quality checking, right? So that we can check it. Okay. Then comes beta downstream processing, जिसमें further you have to separate and purify the product. That's it. That's it, right? Separation and purification is downstream processing, right? If required, suitable preservatives will be added. If there is a drug, the clinical trials has to be done. So this is what downstream processing is. ठीक? बाकी बेटा biotechnology applications में, right? Here, yes, give it a reading to tissue culturing, and I would say that these examples, जैसे BT cotton, BT genes, this Melodygyne incognita, then other than that, insulin, genetically engineered insulin, very simple examples they are. You just give it a reading. Okay, that will be sufficient for you. That will be sufficient for you. Is that clear? Is that clear? So this is what we have discussed today. This is what we are going to discuss today because we are also running late. So again, I'm telling you, look at the sessions here. Sunday 11 a.m. complete 11th biology revision is there. Not like the, complete syllabus. Nahi. This time NCERT points, NCERT readings. Earlier I thought that I will ask the questions from the zoology, from the botany only, but that we have done already, right? So what will I do? I will just do one thing. I will add the NCERT lines there, right? You have to fill that up. You have to solve the questions on the basis of that, right? You have to solve the questions on the basis of that. So in this way, that line to line NCERT will be there in your mind. Line to line NCERT will be there in your mind. Trust me on that, right? Ki yes, theek hai, this is what we have in NCERT. That is what we have in NCERT. So you will be able to revise it. So make sure you like it. Make sure you click on the notify me button, right? Make sure you click on the notify me button. ठीक है? Notification में ये notify me, notification on. Okay, so quickly hit the like button now. Everybody here, hit the like button now, right? Hit the like button now and click on the notify me now. I really want to increase this number, right? quick, hit the like button. I want to see you all here in this particular session, right, bache? So complete revision will be there. After that, there will be complete 12th revision also in the same format. We will start with the ecology and sexual reproduction in flowering plants chapter. So whatever chapters we are not able to touch in today's lecture, we will start from that chapters in the next marathon. So in this way, there is a complete revision. But whatever we have completed today, that is something very, very important. That is something very important, right? That is the major part, right? That is literally the major part. So give it your time, right? Give your best here. Give it your time. Give your best here. Just keep it in your mind, okay? Just keep it in your mind that yes, we have to revise. Baki other teachers like physics marathon is there, physics sessions are there, right? We are just putting so many questions for all of you here in this, what do we say? In our video section. And if you are feeling demotivated, this is the video that you people can watch. This is the video where you guys be able to relate everything. Okay, everything. Ki theek hai, madam, you are right. These are the kind of issues we are facing. Thank you so much for talking about that. And now we are feeling comfortable and okay. Right, so you can watch this video. But as I said, guys, do not forget to join the complete biology marathon for 11th, for 12th, and even the complete syllabus marathon as well. Okay, that's the best way to practice. That is the best way to practice, and we are going to do the same. Okay, that we are going to do the same. And because I don't want my students to struggle, right? Literally, I don't want my students to struggle in that part. Ki ma'am, we are not able to revise from NCERT, right? Yeah, we are struggling with that NCERT part. We are not able to retain it, right? I don't want to hear such thing. Okay, so I'll share the PDF of this session as well, right? You can just use them for quick revision, and from the evolution part, if you have done it before, then it's fine. Otherwise, the topics that we have discussed, that is sufficient for you. I'll put the short video. Hopefully tomorrow I'll put the short video. Okay? Hopefully tomorrow I'll put the short video for the quick revision here. Don't worry about that. Okay? Don't worry about that. So any other doubt? Yes. Anybody is having any other doubt? Anybody is having any other doubt? Okay, so make sure, students, that from the genetics part, you solve all the last 5 to 6 years ke question. From the ecology part, last 10 years question, if you will solve, no need to read anything then. Just solve last 10 years PYQ. Okay, just solve last 10 years PYQ. That is literally going to be very helpful for you for this particular NEET 2025 examination. So for ecology, for genetics, at this point of time, keep revising the questions, okay? Keep revising the question. Do appear in the test as well. You know, now your benchmark test is there on 27th of April. That is this Sunday. Yes, this Sunday, there will be the benchmark test. There will be the benchmark test. That is, and that benchmark test is just like NEET examination. It will be NEET replica. It will tell you about your all India rank, right? It will literally tell you about your all India rank, that this is going to be your all India rank, right? And you'll get a chance to compete with all the aspirants around. Okay, so do give that test, your benchmark test, on 27th April, 2 p.m. Okay, chaliye. So take care. Thank you so much, guys. We'll share the PDF in the Telegram group. Thank you.